if you need to click to play. But I can click it for you. This one for you.
You don't go for now. Good. We thought it was you. We're just checking that everyone's here ready. So we'll start in 1 minute. Okay. Start. So, good morning, everybody. It's nice to see everybody, even though it's quite an empty room in such a huge audience. A room like this, it's a bit daunting. But after last night's party in New Orleans, I'm not too surprised, actually. Hopefully, more people come in during the session. So, I'm Phil Hewitt. I'm from Merck in Germany. So, I'm an investigative toxicologist. My co-chair is.
Good morning, everyone. I'm Connie Liebachen. I'm a scientist at a company in Madison, Wisconsin called STEM Farm.
Okay, so it's a pleasure to welcome you all to this session, which is Predictive Toxicology MPS for AOP, which I assume everyone knows as Adverse Outcome Pathway. And so even the slides are ready for you, Laura. So it's a pleasure to introduce the first speaker this morning. And that's Laura Sutter-Dick. And maybe introduce yourself, because I have no idea what FHNW stands for.
I'm Laura Sutter-Dick. I work for the Applied Science University, Northwestern Switzerland. That's the acronym. I realized Phil was talking in my time. So anyways, and I would like to present here this concept of AOP chip, something which is a little bit different from what many people have been working on presenting in this conference, because we're trying to put everything together, all the cells together, all the organs together, multi-organ chips and so on. And I'm trying to use the microphysiological systems to actually deconvolute specific questions. So going a little bit in the other direction. So let's see how this works. Okay, so we started the idea, the concept in general should be for AOPs, but we started with one specific example, and that's fibrosis. Why fibrosis? Well, on one hand side, I've been working in fibrosis for many years with 3D models, multicellular models, but also as a disease, is an endpoint in toxicology. Several etiologies will lead to liver fibrosis. And it's also a very well described AOP. So I think there is also an important unmet medical need. So there are not many therapies. So there are many reasons why fibrosis was, we consider fibrosis a very good example to move forward with this. So the concept in general, and I think this is just, this is just a schematic to show that you can consider a microphysiological system. There are many flavors. We've seen beautiful presentations here, but you always have a sequence of connected compartments, could be cells or tissues or organs, whatever. You have often valves interconnection, and we have, by some means, you generate some flow. So this is just a general scheme of a microphysiological system, which applies to everybody. But if you look below, there is an AOP, an adverse outcome pathway, and this is specifically the adverse outcome pathway 38, which is liver fibrosis. And you can see, if you look at the schematic, that it's also a sequence of events which lead to the adverse outcome pathway. So we have several key events here. The hepatocellular damage, then we have Kupffer cell activation, we have stellate cell activation. And each of these key events will generate a signal that will prompt the next key event in a key event relationship, and this will generate the next one, and eventually we'll have a problem. So I thought that this looks very, like it just seems like a similarity in the architecture, and I thought we should try to give it a try whether we can see that. Because the big problem with AOPs is that the key event relation, to quantify these key events, is very difficult, because if you have animal data, or even if you have a multicellular system with all the cells are mixed together, you don't really know which cell is generating which signal or is undergoing which process. So the goal was to provide a model that we can to quantify these key events and the key event relationships, and to see how these AOPs may or may not progress. In my mind, I always picture this as a kind of a domino stones, and because you can have a little bit of a change, an adaptive change that may not lead to an adverse outcome. But once you go over the threshold, point of no return, then you will have a problem. So we need a system that we can use to quantify this. So liver fibrosis is the one we chose, and I want to go into a little detail into the AOP to show you which are the main players in this. So this is a depiction of the liver, and we will see in that very nice depiction that my PhD student generated, we have the hepatocytes, we have the Kupffer cells and the inactivated stellate cells, and they're the three main cell types which are part of this AOP liver fibrosis. Once the injury starts, we have a first event, and you can see even depicted here, the hepatocyte is the first key event. The hepatocyte starts suffering, it changes, it has some toxicity, we can see some toxicity, and we can use specific cells. In this case, we use HepRG cells in 2D or 3D to represent this hepatocellular damage. That would be the first stop. The second stop are the THP1 cells that we use as a surrogate for the activation of Kupffer cells, which is what happened here. Once the hepatocellular damage is sufficient, Kupffer cells will be activated, and we can measure that using THP1s. And the third cell type we use is the stellate cell. And the stellate cell is my favorite. We study a lot in the stellate cells, but it's also the actuator of the fibrosis, because it's the cell that ultimately will get activated and will generate the extracellular matrix. We can see depicted here that we have extracellular matrix deposition in the picture, and we can work with immortalized stellate cells in 2D or 3D. So we have three different cell types that they are one after the other in a participant of this adverse outcome pathway, and the idea is to connect them together in a microphysiological system. This is a very busy slide. I will not go into detail. In the middle here, you can see in this row are actually the same events that you see in the AOP, but there's many endpoints on parameters that are, of course, can be measured when you're trying to quantify the AOP. And that's what I wanted to represent with these slides. So we have biochemical assay, we have cell health, we can have gene expression and so on for the specific pathways which are involved to each of the key events we discussed. We selected a few of them for our approach. So for all the cell types, we always check the viability, but you can see for the hepatocyte, it's also important to measure glutathione content, for example, because oxidative stress that maybe arise, and albumin production, because it's a functional parameter. Hepatocytes and also our cells should produce albumin when they are healthy. And what you see in blue for all of this, and we are developing within this project, are sensors, because the idea would be also to be able to quantify or to assess what is happening with these cells on during the experiment, monitoring it over time. For the Kupffer cells, they are, well, surrogate, the THP1 surrogate for Kupffer cell, they're macrophages. We can see whether they can be activated to M1 or M2 phenotype. We can look at expression markers and cytokine release, and of course, also evaluate with sensors like glucose, lactate, and pH. We can follow them live. And for the fourth, oops, I went one too much. For the stellate cell, we can say that one of the main things that we need to see is whether they are activated and they are producing extracellular matrix. For that, we have a specific activation marker, alpha SMA, where you will see a few times on these slides, and then extracellular matrix proteins, like in this case, I listed collagen 1, but fibronectin on others. And of course, incorporating sensors. So that was the general idea. To do that, so what we require is, on the one hand side, the biology. I'm a biologist, so my presentation will focus mainly on this. Of course, the development of a suitable microphysiological system that would allow us to incorporate sensors and also to take the cells out onto culture for a period of time. Fibrosis, I forgot to mention, is a chronic event, so you need to keep the cells over time. You need to stimulate them over time for that. And here the sensors that need to be low cost, that you can incorporate in your microphysiological system. So I will focus first in the biology, because one of the main things that could fail here, that was a big risk when we started this, is I'm assuming, or the assumption was that the secreted factors are the ones who are responsible for the propagation. If we have the cells connected as here, with the 3 cell types physically separated, just connected by the microfluidic, we are not having, of course, a cell-cell contact. And this may be a problem. So the first thing that we wanted to check is whether the soluble factors are sufficient to really propagate this AOP as described. For this, we did 2 sets of experiments. This is the first set of experiments I'm gonna describe. It was a very simple plan experiment. Took a bit of time, but it's still simple. We used the parenchymal cells, the FRGs in 2D and 3D, the THP1s I introduced already as Kupffer cell surrogate, and the stellate cells in 2D or 3D. And we just cultured them separately, and we work with conditioned medium. So we just very simply passed the medium from one well to the next to see whether we could carry over the effect and how that worked. So here, the first step, and you see here in the top, the box of the study design. So we are now in the first red box, the hepatocytes. We checked what happens to these hepatocytes. We treated the hepatocytes for 3 days with compounds, for example, thiacetamide and methotrexate. They are both profibrotic compounds. And we also incorporated acetaminophen. Acetaminophen causes hepatocellular injury, we know that, but it doesn't lead to clinically to fibrosis, to liver fibrosis. We could observe, if I go quickly through that the cells produce albumin. We could see that we have some effects with thiacetamide and acetaminophen. So, and they have a decrease in viability and a decrease in albumin production. So the first key event was the damage of the hepatocytes, or the FRGs in this case, was given. Just to a note, methotrexate, this was one of our most used model compounds, had very slight effects on the cells. And this is due to the fact that these cultures were only for 3 days. We have a lot of experience with this, and we need a chronic exposure. So we move to the second box here, the second actuator, the THP1 cells. So the first thing we looked is whether they could be activated, and they could be activated by LPS. You see here the secretion of TNF alpha. That was to see that the cells are working correctly in the conditions that we're culturing. But which is more important for the fibrosis is that we have here gene expression of TGF beta, a profibrotic cytokine, which is increased with the 3 compounds that we are treated. The release was also increased. The error bars are a bit too large for my taste. We are repeating this, but we can see definitely a trend on a very clear effect on the gene expression level. So we can see that this second step is also taking place. So moving to the next step, to the third step, we're looking at the stellate cells, and this is what it gets more exciting as well, maybe because I do like these cells very much. If we treat FRG cells here with TGF beta and we carry over the medium, we can see that the stellate cells get activated. We can see here this increased expression of the extracellular matrix proteins and alpha SMA. And we can see in a 2D depiction here the activation. These are the negative controls. I don't want to go into detail, but if you look here, TGF-beta causes alpha SMA expression and stress fibers that we can see very well. The same is for methotrexate and thiacetamide, and which is most important as well, acetaminophen. We have a background expression of alpha SMA, but we have no development of stress fibers. So acetaminophen is not activating the cells. So we were quite happy that we see with soluble factors, we can really carry on the effect. We didn't have, and we didn't seem to have also a direct activation through the compounds directly of the stellate cell, because if the stellate cell would be activated, you can just make an assay with just the stellate cell. But we could see that we needed that. So the next step was to generate a system where we can really have the cells, and again, we're the same three types, so I don't need to introduce them again. The EPRGs, the THP1s, and the stellate cells in 3 different chambers. And what we had, and just is again for the proof of concept, we cultured the cells in these different compartments. And in one setup, we have high walls, so the medium could not move across over the compartments. And in the other, we have low walls, which allowed that the medium could flow very freely between one and the other. So we had the cells independently cultured, or the cells where we have a possibility of diffusion. And we placed them on a rocker so that we can see the cells moving back and forward. And we treated the cells with methotrexate being one of the main compounds that we're discussing, and TGF-beta1, which is just directly a profibrotic compound. I think this is a very, very nice set of results. First, we cultured the cells for a week now. We're trying to go to this chronic. We know we need 1 week to 10 days to really generate a good fibrotic phenotype. So we cultured the cells for at least 1 week, and they were viable in the system, which is of course important. This is the viability data. But what is most important, I think, is this depiction here. And this represents our cells, which have been without contact, so cultured independently. We have the stellate cells here. They get no activate. We have no activation. You see the alpha SMA staining. So if you put the methotrexate directly onto this into these microtissues, we see no activation. But if we have the possibility, if we give the cells the possibility to exchange the medium, meaning all the soluble factors that are generated, we can really see the activation. So this is, I think, a very important result, because it proves that we really can see this activation based on soluble factors. Of course, there might be cell-cell contact factors that also influence fibrosis, but we're now focusing on the soluble factors that we can then measure to generate a quantitative AOP. What we see here on the left as albumin staining, I didn't go much into detail, the EPRG cells, that they have some decrease in albumin staining, as we expect. So we were really quite pleased with the fact that we can see this the biological proof of concept. So we're starting now to move, and again, I'm presenting mainly the biology. I'm just going to give you a flavor of what we're doing. Otherwise, defining and designing a microfluidic device. This is with colleagues from HESO in Switzerland as well. Sophia Alexandra Homsi in her work, where we're trying to generate a microfluidic system. We will then, of course, upscale it, but first we need to find the right system to connect, to make valves which can open, that we can culture the cells independently, and at some given stage, open it, incorporating. So we have tried, for example, with FC40 to generate this interruption and then open it. We need access to the cells from the top, because we want to also make imaging. We need to get rid of, take up the cells for messenger RNA, for example, gene expression. We also want to incorporate the sensors. So we're working on that to generate this microfluidic system. And, there you go, sorry. And we are also working on, this is the colleagues and CSEM, also in Switzerland, they are working on generating specific sensors. They should be sensitive enough and also low cost. So there you can see here a printed sheet of sensors. They're about 2 millimeters in diameter. They're working on miniaturizing further. And we have at the moment already established pH, glucose on lactate sensors that were also tested for longevity. I mean, how long can you keep them working in cell culture medium in the appropriate matrix? And we can really measure this for the live monitoring of what's happening. We are also discussing to incorporate reactive oxygen species sensors. There are a couple of species that we are discussing that we will like to incorporate because it's important to see how the cells are performing. Because the main goal is to really generate quantitative data. We need to measure what is happening with these cells, and we need to know how much cell damage or how much cytokine release or how much reactive oxygen species is necessary to really trigger the next step, because a bit of change, it doesn't harm. And we see that also. I mean, I'm also interested in using these to try to elucidate a bit more what's happening with this acetaminophen. It causes hepatocellular damage. We even see TGF-beta expression, but we don't see fibrosis in our model that I'm not presenting here. We don't see fibrosis in the clinic. So what makes a difference there into this chronic slow release and the acute hepatotoxicity that may or may not lead to fibrosis? I think I hope I could convince you. At first, that is, I think it's a very novel concept. We're taking that cell, the things apart instead of putting them together. So it's not a multi-organ chip; it's a sequential cell chip, which I think reflects this AOP and may be useful for many other AOPs, hopefully in the future. I think the data, I hope I could convince you that the data support the feasibility biologically to do this. We have some, for the microfluidics, we have some concepts which are in place, have been developed. We are working with these disposable low-cost sensors because they need to be integrated and you want to throw them away. At the end of the day, we would like to generate something that can be used repeatedly. Now we are in the prototype time. And also that this contact-free development of the liver fibrosis has been proven by the data we generated. So the ideal team, I mean, I'm at the Fachhochschule or Applied Science University. I'm specialized in the biology. I've been, as I said, working many years with multicellular three-dimensional systems for liver fibrosis, under other things. But also we have the colleagues working with microfluidics and the colleagues working with sensors. We are all together working on this. The project is obviously at the moment not completed. This is clear. We have a good concept, and I think we have a biological proof of concept. We have a couple of more years to go, and we will making progress. So hopefully I can report soon another time, because we can use it for other AOPs as well, for safety assessment, for antifibrotic therapies, and of course 3Rs is very high, very largely written here. So with this, I just need to thank all the people in the 3 different organizations which are part of the small consortium, and mostly thank also the Bridge Discovery grant from the Swiss National Research Foundation, which is funding this research. And thank you very much for your attention. And I still have 2 minutes.
You have more. Thanks, Laura, for a great presentation. We have some minutes left for questions. There's already one there.
Robert Strange from University of Latvia. First of all, very nice talk. I was wondering, could you comment a bit more on those sensors? Like, is that real-time reading within the channel, or is that you're reading in the outlets, using hydrostatic flow?
The idea with the sensors is to really incorporating, not in the channels, but on very nearby to the cell wells, actually, to just get them, be able to get them from the side, really. And of course, we have to connect them to an electronic device to measure the reading. But this is the idea, to have them there. And that's why they had to perform well. Maybe they're not the best performing sensors, but they need to be low cost and disposable. So this is what we're fine tuning these details there. Exactly.
I also have a question related to the sensors. Then obviously I understand why you do it. This is more for the cell health, but if you want to have data for your specific outputs, are you also thinking of a specific fibrosis biomarker sensors or DILI biomarker sensors?
Yes, but I mean, this is, again, I'm a little bit out of my depth with the sensor technology, but I think we don't have at the moment the technology to measure. I mean, it would be great to have and incorporate additionally even interleukins, for example, or TGF-beta. I mean, but I don't know how feasible that is. So we started to look into to follow the cell health because this is something which we know we can achieve in a relatively short time. But we are working on thinking at least on other sensors that we can use. I mean, I think sometimes about lateral flow, for example.
I'm pretty sure there are ALT sensors out there, but I'm sure in vivo, I'm not sure whether they work. But
yeah,
it's a great concept. I like the idea of the AOPs and being able to separate everything out. I think we still have 1 minute. I have one last. Oh, there's a question.
Thank you very much for the very interesting talk. And you are focusing on the soluble factors. So that depends on so the effect of soluble factor depends on the concentration. And the soluble factor itself is produced by the cells. So how did you select the good amount of the cell? So how do you set the number of the cell you cultured for each cell types? Yeah.
Okay, this is a good question, and I would say that we have a bit of empirical experience more than a very reasoned approach. I mean, we know what we can achieve, and we're working with in the 3D. We're having for the stellate cells and for the APRGs, we're working 3D, so spheroids. They need to be small because if you go above 250, 300 micrometers in diameter, they will start developing a necrotic core that we don't want. So we have to limit the cell numbers of that. So we're trying to get the cell numbers. It's not a physiological proportion between the cells, but we're just looking what is best for each cell type at the moment. It may be something that in the future we could adjust, actually. This is a good point. But at the moment, we are just making it as good as possible for each of the cell types.
So for the concept of proof experiment, do you think it is not necessary to the cell ratio and physiological or not?
No, I don't think so, because this is maybe something, of course, I didn't present today, but I've been working on fibrosis, and we make spheroids with the 3 cell types, and we have a non-physiological condition, proportion, and you really can elicit fibrosis. Because if you're only working with 2,000 cells, and you only have 3% stellate cells, then you can never see anything. So you have to kind of force the system a little bit. Again, it's a model. It's not the liver. I haven't even mentioned the endothelial cells, for example, which may play a role, but it's a model. So we try to make each cell is happy or maintained properly, and then we go from there.
Thank you.
Okay, I think I have to go because I'm chewing on the next speakers. Okay,
So-
thank you very much, and especially for people who came after the party, came so early to the first talk.
So our next speaker will be Daniel Navid Tevakal, and he is from Columbia University.
Hi, everyone. My name is Navid Tavakol, and I'm a PhD candidate in Dr. Gordana Vunyak-Novakovic's lab at Columbia University. I want to first thank the organizers for having me here today, and also letting us talk about our work on an astronaut on a chip, a human multi-organ platform for assessing the extended effects of cosmic radiation. So one of the greatest risks of space travel and intended space travel to the moon and Mars are really the effects of cosmic radiation. And so what this means is that there are a number of different radiation sources that come into contact with either a spacecraft or humans that are in space. And these risks are actually hundreds of times greater than the amounts of radiation that we see on Earth, and also in like airplane travel, for example. And so what we're trying to do is better understand these effects in both the context of galactic cosmic rays, which can include a number of different ion sources in space, but also the interaction of these radiation sources on a spacecraft intended to go to Mars. And so the framework that we're really using in some of these studies is really to understand the predicted radiation risk. So what people have already identified over the past maybe 50 years or so on damages to human health that might be a result of any types of radiation. This includes acute radiation toxicities, malignancies of the blood, cardiac arrhythmias, et cetera, that might be in a problem for different human health. And the crowd here is really familiar with the organ-on-a-chip systems and different microphysiological systems that can better help us understand physiology of human health in these contexts of radiation that you might not be able to mimic in other human contexts. Especially animal models, they're kind of the gold standard for radiation research at this point, and people have been using any human system, so whether that's ex vivo cell culture, to better understand how radiation may affect human cells. And what we hope to do here is really be able to take a patient sample or an astronaut sample of their blood, reprogram these cells into iPSCs, and then differentiate these pluripotent stem cells into a variety of different cell types. The goal here is really to envision a patient on a chip where you have one specific organ or multiple different organs interacting together to be able to study different radiation sources, different doses of radiation, and also the timelines, because we're really concerned about long-term space travel and how smaller doses of radiation over a longer period of time may affect human health. And some of these things are a little bit harder to study in vitro systems because you need really those longer time courses, but we're hoping to get there over the next 10 years or so. And so what our work is really focused on is engineering a human organ-on-a-chip system to study the systemic effects of radiation toxicity in this Envision Mars mission. And so we've chosen 3 model systems in this approach: the heart, bone marrow, liver, and then a vasculature barrier, so a compartment to help separate our individual tissue niches from a recirculating compartment. And then study these effects of these different organs to photon radiation and neutron radiation. And I'll go a little bit more in depth into the differences there. We hope to study both acute versus chronic exposure timelines. And so in a traditional setting where you have accidental radiation exposure, this would come in an acute context, but this isn't really a good mimic for what you would see in space, because these are, again, smaller doses over longer periods of time. What we hope to do in future work is really benchmark our engineered tissues and what we're seeing in vitro to any human metrics, so data available from either patients or people who have gone to space and come back, or accidental exposure from different nuclear disasters in our history. And then really to be able to identify radioprotective measures that could be able to mitigate some of the radiation effects that we would see in an intended Mars mission. And so what we did in our study here is really use a source of high LET radiation, which is mimicking neutron radiation. And so these are the effects that you would see when galactic cosmic rays interact with the spacecraft. So not necessarily all the different ion sources that you would see in space, but a portion of them that the actual astronauts would sense. And so we have a facility actually conveniently at Columbia called Nevis Laboratories, where they have these neutron sources and are able to mimic some of the effects of neutron radiation outside of space. And here's just a graph comparing where the GCR spectrum is in space and what you could get more similarly in some of the facilities that people are looking at simulated galactic cosmic radiation. For example, Brookhaven Laboratories in Long Island of New York also has this type of radiation that we're hoping to get to in the next few years. And then the spectrum of high LET sources that we'll see in our Columbia neutron facility. And so my colleagues already talked a little bit earlier in the in the meeting, Casey and Diogo, on our Organs on a Chip platform. But a basic introduction is that we have a platform that we've developed over the past 5 or 10 years that really is able to take patient samples, whether primary cells or iPSC-derived cells, engineer individual tissues in their own tissue-specific environments, and then integrate them together in a context where you have each individual tissue in its own tissue-specific media, so it's individual niche, and then have a endothelial barrier beneath this that can help separate the media that is for that individual tissue and a common recirculating media. And so the idea here is that you could have each tissue matured separately and then brought together and study some of these systemic interactions in vitro. And so what we look for in our platforms is really to make a platform that's modular, so you can add in any tissue type, mature these tissues over weeks to months, study their connectiveness in the systemic context for a period of 4 weeks, and then try to integrate functional readouts both online and also at the endpoint of our cultures. And so what we're hoping to do is really build complexity to these models. So Casey's paper was published just last month, actually, so you can read a little bit more about the platform itself. But in this work, we're integrating the liver, heart, and bone marrow, which are different organs that were intended for both some of the more sensitive effects of cosmic radiation in the bone marrow and heart, and then liver as an organ that can help metabolize drugs when we put drugs into our platforms. And so just a brief overview of our engineered model of the human bone marrow. We use decellularized bone in a hydrogel system to integrate osteoblasts, mesenchymal stromal cells, endothelial cells, and cord blood-derived hematopoietic stem and progenitor cells. And we can see over a period of 1 week and 2 weeks here that we can maintain the hematopoietic stem and progenitors, and then also over a period of 2 weeks, see myeloid differentiation of these cells into different phenotypes. And so this is just brief characterization of the different stromal populations that are in the tissue niche, as well as the blood cells forming from the engineered human bone marrow. And so just looking at the isolated effects of radiation on the engineered human bone marrow, so without combining it in the systemic context, we see that there's an immediate increase in inflammatory cytokines from the engineered tissue in response to this 1 gray of neutron radiation. We also see the structure of the tissue is affected in response to the higher doses of radiation. And we chose a photon dose and a neutron dose to really compare. Photon doses are more similar to what you would see in cancer therapy, and then the neutron doses are more similar to what you would see in the secondary effects of radiation. And so the neutron doses are about 4 times as potent as the photon doses, which is why we have the differential dose in some of these studies. But we see over a period of 3 weeks that we actually have a reduction of CD45 positive blood cells coming out of the tissue niche over in response to radiation, as well as an increase in median fluorescent intensity of CD45 on those cells, indicating that these cells are either differentiating and becoming larger. And with some of our molecular characterization, we've been able to corroborate some of these findings. So we perform single cell sequencing on our engineered tissues in response to radiation, and we saw that in the photon and neutron doses, there was actually a shift in the population of myeloid cells that was appearing. And we saw a large increase in the population of CD14 positive cells, CD68 positive cells, and a large portion of these cells in this subcluster here is actually populations of monocytes and macrophages that are differentiating towards a M2-like phenotype, so reparative phenotype after 3 weeks of 3 weeks post-radiation. And so the gene ontology terms that are interacting in the system are really extracellular matrix organization and migration of these myeloid cells. And for us, this gives some indication that the system itself is trying to repair itself post-radiation damage. But as we noticed in the previous slide, a lot of the production of the blood cells is actually affected early on with this acute exposure. And so it gives some indication into some of the longer term interests that we have of looking at smaller doses of radiation over longer periods of time, and if this effect is still seen in those cases. When we looked at the effects of radiation on engineered human heart muscle, we noticed, or actually, sorry, a brief overview over engineered human heart muscle tissue. So over the period of the past 10 years or so, our group has developed a platform where we have a pillar system where engineered iPSC-derived cardiomyocytes and primary cardiac fibroblasts are integrated into a fibrin hydrogel and then electromechanically stimulated over a period of 4 weeks to mature the tissue. And so this is published over the past 5 years or so. And then we have a small adaptation over the past year where we have a smaller tissue format so we can integrate this into higher throughput studies. And so these engineered human heart tissues in response to radiation, we saw actually a decrease in maximum beating frequency and an increase in excitation threshold for these tissues. And so what this means is that the tissue would actually need more stimulation or more electrical stimulation to reach the same threshold as what would be seen in the healthy control tissue. And an interesting thing that we had noticed as well was that the force generation was actually higher in these neutron treated groups. And so this gave some indication to us, and looking also at the molecular characterization, that there could be an increase in early hypertrophic cardiac tissue phenotype. And this is an area that we're actively exploring with our data. And so getting to the point where we actually use the studies of isolated radiation damage and try to integrate this into a systemic context, we created a system where we engineered each of these 4 different tissues with primary cells or iPSC derived cells, and then integrated them into the systemic context that I described earlier. And so what this means is we have our engineered human bone marrow, as I described before, our engineered human heart tissue, our engineered human liver tissue, which is a combination of hepatocytes and primary fibroblasts in a fibrin hydrogel to mimic some of the metabolic functions of the liver, and then an engineered human vascular barrier where we have endothelial cells and mesenchymal stromal cells to help prevent basically mixing of the media. So we have this individual tissue niche compartment from each individual tissue niche to the circulating media. So we mature these tissues individually over periods of 4 weeks. 4 to 6 weeks, and then integrate them into the systemic context, expose them to either an acute dose of radiation or a protracted dose of radiation. And so this protracted dose is really trying to mimic that smaller dose of radiation over the period of 2 weeks. And then we ended the experiment at that 2-week time point to really characterize what are the effects of what's happening to each of our tissues in response to both an acute dose of radiation and some of the longer term doses of radiation. And so what we saw is we actually saw the same myeloid skewing of the human bone marrow in these systemic contexts. So we noticed an increase in CD11b positive myeloid cells. And what was really interesting is that actually the groups that were treated with the protracted doses of radiation, so smaller doses of radiation, but the same total cumulative dose, actually had a higher proliferation of these CD11b positive myeloid cells. And so it gives some indication and also an increase in myeloid-derived suppressor cells. And so this actually correlates with some of the early findings people are having on increases in myeloid malignancies in response to some of these smaller doses of radiation. But we're doing more molecular characterization to really understand if these cells themselves are inherently changing or if it's a result of, is it a result of the actual dose, or is it a result of the type of dose, so the neutron dose in specific. When we looked at our cardiac muscle phenotype over time, we noticed that early on in the acutely treated multi-organ platform, we saw an increase in cardiac troponin release at that 24-hour mark, but this was actually restored to normal in the later time points, so the 1 to 2 week time points. Which makes sense because in the smaller doses of radiation, the cardiac tissues are actually being stimulated with very little radiation over that at the everyday time point, but in the acute dose, they're getting 0.5 gray of neutron radiation at one time. But what we also noticed is that the maximum contraction velocity, like we had seen in the isolated tissue studies, was actually also increased in the acute dose and protracted dose, higher in the protracted dose than you would see in the acute dose, which corroborates some of what we're seeing with the bone marrow, in that the protracted dose of the smaller doses of radiation might actually be having a more detrimental effect to the tissue itself. And then when we looked at the liver, we noticed that the liver actually didn't have any significant, or not many significant differences in albumin and urea secretion, which is what has been seen clinically, and also what people are seeing with animal studies with the same doses of radiation in neutron-specific context. And so, taking all of this together, we're really trying to use our organs on a chip systems to really study radiation and the effects of cosmic radiation on human tissues. These patient-specific models of bone marrow, heart, liver, vasculature can answer specific questions, whether that's dosing or the use of radioprotective agents. We've been looking at some radioprotective agents for the hematopoietic system, like GCSF, as well as some other predicted measures that people have used in previous studies for neutron-specific contexts. And then using the crosstalk between multiple organ systems in the systemic context can really enable studies of astronauts on a chip, and taking a specific astronaut's blood, creating IPS cells, and then being able to see that specific astronaut's, the effects of that type of radiation in that specific astronaut over this period of time. And we're really excited for this because it can really inform future studies and what people are really intending to do in space travel as well. And I just want to thank my lab, the entire lab, especially Casey and Diogo are here at the conference. So if you have any questions, please feel free to reach out to us. And then also NASA's TRISH initiative, which has been extremely supportive in funding this work, but also really helping connect us to the space biologists that are doing these studies in animals, and being able to try to compare what we see and what we see in what others have seen in radiation specific contexts. So thank you so much.
Thanks, Daniel. Awesome talk. Brings me back to being a little boy and I want to come to Colombia. Are there any questions from the audience? I have maybe some more generic questions. You using the iPSC cells? How have you checked how confident you are, how mature these tissues are when you actually put them into your chip?
Yeah, definitely a very great point. So I think we're still developing the right metrics to characterize our tissues. So we've done a lot of work in the cardiac context over the past 5-10 years. And with our other tissues, we're really trying to validate our tissues and benchmark them as close as we can to available human data. And until that point, we're really trying to, in parallel, really study the effects of radiation in the both primary context, but also IPS, and see if there are differences. And if there are differences, is that a lack of maturity of the cells, or also are those cells not performing as well because of whatever artificial systems we're kind of developing? And so I think that's why the benchmarking is so critical in our studies, at least.
Have you already got things set up to get astronaut blood? Was it a wish?
Yeah, so astronaut blood, we haven't specifically, but the goal is really over the next few months and year really is to compare the effects that we're seeing with these neutron radiations to the simulated galactic cosmic rays at the Brookhaven facility I mentioned before. So the idea is at that point, we'll hopefully be at the right, I guess, vantage point for everyone to kind of come together and then either provide astronaut blood or collaborate with the people who are doing some of these other studies in vitro.
So I have a question pertaining to devices and biomaterials. So how do you control for the radiation's impact on the device and the biomaterials and leachables that you might have?
That is a very good question. So we haven't specifically looked at it in this context, but I think for all the materials we're using, they're not actually in our system, they're not actually they don't become radioactive after that exposure. But I think if we're using materials that do become radioactive, we have to do a further step back and see what actually we can
Yeah,
mitigate
I guess
there.
the question is more about what is it doing to the material rather than it becoming radioactive.
Oh, so we haven't looked into that, but I think that's a very good question that we can investigate.
I have a question. Do you also have a possibility to measure for the cardiac for the muscle? Can you measure the strength of the intensity of the contract, the force actually of the
Yeah, so
contraction?
I showed some of the functional data, but yeah, we can look at the
Okay.
force as well, and we've looked at that data.
Okay, it would be interesting to use this system also to study microgravity or things, because this has a lot of influences. I was learning recently, also in cartilage and other tissues, in astronauts, they suffer because of that. But of course, it's a different question. So,
Definitely, yeah. So I think for this study, we chose the specific organs we chose for one, to look at the most sensitive or some of the most sensitive organs, and then also in the Trish work, we're really trying to develop radiation countermeasure tools, and so the liver is really critical in this. And I think in future studies we can integrate more organs. It just I think limitation of systems.
of course.
But, yeah. Thank you.
Okay, brilliant. Thanks a lot, Daniel. If there's no more questions, we should move on. So it's a pleasure to introduce the next speaker, and that's Ilona Weil from Boehringer Ingelheim in Germany. And she's going to talk about integrated human intestine liver on a chip.
Thank you. So good morning, everyone. It's really a pleasure for me to give a talk today, as I switched just a couple of weeks ago from a poster presentation to the talk. So I'm a postdoc at BI in the non-clinical drug safety department in Germany. And I'm in the investigative toxicology group. So I'm not sure if you're all familiar with investigative toxicology. So I would like to give you a short overview what our goals are and how we can use MPS in investigative toxicology. So in investigative toxicology, we support the whole drug development process. In the early stages of the drug development, we provide risk assessments for targets or chemical classes. And the goal is to have a risk anticipation. And in later stages of the drug development, for example, in non-clinical studies, in clinical trials, or even when a drug is already on the market, we might get safety alerts. And whenever we are getting a safety alert, we try to elucidate the mechanism of toxicity. And here the goal is to have a de-risking and to propose de-risking strategies and also analyze its relevance for humans. So we can use MPS for both ways, as a tool for predictive toxicology, but also for retrospective mechanistic investigations. And today I would like to focus on a retrospective mechanistic investigation. As our first MPS case study, we chose agonists for the free fatty acid receptor 1, which is also known as GPR40. GPR40 agonists have been developed for the treatment of type 2 diabetes mellitus. And probably the best known GPR40 agonist is fasagliflozin. It has been developed by Takeda, and it was a very promising candidate in the preclinical studies. It made it to the clinic, and then in the clinical phase 3, it had to be stopped due to drug-induced liver injury. And this was especially interesting for Boehringer Ingelheim, as we had a competitor compound at that time, which we then also stopped due to safety concerns. So until now, many studies have been performed to analyze the mechanism of toxicity, but it's not yet fully elucidated. We are currently investigating the MPS from tissues. And I'm sure that most of you already know the system, as we already had several talks about the system, also quite a lot of poster presentations yesterday. So this is just a very short overview. Here you can see the chips. We have a recirculating system of the medium in the chips. We have on-chip peristaltic micropumps, and Tissues offers chips for a combination of organ models. They offer chips for a combination of 2 organ models, 3 organ models, or even 4 different organ models. And one advantage for us is that we are very flexible in the combination of tissue models. We can insert the tissue models in the chips that we have already worked with in static cell culture. Another advantage is that the chips are compatible with live tissue imaging. And also we have permanent access to the different organ compartments to sample media, for example, or to add compounds. For our GPR40 agonist study, we wanted to have a closed in vivo-like situation. So we chose a combination of intestine and liver, and this combination gives us the possibility to mimic an oral substance uptake. We can add the compound to the intestine compartment, which will then be transported through the intestinal cell barrier in the microfluidic channels to the liver compartment. And we can also compare it to an intravenous treatment. And we are currently investigating a 2 weeks repeated dose substance testing. We have an adaptation period of the cells in the chip for 1 day. And after that, we start with the substance treatment. We treat the cells each day with the GPR40 agonists, and also each day we are analyzing the medium. We are analyzing for cytotoxicity with LDH. We are analyzing the cytokine release, liver function with urea secretion and albumin, liver injury with AST, ALT, and GLDH. And as we want to have a really detailed insight in the mechanism of toxicity, in the end, also we analyze the cells with high content imaging. We analyze their ATP content, and we perform a next generation sequencing. So it's really a high content experiment as we have a huge variety of different endpoints. For the cell models, we are currently using ready-made commercially available cell models. For the intestine, we use the epi-intestinal cell model from Mattek. It's a transfer cell model, which you can see here. It's made from primary human small intestinal epithelia cells from a single donor. And already here in the bright field overview, you can see the nice villi-like structure, which you can also see then here in the cryo sections, stained with antibodies and H&E. For the liver, we used the 3D Insight human liver microtissues from Inspiro. They offer human multi-donor primary hepatocytes in a co-culture with Kupffer cells and liver endothelia cells. And we add 45 of these spheroids in the chip. Now, when we started working with spheroids in the chips, first of all, we realized that we have some challenges here to overcome. We wanted to have an easy way to insert the spheroids in the chip, but also being able to remove the spheroids afterwards from the chips. So we started by just simply adding the spheroids in the organ compartment. And what we realized after a period of 14 days in the chips, we got an aggregation of the spheroids to the PDMS borders in the compartments. So it was impossible for us to remove the spheroids from the chips. And what we also observed was fusion of the spheroids, which then led to necrotic cores. So it was impossible for us to get reproducible data with this setup. So fortunately, Tissues is now also offering chips with ultra-low attachment DIN arrays. And these DIN arrays gives us the possibility to pool the spheroids in the organ compartment, but still the spheroids are separated from each other. As you can see, we have here small little cavities where we add the spheroids, and it's not possible for the spheroids anymore to fusion. And here we could see, we could observe no shrinking of the spheroids. The diameters of the spheroids of day 0 was comparable to their diameter after 13 days. And also we got a stable urea production over 14 days and nicely reproducible data. So today I will show you the long-term repeated oral treatment with the GPR40 agonists. As you probably know, we have to focus on some concentrations in the chips as we still have a quite low throughput. So here we chose to work now with concentrations which are not cytotoxic, as we wanted to get a detailed insight in the mechanism of toxicity. We chose to work with a slightly lower concentration of our BI competitor compared to fasagliflozin, as the BI competitor is also, it's slightly more toxic, but also slightly more potent in vivo. And so with the concentrations we chose, we measured the LDH value each day. And here we couldn't see any significant differences to the control. And what we found was very interesting is that in the intestine, we even got lower LDH values for the treated cells compared to the control. We also made sure that we have a stable barrier function. After 14 days of treatment, we investigated the barrier function of the intestine model, and here we couldn't see any significant changes with the concentrations we used. Also, we couldn't see any distinct differences in the histology of the cells, which you can see here in the cryo section for the control, and here the two different GPR40 agonists. So, of course, we were also interested in the concentration which we actually have then in the liver compartment. So we add 200 micromolar of fasagliflozin to the intestine compartment, which will then get absorbed, transported through the intestinal cell barrier, distributed in the chip, and then eventually gets metabolized in the liver spheroids. And what we found is that we actually have a quite low concentration in the liver compartment. In the beginning, we had only 2 micromolar, and then it increased to around 10 micromolars. So this is actually less than 5% of what we added to the intestine compartment. And there are several reasons for that. So one, of course, might be metabolism. It might be already metabolized in the intestine cells, but then also in the liver spheroids. And another issue or challenge what we have with GPR40 agonists is that they are highly bound to proteins. So it's known for fasagliflozin that it has a human protein binding of 99.8%. And we have some proteins in our cell culture medium. And an issue which we, of course, always have is non-specific binding to PDMS. Now we had a closer look at the spheroids. And in the bright field image, on first view, we couldn't see any differences between the control spheroids and the treated spheroids with the two different GPR40 agonists. We also checked the spheroid area, and here we could see that our treated spheroids were slightly smaller. We also checked the liver function with urea secretion, and here we could also see the urea secretion for the treated spheroids was slightly lower compared to the control condition. ATP content also slightly lower compared to the control. And then we also checked the mitochondrial membrane potential with a mitotracker, and here also we couldn't see any significant differences. Now, in the next step, we performed the high content imaging and analysis of hepatic transporters. We performed 2 different live stainings. One was CLF, which is a substrate for BSEP, and one was CDCF, which is a substrate for MRP2. And for BSEP, we got quite interesting results. In the control spheroids, we can see nicely the bile canaliculi, but in the treated spheroids, there are hardly any bile canaliculi. So we also confirmed this with the high content analysis. And here we could see a significant decrease of biocanaliculi area. We also checked if we have a lipid disorder with anyl red staining. And here we could see for both of the two different GPR40 agonists that we have an accumulation of lipids in the hepatocytes, which you can also see here when we counted the NIRAD positive cells, that we have a significant increase of lipids in the hepatocytes after treatment with GPR40 agonists. So as I said before, we also checked the cytokine release in the cell culture medium, and we checked a huge panel of different cytokines. And what we found was that actually most cytokines were below the detection range. We could only measure IL-8 and MCP1. And for both of them, we couldn't see any significant increase of cytokine release after treatment with the GPR40 agonists. We also performed a static cell culture experiment with the spheroids where we induced the cytokine release with LPS. And here, when we tested the different concentrations in the LPS condition and without LPS, we couldn't see any shift of the EC50 value for both different GPR40 agonists. So to sum it all up, we investigated the two GPR40 agonists in concentrations which are not cytotoxic, as we wanted to get a detailed insight in the mechanism of toxicity. We ensured in the intestine cell model that we have a stable barrier function, and we found no distinct differences in the histology. In our high content imaging and analysis, we could see a decreased transport capability of BSAP and accumulation of the lipids in the hepatocytes. So in conclusion, the two GPR40 agonists may lead to cholestasis and steatosis. And as we had no increase in the cytokine release, we have no indication for an inflammation-mediated hepatotoxicity. So we could successfully use our two organ model for a long-term oral exposure of the GPR40 agonists. And what we are currently doing is that we are also performing the next generation sequencing to get an even more detailed insight in the mechanism of toxicity. And with that, I would like to acknowledge all the people who were involved in the project, especially my supervisor, Mario Ballmann, who is also here together with me. And I would like to thank you, of course, for listening. And I'm happy to take any questions you might have.
Thanks, Ilona. Great presentation, and thanks for highlighting investigative talks. Question?
Hi, yes, very nice talk. I'm wondering if you looked at the inhibition of BSEP in a more simpler system.
You mean
vesicles.
Just in spheroids without an MPS?
Well, just like BSEP vesicles or some kind of in vitro conventional assay. Did they show inhibition in those simpler systems?
We haven't done that, but it's published. It's already done by several groups, and several groups have checked it and also found that they have a BSEP inhibition also in other systems.
Okay, thanks. Makes sense, right?
Yeah.
Hi, thank you for the great talk. Just wondering if you looked at CYP activity at all in your liver?
We haven't done that yet. We might do that also, but until now we haven't done that.
Is that going to be part of your next gen sequencing stuff? Thank you.
Hey, thanks for the talk. I was wondering, you showed the differences between BSAP and MRP2. Also, like, due to my knowledge, like MRP2 is also lining the bicanaliculi. I was wondering if you did like a double staining, because it looked like that MRP2 actually the bicanaliculi looked still intact. So if this is like a very transporter specific like effect you observed?
Yeah, so it might be transporter specific. And we hope that with the next generation sequencing, when we look at the expression of the different transporters, I mean, there are a lot more besides BSEP and MRP2. And with that, we hope that we get a more detailed insight in the differences, if it's specific to some transporters or a general effect to all hepatic transporters.
Thank you.
Thanks.
I have a quick question as well. You hypothesized the mechanism.
Mm-hmm.
Is it known from the clinic what the mechanism of DILI was?
So many people already hypothesized a mechanism, and so the inhibition of transporters is hypothesized by different groups until now, also that there is somehow a lipid disorder. Currently, we are also comparing our in vitro data with in-house in vivo data to see what they found and if that is comparable.
Cool. And one last question, if there's no one waiting. You mentioned about the low exposure to the liver in the tissue system. Is it known clinically what the liver is actually exposed to?
I think it's not known, no. As far as I know, not.
Great, thanks.
Thank you.
Our next speaker will be Kristen Bursack from MPS.
Thank you for the introduction, and thank you to the organizers for the invitation to present. I'm very excited to share with you an overview of a study where we employed a high throughput proximal tubule on a chip platform for nephrotoxicity detection. And I think at the very beginning, it's really important to note what a great collaborative effort this was. We had colleagues participating from MPS, as well as from Radboud University, the University of Applied Sciences, Northwestern Switzerland. We actually heard from Laura earlier today. And we also participated together with Utrecht Institute for Pharmaceutical Sciences. I'd also like to point out that we collaborated closely with 3 different pharmaceutical companies. So that's GSK, Roche, and Pfizer, who actually provided blinded compounds for the study. So let's jump in. So drug-induced kidney injury is a dose-limiting factor in pharmacotherapy, and it's often associated with kidney failure in many patients. We know specifically that especially the renal proximal tubules are susceptible to drug-induced kidney injury due to their active role in the secretion of xenobiotics into the urine. Now, currently in preclinical drug development, only about 2% of drugs actually fail due to nephrotoxicity. And this ends up leading to 75% of clinical nephrotoxicity that's not detected in that preclinical phase. So really taken together, these data suggest that in order to prevent drug-induced kidney injury, to save patient lives, we need to detect it earlier on in the drug development pipeline using complex in vitro models that are robust and reproducible. However, many of those out there have a number of limitations, including the complexity of the model, the fact that they're low throughput, maybe they only have a single chip or they're connected to pumps. They often lack the apical and basolateral accessibility of the proximal tubule, and they finally lack the cell-ECM interaction, perhaps due to the presence of a semi-permeable membrane. Now, there is a platform out there that addresses many of these limitations, and that's the MPS organoplate. So perhaps you've already heard about the organoplate previously during the meeting, but I'd like to just give you a bit of an overview. So here we see the organoplate bottom is made up of microscope quality glass. It's situated in a microtiter plate, and our chips are actually embedded within that glass, and we can have anywhere from 40 to 96 individual tissue chips within the organoplate, so nice and high throughput. Now, if we look at the top of the plate, it may look a little bit more familiar to you, similar to other 384 well plates you may have seen before. So you can use a pipette or an automated liquid handler to add your cells, extracellular matrix, and media to build your stratified tissues layer by layer within the microfluidic channels. There should be a video here for you, but it's not starting. I wonder if from the back of the room, would you be able to start the video? Excellent, thank you very much. So now this video will just give you a bit of an overview of how the organoplate works. So we're going to go ahead and zoom in on a single tissue chip. We see we have 3 lanes running in parallel. This is our 3-lane 40. Now there's no physical barriers that are separating the 3 lanes. Instead, we have these short ridges of plastic called phase guides that are capillary pressure barriers. So when you pipette in your extracellular matrix gel, the capillary pressure barriers pin the gel within the channel and prevent spilling over into the perfusion channels. Once solid, we can add cells like proximal tubule cells to that perfusion channel. They adhere to the ECM, and we can begin perfusion by simply placing the plate on a rocker system within the incubator. And the wells of the microtiter plate act as medium reservoirs, such that as the plate rocks back and forth, the media flows through those microfluidic channels, and then it's programmed to rock back in the other direction. And over time, you'll form this 3D proximal tubule structure in that perfusion channel. So there are many flavors of organoplates. I'd say the simplest is first our 2-lane organoplate. So this one has only 2 microfluidic channels in parallel, separated by a single phase guide. And we can have up to 96 chips on this plate. So we use this commonly for our liver model, where we have hepatocytes embedded in the gel and endothelial cells and Kupffer cells against that channel. Now, we already discussed a little bit the organoplate 3-lane, and you'll see that later in the presentation for our proximal tubule model. We've recently developed the organoplate 3-lane 64. So this not only has increased throughput to the 40, but it also is more compatible with high throughput automated liquid handling. And then finally, we have the organoplate graft, which is a bit of a variation on the 3-lane in that the gel channel is open on the top so that you can graft on a spheroid, a tumoroid, or organoid tissue, and perhaps have it vascularized by a bed that's within that ECM. So I hope you can begin to appreciate that the organoplate is one platform for all cultures, and it often addresses the drawbacks of many of the classic laboratory setups. I think particularly when thinking about transwell cultures, we are membrane free. We also have, that nice access to apical and basolateral sides. We can introduce perfusion to the cultures, which we find that fluid shear stress enhances the differentiation of the cell types frequently. The throughput, as we already discussed, and it's quite compatible because of that microtiter plate format with those automated liquid handlers, but also plate readers and high content imagers that you already have in the lab. And there's some really nice examples of some beautiful confocal images you can see on the screen. So we can see through that microscope quality glass, you can easily capture these beautiful images that are also quantifiable. Okay, so now we can dive into the proximal tubule on a chip and the nephroscreen study that we performed. So to set up our proximal tubule on a chip, we used the organoplate 3-lane and actually used 2 different types of cell lines. So the first was a conditionally immortalized proximal tubule epithelial cell that overexpressed the OAT1, organic anion transporter 1, which is an uptake transporter that plays a role in toxicity in the kidney. And the other was using immortalized RP-Tex from Sigma. And we found when we exposed them, when we added them to that perfusion channel, exposed them to shear stress, we observed some really nice differentiation of the cultures where we observe an apical and basolateral side of those proximal tubules. So to assess drug-induced kidney injury, we used 4 model nephrotoxic drugs, in addition to those 8 blinded compounds that I mentioned earlier that were supplied by pharmaceutical companies. For each drug, we used 2 concentrations and selected 2 time points. And we evaluated a number of different endpoints across multiple laboratories. And we'll see that in the next slide. And I think that really lends to the robustness and the reproducibility of the platform. So here we have a bit of our screening workflow, each for the OAT1 cells and the RP-TEX. And we can see that each rectangle represents the location where that assay was performed. So for example, on the left-hand side, if we look at the OAT1 cells, Mehmet has performed all of those that are highlighted in green, while folks at Radboud University, all of those in blue. And then at the University of Applied Sciences, those rectangles in red. So really big collaborative effort. And then in the next coming slides, we're not going to review all of the data. I'll just give you a bit of an overview. So if you are interested in more information, I invite you to visit our publication, which is listed below. So first is the barrier integrity assay. So this is a measure of the tight junction function of our proximal tubules. And it tells us a little bit about the health of the cultures. And we can do this by using a fluorescent dye, like a FITSI dextran dye, and we add that to the perfusion channel. And if our tubes are intact and leak tight, then you'd expect that fluorescent dye to stay within the perfusion channel. And you can see that over here, we have a nice example of a leak tight tubule, so that fluorescent dye is staying in the top perfusion channel. However, if we've applied a toxicant that's disrupting the cells, disrupting the tight junctions, then you're going to see leaking of the fluorescent dye into the gel channel. And that's what we see in our next fluorescent image. So what we can do is actually quantify the fluorescence intensity in the perfusion channel as well as the gel channel to calculate the apparent permeability. And when the apparent permeability is low, then we actually have a really nice and great intact barrier. However, when the apparent permeability increases, we know we're losing our barrier function. So if we look over on the right-hand side, just a review of our results in the RP-TEX cells, we have one graph on the left that features results from the 24-hour time point and then 48-hour time point to the right of that. And on the y-axis, we have PAP value, and then all of our blinded compounds across the bottom on the x-axis. So I just want to point out one example to give you an idea of how to interpret the graphs. So if we look at the very first gray bar, we see that we have a nice and low PAP value, right? So that means that we have great intact barriers. However, when we apply G1, so that's our first blinded compound, we see a dramatic increase in that PAP value, suggesting that we're losing barrier integrity. And even at the higher dose, you notice that you don't even see a bar there. And that's because when you completely lose the barrier function, you're actually unable to calculate that PAP value. So when you look even at the 48-hour time point, both G1, the low concentration of that blinded compound, and the high concentration of that blinded compound, we've completely lost the barrier function, suggesting that the longer time points induce more toxicity. But overall, the blinded compounds, they did show different effects on the barrier functions. So one of the other assays we employed was the WST8 assay. So it's an indirect measure of viability. And same here, we actually found that there was just varying effects on the different blinded compounds. This was particularly in our OAT1 overexpressing cells. And there was an overall trend for the 48-hour time point to have a greater effect than the 24-hour time point. We also evaluated LDH. So this is a cytotoxicity measure. We know that when cells are under stress, they'll release LDH into the media. So we sampled the media from our blinded compounds, and it was a similar effect observed here. So just different effects for those blinded compounds that was measured by the release of LDH. So this table I just wanted to share with you to so that you could take away the fact that there were a lot of data points collected for this study. So over 2,000, I think we had over 2,600 actually data points that were collected for this study. So I would say it's pretty medium to high throughput evaluation. I definitely don't expect you to be able to read and interpret everything that's going on here. But I hope in the next slide, I can give you some of the insights that we took away from this study. So what we found was that most of the blinded compounds, that was 7 out of 8 of them, resulted in toxicity by at least one of the parameters or in some of the drug transporter interactions. So this is 87% of the time we detected nephrotoxicity, and this is 3-fold better than our current preclinical tests. We observed a decrease in barrier integrity, and that's often coupled with a decrease in cell viability, as well as an increase in LDH release. So we can say that the loss of the barrier integrity is directly associated with the cytotoxicity. We also found that prediction of nephrotoxic liability was highest when the results of multiple assays were combined. So cell viability, LDH release, as well as microRNA release. And then as we kind of were seeing throughout the slides, exposure for 24 and 48 hours provided sufficient, sorry about that, were sufficient to detect drug-induced nephrotoxicity in this case for evaluating acute toxicity. But I do think it's important to note that the platform can also be implemented for maybe compounds that require a longer or subchronic exposure. So I hope you can see that our proximal tubule on a chip is really a robust and transferable platform, which is going to be key for that broad implementation into industry. We're making use of some well-characterized proximal tubule epithelial cell lines, and we like that they're a potential alternative for the use of animal studies. And then finally, I really hope that I've convinced you that the NephroScreen, this high throughput proximal tubule on a chip, is a reliable, standardized, automatable system that was really useful in identifying nephrotoxicants and provides insights in some of the mechanisms of toxicity. So actually, I want to give you a little bit of a teaser and let you know, if we can go back, there we go, how we're building more complexity into our proximal tubule on a chip and modeling ischemia reperfusion injury. So in this case, we've included not only our RP-TEC cells, but also our HUVEC cells in the bottom perfusion channel. And we were able to mimic ischemia reperfusion by exposing the cultures to low oxygen, keeping them under static conditions and having them with low glucose. So we did that for 24 hours. And then the reperfusion part came where we basically just reintroduced them to normal conditions. So perfusion, higher oxygen, normal glucose. And what we observed is that in that ischemia reperfusion condition, we actually disrupted those proximal tubules. And we can see that by the fact that we lost, our proximal tubule cells. There we can see some DNA staining here. We also see some caspase 3-7 staining, suggesting that there's some apoptosis going on. But I think what was really cool is that when we applied adenosine to the culture to try and prevent injury, we observed that the adenosine actually rescued this injury. So lastly, kind of building even further on that proximal tubule, we know that the proximal tubule compartment is not the only part of the kidney. The glomerulus is also an important part of the nephron. So we've collaborated closely with Dr. Laura Perrin at Children's Hospital NLA, who's developed a glomerulus on a chip in the organoplate 3 lane. And we plan to transfer the media from the glomerulus on a chip to the proximal tubule on a chip, as similar as we can right now to be what's going on in vivo. And in our preliminary studies, we used membranous nephropathy as a way to first characterize the system. So membranous nephropathy is a rare kidney disease. It's an autoimmune kidney disease. where patients often exhibit loss of kidney function by seeing albumin in the urine. And these patients, they frequently have a lot of circulating anti-PLA2R antibodies, which are specific to the podocytes in the glomerulus. And so for us, and Laura has done this already, where she's been able to, in the glomerulus, model membranous nephropathy by applying patient serum, and we observe a loss of glomerular function. But our question was, how does this loss of glomerular function actually impact the proximal tubules? So if we look on the right-hand side, after treating the glomerulus and then applying it to the proximal tubule, we first confirmed that we have a loss of glomerular function when we applied the patient's serum to the glomerulus. However, after applying the filtrate from the glomerulus to the proximal tubule chip, we see also that the proximal tubules become damaged because we see an increase in KIM1 secretion. So with that, I really hope I've convinced you that MPS develops human tissue models for better therapies, that we're unlocking novel biology for better and faster assays to have an impact on therapy development. With that, I would really like to acknowledge all of my colleagues at MPS, as well as all of the folks that participated in the nephro screen. And I'd like to thank all of you for your attention, and I'm happy to take any questions.
Thanks, Chris. I think we have time for 1 or 2 questions.
Thank you for your talk. This is Ramin from Kyoto University. I was going to ask if you have investigated formation of bilayer epithelial endothelial until you showed the last picture. And I was wondering if you could make this bilayer more intimate, like culturing HUVEX right in contact with architects. That's question 1. Question 2, the difference between your OAT1 representing cells and the immortalized one. How much is the difference in amount of OAT1 actually?
Good questions. So I think there's a couple ways we can get those proximal tubules and the HUVEX closer together. So we can, and I didn't mention this, for our glomerulus model, we actually culture in one channel the podocytes and the endothelium. So you can kind of build a tube within a tube so that the cells are directly layered on top of each other. So I do think that that could be a potential option. Another one could be making that gel channel, perhaps a little bit smaller. Yeah.
Okay.
And then your second question, would you mind repeating it? I think you were asking the difference between the OAT1 overexpressing cells and the RP-TEC cells.
Yes, yes.
So it's primarily going to be the fact that OAT1 is very highly expressed in those overexpressing cells. I do believe they also have a very nice expression of PGP and MRP2. RP-TECs have them as well, but I'm not sure if you noticed in the table, actually, when we applied our known compounds, we had more of an effect in the OAT1 overexpressing cells than in the RP-TEC cells. And I suspect it's due to the expression of the transporters, higher expression of really, most of the transporters.
Very well. Thank you very much.
Yeah.
I have one quick question, if I'm allowed. When you did this round of the study, you had 2 labs checking it, which is a good idea. Did any of the pharma partners actually set this up or try it?
To my knowledge, they did not, no. Yeah, but that would be interesting. Will they? I don't think they're planning.
And I mean, did you get any information about the 8 compounds? Because they're obviously positive nephrotoxins. Are they clinically positive, only positive in animals?
So I think a little bit of both. And actually there were some in there that weren't nephrotoxic. There was at least 1 or 2, yeah.
And you flagged them up as positive?
So all but 1, yeah.
Interesting. Thank you. We think we should move on. So.
Yeah. All right, so before our last speaker, we've been asked to make a couple of announcements. So one is tonight there's a party and there is going to be a shuttle bus to take you to that party. You're supposed to leave at the Hilton entrance at 7:45. So if you want to go to that sunset party. And then the city tour that was on the schedule, there wasn't enough interest in that, so they've canceled that. And so everyone has 2.5 hours of free time. after this? Hmm? Not right after this, sorry. Don't leave. No, there's another session right here. So finally, our last speaker will be Arunima Arunuma, sorry. And she is from Switzerland. I'm going to leave that because it's ARTORG. I'll let you define that. So thank you very much.
So good morning, everyone, and thank you for having me in this very interesting session. I'm a postdoc in the University of Bern working with organs on chip models, and I will give you a little bit overview on the inhalation possibilities with these devices. Okay, so we heard from a lot of keynote speakers already that how difficult it is to bring a drug to the market. And in this laborious process, of course, we start with thousands of compounds that are researched in laboratories where a very selected hundreds reach into the preclinical development phase. And after the biological effectiveness is established, which is mostly via animal studies now, a handful of them reach into the preclinical development or the phase trial phase. So that is how the pharma is a bit in crisis now. So if you see the graph on the left, on the x-axis, it is the number of drugs that could be brought to the market with $1 billion US. And in 2010, not even a single drug could be brought with that amount of money. Moreover, it takes around 10 to 15 years for a drug to enter a market. And in today's time, it will cost around $2.5 billion US. So if some of you are into cars, you can probably get 16,600 Tesla Model X with that amount of money. So just to give you an estimate on how huge the amount is. And if you look into the numbers, the respiratory field faces the most amount of attrition rate. And this is mostly because the crucial basic scientific discoveries, they fail to integrate into the drug development pipeline and are often lost in translation within this valley of death. So currently, to study the lung, there are these 4 broad categories of models existing. On one hand, exploration of the physiological functions, or if you want to have systemic interaction between the organs, of course, an animal model is essential in that case. But on the other hand, they also do not exhibit the same kind of pathologies that we have, and they're also questionable ethically. For ex vivo, the precision cut lung slices have garnered a lot of attention lately, and they are excellent models that represent the native lung tissue architecture. But on the other hand, the scarcity of the animal or the human lung tissues makes it a little bit difficult to scale it up for high throughput studies. On the other hand, classical in vitro models are very high throughput, but their translation to the clinic is not always the best. So what if we now combine the technologies that we have currently and integrate biomechanical, biophysical cues into these complex in vitro devices? And this is of course possible with MPS systems, and there can be with organs on chip or even in transfer formats. I will mostly focus on the lung on chip with this. So now if we look into the current drug development pipeline, we arrive at a scenario where we can take a much more stratified approach rather than a unidirectional one, and rightly integrate these complex technologies within the drug development pipeline, where we have the ability to include patient-derived cell materials, the blood flow, air-liquid interface, and so on. Just to give you an overview, so organs on chips were developed around 12 years ago when scientists really combined the fluidic technologies, 3D cell culture techniques, and analytical methods, and to represent a smallest functional unit of an organ. And if we look into the building blocks of what is required to make an organ on chip or even a lung on chip, and if we look into the engineering parameters, so as a function of a tissue is to really provide a relevant atmosphere or environment for the cells to maintain their specific function. Here, the material properties, geometry, and the flow conditions needs to be accurately controlled. On the other hand, for the biological parameters, the most important consideration is, of course, the source of cells. But secondly, the growth, differentiation, and culture of these cells also require a thorough understanding on their different biomechanical and biochemical cues that are required. So we saw a lot of parameters that we can play with, really, to have a very complex model for the lung. But it's also important to keep in mind that there are several models, and they would each represent a different function of the lung. And it really depends on the research question of interest that we have in mind. So I would discuss maybe 3 lung-on-chip models that are robust and now commercially available. So the first is the first lung-on-chip that was developed by Hu and Ingber in the Wyss Institute. We have heard a lot about this device already. It's commercialized by Emulate. The membrane is quite thin, it's around 10 micron, and they can have stretch with the lateral channels on the ends of the size. Next, we have the organs on chip from our group in the University of Bern. It has 6 independent alveolar units, and currently the modified version has 12, which is now commercialized by Alveolix. The membrane is quite thin, it's around 3.5 micrometer, and we can replicate 3-dimensional stretch by using a microdiaphragm. So I will explain in detail a little bit later in the talk. And finally is the third one is from the Diamond MART, and it's commercialized by Dynamic 42 now. The membrane is around 12 micron thick, and you can simulate the blood flow in the basolateral side. However, the implication to add include stretch in this device is not possible currently. So coming to the Alveolix lung-on-chip system, how it looks is this plate is in the format of a standard 96-well plate. It fits right into this black docking station, and the breather and the exchanger have really intuitive touchscreen-based system. And with the plate, it has 2 modular chips, which has 6 independent alveolar units. Each membrane is very thin, porous, elastic, and is made of biocompatible silicone. And what was important for us is to really replicate the dynamic lung alveolar barrier. So for this, there was an inclusion of the microdiaphragm, which is connected with an external electropneumatic device, which is called the breather on the left-hand picture. And this really helps in the deflection of the membrane, which has the cells in it. So the cells are exposed to physiological levels of cyclic stretch. They're at booth 124 if you want to really see the system in person. Now, important was also to establish a relevant cellular model. And of course, with alveolar epithelial cells, it's important to understand that it's not readily, we do not readily get primary cells that often. So that's why we used this immortalized human alveolar epithelial cells. The paper was recently accepted and you can find it. So these ASIC cells grew really nicely. And what was important for us to validate was to see if they can form a really tight barrier and they could be cultured on the chip for long-term conditions. So in the picture you see it's around day 29 on the chip. They see display a very robust and tight barrier. They have very nice tight junction proteins with the zolenoccludins. And we also measured the transepithelial barrier resistance to check if they have a tight barrier. Which is very important for toxicity studies because that is the disruption of the barrier is the first real hallmark. And we did it in 2 different conditions. So in submerged conditions, we could see that the cells could maintain a very tight barrier, about 1,000 ohm centimeters square. But also in relevant air-liquid interface culture conditions, the cells could also maintain a very tight barrier and could be cultured for until a month on the chip. And next, we also wanted to see if these cell lines could produce the relevant or express the relevant biomarkers which are needed for the specific studies. So if we see, so we checked some of the epithelial markers, 81-like markers and 82, and we can see already that they express also in gene level, which I haven't shown here, some of these specific markers. And interestingly, they also display some of or express some of the 82-like markers like SPC and APCA3. And the SPC expression was also increased when we keep these cells an air-liquid interface on the chip. And since this study was performed during the COVID pandemic, we also checked if these cells could be used for COVID-19 studies. And they do express all the 4 important host cell factors that are required for the SARS-CoV-2 infection. Namely the ACE2, MPS2, furin, and NRP1 in both gene and protein level. And if you are interested to know more about these cells, we have a poster at 162. You can ask more or have more information on this cell line. So next, it's important that we are exposed to, due to the growing urbanization, that there is an increased emission of particles as byproducts into our environment. And inhalation is the major point of entry to our systemic circuit for these inhaled toxicants. So particles less than 1 micrometer, they can gain access to the distal lung and reach the lung alveolar barrier, whereas particles from 2.5 to 10 micrometer are really stuck into the upper airways where they have the cells blanketed with the protective mucus. So now with the surge of exposure devices that we have, it makes sense that we combine them with the realm of the lung on chip systems to really have robust platforms for inhalation toxicity studies. So coming from this, we use this Cloud AX12 system, which is developed by Vitrocell specifically for the AX12 lung on chip system. It has a nebulizer on the top, which uses vibrating mesh technology to really make a cloud of the aerosols from the liquids that we put on top. It has this vacuum exposure chamber. And the interesting thing is that they have a quartz crystal microbalance, which is integrated within the system. So this allows us to have real-time dose deposition exposure so we can calculate the amount of dose exactly reaching the cells. And yes, they're also at boot 113 if you want to see the system. So to validate the system, we wanted to use a potential toxic compound that has a human relevance. And here we use the polyhexamethylene guanidine, or PHMG for short. It was used as a humidifier disinfectant in Korea for a long term. And it was recently around 2014 that they banned this substance because they were causing an unknown lung injury and acute toxicity. And from then onwards, this compound is heavily used for toxicity studies. So what we see is after 24 hours of nebulization of this compound, the cell barrier looks absolutely disrupted comparing with the control samples. And we wanted to make it a little bit more complex to study how this affects it. So we have now differentiated THP1 macrophages within the system on the apical side, human alveolar epithelial cells, the immortalized ones. On the apical side and on the basal side, we have human lung endothelial cells. So now we subject this cellular co-culture to the PHMG, and we tested 3 different conditions. So we tested it in the standard submerged culture conditions. We nebulized them in air-liquid interface, and then we combined air-liquid interface with cyclic stretch. So I would say that within the submerged and air-liquid interface conditions, we didn't see really a significant differences when we nebulize the compound. But it's so interesting that when we combine it with cyclic stretch, we see a massive increase disruption of the barrier. So starting as early as 4 hours, and it's very consistent until 24 to 48 hours. So this really suggests that the combination of these relevant parameters like cyclic stretch and air-liquid interface is really important because it sensitizes the barrier to these compounds or these inhaled compounds. Furthermore, we wanted to also use or test nanoparticles if that's compatible with these devices. So we used healthy human, so for primary lung alveolar epithelial cells from healthy human lung tissue sections. And here we nebulize it with the zinc oxide nanoparticles, which is sometimes when the factory workers are exposed to a higher amount, it can lead to occupational COPD. So here we compared it with static and dynamic, and we really saw there was an increased cytotoxicity when we nebulized zinc oxide nanoparticles in the dynamic state after 24 hours. And furthermore, we also tested it with 2 different nanoparticles. So here we included titanium dioxide. And again, we use here primary human alveolar epithelial cells. And what was interesting was that after 24 hours, you can immediately see a drastic disruption of the barrier with the titanium nanoparticles alone. And what we also measured was the LDH release to measure the cytotoxicity percentage. So you can already heard how the LDH assay works in the last talk. So here, what we can find out is that when we are nebulizing with a high dose of the titanium nanoparticles, we see a significant toxicity in the cyclic stretch condition. But what we also see here is that we have an opportunity to also mix different nanoparticles in a physiological dose to have the same amount of cytotoxicity. So this also gives us a chance to think about including or mixing a different kind of nanoparticles, because of course in the air, it's not just one kind of particle that we are exposed to. So moving on, one of the common most irritant that we have in the atmosphere is the cigarette smoke. And when you're smoking, you're inhaling the smoke with your mouth, but then you're exhaling with the nose and mouth. So currently, we are trying to develop a cigarette smoke exposure model. And I would like to take this opportunity to tell you that the animal models that are currently in place is either you put the animal in a exposure chamber and their whole body is exposed to the cigarette smoke. And in that case, they have these systemic side effects, which is really not relevant. Or they expose the nose of the mouse or the rats to the cigarette smoke, which is also not quite relatable to the nose and mouth smoking that we have in the human. So this is very important to keep in mind for future model development, these issues. And I think since 85% of the heavy smokers develop COPD at the later stages of their life, it is really important that we know how the cigarette smoke affects on a cellular and a tissue level. So what we have here is a cigarette smoke exposure chamber called the continuous flow AX12 system. And it has a climatic chamber, so the humidity and the temperature is accurately maintained, and there is no variation from handling. And there is the AX12 lung on chip is rightly fits into the docking station on a heated chamber so that we can maintain the 37 degrees Celsius temperature on it. So I will show you some videos of the smoking robot. They have the option to have 10 different cigarettes into the module, and I can go once. And they really give us an opportunity to have a dilution or module for the cigarette smoke. So now I want to say that there are two standardized profiles for cigarette smoke exposure in vitro that are there in the field. One is the ISO profile and another is the Health Canada Intense regimen. And it has been seen in studies that if we compare the tar nicotine carbon monoxide profile of the human smokers, it relates more towards the Health Canada Intense regimen. But the ISO profile is still used to sometimes mimic more mild smoking regimens. So just for reference, it has 6 cigarettes, which are where 2 cigarettes are smoked simultaneously in one cycle. And what we wanted to see is really a very drastic difference in alveolar barrier disruption after 4 hours of the cigarette smoke exposure on the cells. And this was very important to see because this is also the first hallmark that happens when you smoke. Next, we also check into some of the inflammatory markers because they are really very nice prognostic or diagnostic tools to see how the ongoing inflammation. And we really was interested that this IL-6, IL-8 profiles was very relevant and could be seen in the COPD smokers from the clinical data. So increased IL-6 TNF-alpha has been shown in COPD as well as in smokers, which also we see in our in vitro device. And also the decreased IL-8 has been shown from COPD smoker patients, which also we see very, very nicely in our platform. So this gives us an opportunity to think that we can somehow mimic some of the biomarker expression with these in vitro devices. And furthermore, we also checked the reactive oxygen species generation. And we also see here that when we imply or include stretch, that somehow increases the sensitivity of the barrier and increases the ROS production 4-fold higher than when it's just in liquid interface and static medium. So finally, wrapping up, some of the next steps that we are working towards is, of course, we make it a little bit more complex. So we try to replace all the THP1 macrophages to actually use primary alveolar macrophages. Also, maybe at some point we include stromal cells into the system. We are also trying to set up that we use omics sequencing, so maybe bulk or bulk RNA or single cell RNA into the platform and really have a comparison on a genetic level. And of course, we started out just with the classical cigarettes, but this system or setup gives us an opportunity actually to also compare it with the vaping of the heated tobacco products. And I think this would be very interesting to see how they differently affects on a cellular level. And finally, with the Cloud X12, we are not only trying to just mimic a disease platform, now we are also trying to do some inhaled corticosteroid treatments to see if somehow that negates the inflammatory reactions with these. And lastly, I would just to say that for the next generation lung on chips, it needs to be more complex. It needs to be biomimetic. It needs to be also compatible with different laboratory equipments or the standard ones. And we have to try to be more high throughput so that it's relevant for the pharma at some stage. And finally, I think it's more of a team effort that where the clinicians, academic, industry, and the regulatory bodies come together really with a single goal that we can advance these technologies to precision and personalized medicine. So with that thought in mind, I would like to end my talk and really acknowledge my group in the University of Bern. Both Alveolix and Vitrocell, they have their booths, please visit them. And thank you all for attention and happy to take questions.
Thanks very much for an excellent presentation. Are there any questions?
Thank you for your nice talk. I'm Sakai from Tokyo University, Japan. And yeah, actually, the first question is about the cell line. I didn't notice about that cell line, but it's basically type 2 cell, right? Type 2 adipose cell.
It's basically type 1, but they also express a little bit of type 2 markers in the beginning, but when they grow on an air-liquid interface on the chip, we see that's more expression of type 2 markers.
Okay, how is the thickness of the cell layer?
Of the cell?
Thickness of the cell layer.
It's actually in a monolayer, so it's not the A549, it doesn't form that much multilayer, which is also a good benefit that it's really on a monolayer format.
Yeah, even in the monolayer, type 2, the thickness of the type 1 epithelial cells are very thin. Can that cell line realize such kind of normal situation?
It should, because we had done very preliminary studies on the thickness. It ranges from 10 to 15, but I cannot say for sure. But we have to check that again. But we did very preliminary studies. And you can check the monolayer, maybe on the poster, they have a picture for that.
Yeah, but please check because
of course,
because type 1 cell have a very unique, very large cell, but very thin, and showing very high TR value, very high TR value. Okay,
and we do see high TR actually in these cells.
And another question is that generally speaking, when you apply stretch movement, all the susceptibility against the stimulation are enhanced. So toxicity generally enhanced, according to your result, right?
We see very definitive cases that it is enhanced. Somehow it sensitizes the barrier much better.
And particularly in the toriculture, such kind of toxicity is enhanced by stretching. So it means that, as you said, cytokine, inflammatory cytokine are strongly involved, right?
We did check a very handful of cytokine inflammation cytokines with ELISA, and there is an increase. We haven't done a relief of full proof panel of the cytokines yet.
Okay, thank you. Wonderful.
Hi, thank you for the really nice talk. Some really interesting results there. I just wanted to ask again just about your cultures. So how long do you see, or how long does it take for your cultures to reach sort of what you see as optimal differentiation post-ALI? And then when do you use those cultures in your experiments?
So generally we see around day 14 to 16, but really ALI helps to differentiate them faster on the chip, that is for sure. But after ALI starting, it's usually 5 to 7 days that we see really that the SPC secretion is really high.
And I guess a follow-up on that, obviously you see your tier increasing. Was it over 28 days? So would you use it as far as that sort of final time point where you get the really high tier?
Exactly. So that is the idea, that we tracked the tier for long term so that we can have that plateau region where the cells are in the high tier, so that that plateau region could be utilized for when we have the disease induction or even the treatment induction, so that we really see if there is any toxicity, that we could see the barrier dropping.
Sorry, can I just, one more follow-up question now? I guess this is a wider question in sort of the lung field. Is high tier in an alveolar model a good thing? Do we want to see high tier, or do we want to be able to see permeability within those models? What's your opinion?
I think generally depends on your question. So if you specifically ask for toxicity studies, then yes. But if you're more looking into, because the primary 82 cells that we have from the human patients, they do not really have height here, but they are 82 cells. So if you're more focused on having really like a 100% 82 cell population in your model, definitely you have to go for the primary cells. But yeah, for toxicity studies, I think a high tier or a high tight barrier is really important to see any effects.
Thank you.
Thank you.
Great. Thank you very much. And before we leave, I'll just say thank you to the audience. So we actually get people in the end. And also a big thank you to all the speakers this morning. I think we had a great diverse set of presentations and actually real life use cases of these MPS systems. So from my side, thank you, and see you again in about 5 minutes back in here. So
Let's get started. So welcome everybody to the roundtable today discussion about regular acceptance of MPS systems. So we are happy that more and more people are joining. The doors won't be closed. Please come in after the very fantastic party last night. And we are hoping that to have an engaged discussion, a lively engaged discussion with you afterwards. So please stand up and ask your questions. So today we will try to look at the term regulatory acceptance from different angles to demystify also a little bit this term and look at this from the perspectives of the regulators, of course, but then also from the users like pharma companies, cosmetics, chemistry, chemical industry, agrochemicals, food industry. So is this, are there different approaches? Is such an approach, is it the same approach for all these different areas? So on the other hand, you know, looking into the submissions to the regulators, what could be obstacles? Why are not so many submissions currently getting through and more MPS systems are used in this context? So we will kick off the discussion with an example of a MPS system, which is the skin in vitro model, which is a regulatory accepted MPS model for a skin irritation assay in replacement of in vivo as an example, and to highlight the journey of such a regulatory acceptance. But before we go into this detail, I would like to welcome also the panelists, and I would like to ask them to briefly introduce themselves with their name, affiliation, and what is your connection to MPS. So my name is Stefan Kustermann. I'm from Hoffmann La Roche, and next to me as my co-chair is Donna.
I am Donna Mendrick from NCTR FDA, and along with Susie Fitzpatrick, we co-chair FDA's Alternative Method Working Group.
So is this on? I guess not. Yeah, okay, Susie Fitzpatrick, FDA, as Donna said, co-chair of the Alternative Methods Work Group, which is all of FDA.
Jean-Loup Dorn from European Food Safety Authority. Connections with MPS is the fact that EFSA is working to implement the use of new approach methodologies in risk assessment. Thank you.
Hello, good morning. My name is Anne Gourmelon. I work for the OECD and more specifically the test guidelines program. And my connection to MPS is that we are trying to evolve our products, our practices to uptake innovative methods in standards and guidelines and frameworks.
Good morning, everybody. My name is Maurice Whelan. I work at the European Commission's Joint Research Centre in Italy. I'm head of the Chemical Safety and Alternative Methods Unit, and I'm also head of the EU Reference Laboratory for Alternatives to Animal Testing, better known as ECFAM. And our role is really, we're part of, let's say, the risk management and policy part of the European Union. And we offer scientific advice and support to risk managers and the policy makers, and in particular, we're promoting new approaches to risk assessment.
My name is Seiichi Ishida from National Institute of Health Sciences in Japan and also the Sojo University. And so my scientific interest is the hepatotoxicity and also the regulatory sciences. And I'm being involved in the Japanese MPS project, and I'm organizing the standardization program. Thank you.
So as next, I would like to ask Maurice to start the presentation. I don't know, can you pull up the slides from behind?
Okay, great. Well, my job is just to kick the proceedings off by telling you a story about the very first MPS to gain international regulatory acceptance for toxicity testing, the reconstructed human epidermis. Next slide, please. Oh, I have to do it. So this is a reconstructed human epidermis, close up of it. Basically made from primary human keratinocytes. differentiated into 3 layers of the epidermis, the basal layer, the spinous layer, and the granule layer. And then on top of that, you've got that intercellular lamella made up of, the lipids you find in the skin. So, a real work of science and a work of art, really. So this has become, a key component, a model, the test system in a number of toxicological testing methods. It started out looking at skin corrosion, then developed into a method for irritation as well, for phototoxicity, and then more recently for testing chemicals for their potential to cause skin allergy or sensitization and genotoxicity. Now, they've also, these methods have become international test guidelines at the OECD. So for those you don't know, like what's an OECD test guideline, why it's important? Well, there are 38 member countries in the OECD, and an OECD test guideline is really an agreement between these countries on these methods as being standard methods that underpin the mutual acceptance of data. So if you produce toxicological property data in one member country, it'll be automatically accepted in another country. It also forms the base of being able to carry out these tests under GLP, which is very important for regulatory needs. So test guidelines are really the highest bar, you'd say, in international acceptance. But what we also have emerged too was performance standards to basically speed up validation of methods which are what we call me too or similar. Because in TG 439, the one on skin irritation, for example, there are actually 7 different methods, 7 different models, 7 different commercial suppliers of those models, all incorporated into the same guideline. So all these methods produce the same information, but in different ways. These then have been elaborated, particularly the skin corrosion and irritation have been brought into testing strategies in IATA so that really you can do a sequential testing and assessment approach for full characterization of your compound, and particularly to help you, for example, decide what class of the GHS your compound would fall into. And then what was important too in more recent years is to develop what we call a good licensing practice. And this guidance document 298 really describes how we can get that right balance between providing accessibility to all these guidelines in different countries by different users, but maintaining the possibility, of course, that there's a commercial activity to provide the models and the methods and so on. As was mentioned, this was kind of groundbreaking because it was really the first replacement method to corrosion irritation for a 3 hours method. And there was, really strong societal driver. This story started, during the 90s, late 90s, early 2000s. And that was even before in the EU, for example, we had the marketing ban on cosmetics that have ingredients tested on animals, which really kicked in 2013. Validation was a central element to all of this, and really the driver in Europe in particular was to valorize the output of research projects. So there was a lot of research money being invested in developing these models, both privately in industry, but also through European funded research programs. This was significant commitment from multiple parties, and it also involved a lot of international collaboration, particularly between partners of the International Cooperation Alternative Testing Methods, of which we're a part. One very important aspect during that whole validation period was ensuring consistency in production of these models. And that involved agreement on particular technical scientific criteria that producers could really use to ensure consistency in batches, consistency across production sites, and consistency internationally between countries developing these models. And that led also to allowing to apply quality systems like good manufacturing practice. All of this also was a strong driver of innovation. So as more validation and standardization was being carried out, this was also helping drive the innovation of the model itself. So over the years, we've seen other functionality, let's say, added to these models, which includes, you know, the metabolic competence, immune response, full thickness models have appeared now, much more high content readouts like transcriptomics and so on. So we've seen this continual evolution of the model itself. So here's some take-home message, I suppose. First was that, look, validation was the key driver behind optimization, standardization, building confidence, gaining acceptance ultimately, and very much driving the MPS model development, both from the, let's say, the scientific sophistication of the models, but also in being able to produce, you know, reproducible, reliable models. I mentioned the importance of this emergence of performance standards. So because with particularly there was you know, a number of different producers, a number of different models, all wanting to measure, you know, skin irritation, it meant that we needed to find a way of being able to allow this variety of systems and methods to coexist, but all conform to, you know, the same standards. And I think that's something potentially that as we move forward will be an important aspect to embrace the diversity, but equally ensure confidence and so on. The other thing too is, look, it did involve major investment from public and private sectors working together. You don't do this on a Friday afternoon. It required significant concerted effort and investment over time, but the dividends are high. And finally, I'd say, you know, it was an incremental process that picked up pace as more confidence was built. So as I said, we started, you know, with the first kind of output of research results in 98, 99, the late 90s, and then that triggered the validation. First validation study was skin corrosion, which led into the skin irritation validation. And, you know, that led in then to other methods being validated against the performance standards. And those guidelines have actually been updated 4 or 5 times and would know better over a decade of time. So as the science and technology moved forward, these guidelines then were then updated to incorporate these developments. So I'll stop there. And I just wanted to mention, if you want to read more about our validation work in particular and what we're doing, developing and promoting non-animal approaches, we've just published our 2021 annual report. I have a few copies here if anybody's interested, but you can download it from our website. And the other thing is we also have a tracking system for getting information on methods which are, you know, undergoing some evaluation or validation towards regulatory acceptance in very different contexts. And it's called TSAR, T-S-A-R. You'll find details in the report. And you can have access to all of the historic validation reports and independent peer review opinions and so on of these methods. So thank you very much.
Thank you, Maurice. So one of the topics that Stefan had introduced very early on, and we want to make sure is clear in this meeting, is different product types have different regulatory backgrounds. So for example, in FDA, there are obviously multiple regulatory centers, each work on their own regulatory laws. So there is a difference between trying to look at an alternative method, for example, from foods, from drugs, from chemicals, et cetera. So I think I'd like to ask Susie to start off the question, which is, you know, how are the approaches similar and different when you go across product types?
Is this on? Okay. How is what similar, different?
What's similar and different between different product types?
Well, obviously, foods is an area that touches you every day, every American or everyone in the world every day. And so food safety is an important area for us. And because we have not very much regulatory authority over it, looking at new approach methods is very important for us for chemicals. They're really for chemicals. And that's why we, Jean-Luc and I, are both part of what we're putting together an international collaboration on looking at introducing NAMs to the chemical sector, which I'll make a little commercial at the end of this meeting for everyone expanding themselves out of the drug box and looking at the broader picture of chemicals. So I think we need them, I think, more than anyone, and we're hopefully working internationally to achieve that. You might want to add something.
Yeah, maybe just something to add regarding chemicals that don't belong to anybody, to industry, contaminants. Nobody is necessarily dealing with them. So maybe these methodologies are very useful. We don't have any in vivo data. We may be able to compare with historical data on related contaminants, but maybe comparative studies might be useful in this context.
Any other opinions?
So the big difference between the chemicals and the pharmaceutical is that we have the human data in pharmaceutical field, but we don't have the, we only have the basically animal data in chemicals. So we need to think about what is the standard for each field.
Yeah, and yeah, another difference, I guess, between pharma and chemicals in general, be they pesticides or biocides or cosmetics or industrial chemicals, regarding we are talking about the regulatory acceptance. I guess for the pharma sector, you can accept high cost of MPS, and that goes with the innovation, right? But in the chemical safety testing, I think that can be a barrier to the uptake of innovation. And that's also one reason why it's taking more time to reach the regulatory acceptance until you have innovation that has a business model that is sustainable for testing a large number of chemicals.
I suppose I can add to that. I mean, differences. One is what chemical entities are actually subject to regulation. I mean, there are hundreds of thousands of chemicals in circulation, but only a very small amount of those are actually subject to regulatory oversight. That's the first thing to take into account. Secondly is, depending on the sector, there are different information requirements. In some sectors, there are actually no information requirements. Agencies have to make do with whatever data they can get their hands on, but not necessarily from a registrant. Whereas in sectors like pesticides or industrial chemicals, there's a requirement for companies, registrants to provide certain sets of data. And that amount of information can vary depending on different factors. In Europe, for example, for industrial chemicals, it depends on the tonnage of the chemical that's produced or imported. Bigger volumes of chemicals in circulation, more information is required. In pesticides, you have a complete package of data that's a requirement for assessment, whereas in cosmetics, it's only certain types of ingredients like hair dye or UV filters that are subject to scrutiny. Otherwise, the companies are trusted to carry out a safety assessment internally. So the other thing that varies significantly, and I think people don't realize this, is the actual assessment processes vary between sectors and agencies. So if you're talking about industrial chemical, like Anne mentioned, where money is tight, then it's much more of what some people call a tick box, you know, a standard information requirement that's satisfied with a standard method, and trying to make the whole process as efficient as we can. Whereas if you're talking about a pesticide active ingredient, they are much more sophisticated assessment processes where you've got a lot of experts sitting around for a long time discussing all kinds of data, whether not only the standard required data, but also any data taken from the scientific literature. And the final thing I'd say is don't forget societal concern. There are certain toxicological endpoints that we're as a society more concerned about, like cancer, for example. And also we have sensitivities when it comes to you know, the type of information, how it's generated depending on the product we use. And we all know we have a cosmetics ban in Europe, and but we don't have it in other sectors because that reflects societal concern, and we shouldn't be dismissive of that either. You know? So thanks.
Thanks. So I'm hearing that first, the different areas, they desperately need the MPS models because this is a good way to sort out a lot of things, but then there's also different regulations in the different areas. So getting back to the example Maurice has presented, I'm still wondering, being of course very biased from a pharmaceutical company perspective, I'm still wondering, is that Is that then in the end the way we should always aim for? I mean, or are there other ways, I mean, to gain regulatory acceptance? Do we always need to go the full way until having that qualified and validated the systems, or are there also case-by-case examples? And could you think of them to ease also the way for the developers and the applicants to submit their data to the regulators? I don't know who wants to comment on that.
So, I mean, at FDA, people are very interested in seeing new types of data. And a lot of applicants do come in, from my understanding, talk with Cedar colleagues and others, they do come in with new types of data. The question is, when do you want the FDA or even yourselves to take regulatory action based on that data? And are you trying to replace an assay? So the barrier is different depending on what your purpose is. You know, again, if you're trying to replace an animal study, obviously the barrier to acceptance for everybody is not just the FDA, but your colleagues within your companies and everybody is going to be much higher than coming in with an assay saying, here's a supplemental piece of information. And again, people do that all the time. So you don't have to wait till regulatory acceptance to start sending in new types of data. You know, in fact, you can validate an assay within a company and be specific to the company. It's just usually takes a lot of work, so it tends to be more consortium based a lot of times. But again, don't think you have to wait until there's some gold stamp that says now it's validated, now I'm safe to come in, because that's not how this is going to work. It's not how it's worked with other types of new technologies. You know, people start adding data slowly. You know, for example, they're trying to explain away a problem they've seen, might use the supplemental information. Again, looking at efficacy doesn't usually have as high a bar as safety. So all different ways that you can start submitting data to regulatory agencies and get everyone comfortable, getting your own companies comfortable submitting data and getting the regulators comfortable looking at it.
Yeah, to add maybe to that, in the chemical sector, I guess regarding standards, we stopped a bit talking about replacement and cancellation or ban of animal testing, because we realized that innovative methods, as they become new standards, new guidelines, that they can continue to coexist with animal testing. And it's not really a problem as long as the innovative method has well-defined applicability, and the regulators know exactly what they can do with it, what they cannot do, what are the limitations, and then it's fine. And so replacement will come, but we need to allow pioneer methods to come through, to become the new standards, and little by little, we expect that, of course, animal testing will become obsolete.
I might add to that. Well, yeah, first of all, I heard there was a lot of emphasis in this meeting about the replacement of animal testing and animal. Which, I mean, obviously, I'm head of ECFAM, and we get our mandate from the European Directive on Protecting Animals Used for Scientific Purposes. So don't get me wrong, that's a major policy. But there are major other drivers as well that we should be talking a lot more about. First, as I said, addressing disease burden in our societies. I mean, we have hundreds of billions being invested in Europe at the moment to beat cancer. That's our Beating Cancer plan. And we all know in certain contexts, chemicals contribute to the burden of cancer, but only a fraction of the chemicals in circulation in the EU are actually assessed in any way for carcinogenicity. Why? Well, because it's too expensive, takes too long. It's an economic barrier as much as it's a scientific barrier. The other thing is, we want our industries to do well. We were hammered after COVID, and now we're, heading into another economic crisis caused by a terrible war. So, we're all looking to find ways of making our economies efficient and effective and competitive and facilitate trade. And the other thing is, we want to move into an era of safe and sustainable by design. We don't want to be putting carcinogens and skin sensitizers into consumer products. And we're doing that a lot at the moment. And so I think we need to, and of course, we want to move away from animal testing as well. So I think that's the important to, when we think about acceptance and what does acceptance mean, It's way more than purely showing the scientific validity of something. It's fitting it in within a whole, a value added proposition, But I would say about in terms of acceptance, I mean, I always struggle to find language to describe this, but I'm going to give it a shot. What I described there was what we call universal acceptance, and it was acceptance of methods, not data. It's acceptance of methods. The bar was so high because at that level of acceptance, those test methods can be used in multiple sectors, multiple countries, multiple uses used by multiple agencies. It's the highest. That's it. It's a gold standard. Not because of the method itself per se. It's golden because it is a universally accepted standard. Okay. But then you have acceptance in specific contexts of use. And that's why often I think you see this contrast between, in the pharma, it's all context of use. Of course it is. But in the other sectors, now keep in mind, pharmaceutical sector rely heavily on OECD test guidelines, particularly for their preclinical approval. So, I think, there's this artificial separation happening there. I'd have to push back on Donna and say, look, validation is an intrinsic part of the scientific process. For you to yourself to believe your own results, you carry out validation. I hope you do anyway. To convince the colleague down the corridor, you do validation. To convince your peers at this conference, you're doing some kind of. So it is an intrinsic part of the scientific process. So I would say to you that the same principles that were followed in those validation studies are the very same principles that a pharmaceutical company making an IND submission or whatever to a regulator on a very case-specific, context of use is showing qualification data, is showing validation data. It's just done within in situ confidentially, and it's only good for that particular context. So, scientifically, let's say there's no difference. Everybody's still having to do validation and qualification. Just the difference is that universality of acceptance then. Okay, hope that helps.
So let me add for chemicals, a lot of which don't have an owner to mention, and a lot of which are in food or water that you're exposed to every day and your children are exposed to. It's not acceptable if we find, we already know in advance, we know that animal studies aren't the right tools for us, not saying they don't give good data, but saying it's not acceptable if we see something in your children's food and we say, as a regulator, we'll be back to you in a couple of years and tell you whether that's good or not, because we have to do an animal study. So we're looking at a lot of mechanisms to work with the industry to help us get new tools. We created a public-private partnership with which the food industry, the Humane Society and others are there to talk about what new approaches we can use for chemicals. We're talking internationally with it. Donna and I have a seminar series through the Alternative Methods Work Group. We invite people to come in and present your new methods, and sometimes we adopt one. We've adopted one. I know our agency has adopted one to look at a better way to look at ADME. So I think there's a lot of ways to work with the agency. Our agency, I know you can work with the other agencies through Talks 21 to work on this together as more of a partnership now than it ever was before to bring these new methods. Because we, as a regulator myself, I recognize I need new tools to do my job better, and that's what we're going to encourage everyone here to.
Yeah, maybe Stefan started the discussion on the fact that we need to demystify what is regulatory acceptance. The thing is, regulators are two communities, and it's first the risk assessors who do the assessment of the chemical and the risk managers who write the way it should be done. The issue also is the fact that it's set in stone that we need animal studies. So in order to change this legislation, maybe what we want to do is to start simple and, let's say, to be practical, we have the hazard identification, we have the hazard characterization, we have a list of endpoints that are set in stone for genotoxicity, as an example that Maurice just gave. We have liver toxicity or whatever. We could look across these different endpoints and compare them with the traditional way they've been set, let's say for genotox, AIMS test or other genotox tests, use MPS and compare for chemicals for which we have the data across for a range of endpoints. Then then the most difficult question would be to derive a safe level using MPS. So hazard characterization, dose response modeling for a particular chemical. But again, we can compare that with historical data. And maybe starting simple like this could lead to demonstrating that we can use such data sets and see how far they are from the traditional ways of doing it. Thank you.
Excellent. And really, animal tests are, for a regulator, just a tool to get questions answered. And so if we look at the questions we want to answer from those and say, do I have a new way to answer that question? And for this product that I'm looking at, do I really need that question answered? It depending on how you're using it, you know, and how much confidence I need in the answer. If I'm looking at it for priority setting, I can take a lot more uncertainty than I can for a pivotal study, but it's really just answering questions. All the animal studies are, in my opinion, and we need to look back at what we want answered, what we thought it answered with that study, and can we answer it a different way, you know, as good or better than the way we're using it now.
Maybe just a couple of last two things.
Yeah.
What we want also to do is to structure the way we put all the evidence together using this so-called weight of evidence approach. And doing these comparative studies allows us to do that for all the different lines of evidence.
Right, that's right.
Okay, so I'm hearing there's multiple ways. It's not always the way of the skin irritation model we have to go to gain regulatory acceptance. So overall, I would say with that multitude of ways to get to the regulators, so where is then the gap? I mean, are you getting already enough submissions with regard to MPS model systems? Are you happy with that, or do you see certain obstacles which could impede this process? And what could these be?
I'd suggest to you what the issue is, and let's just stop talking about animal testing for a moment and go back to basics, which is most of the reasons why MPS per se will not be regulatory accepted. It's the methods that will be proposed or the data derived from it will be subject to acceptance or not. But the first thing is lack of purpose. I've heard the last couple of days, fit for purpose, yeah, fit for purpose. Very few developers actually are able to define the purpose of what they're doing. It's wonderful research, but it has no, from a regulatory perspective, it has no utility. So for example, we don't regulate chemicals in Europe based on liver toxicity. Information required, it doesn't exist. Liver toxicity is important, of course, but we don't. So there's the first problem is how in the name of God can we even talk about regulatory acceptance if we can't evaluate fitness for purpose because the purpose isn't clear, okay? The second thing is understanding the decision-making context. Vast majority of MPS developers who are claiming that, you know, want to have their method or MPS-based system considered for regulatory acceptance have no idea about how decisions are made in a regulatory context and make no effort to find out. Thankfully, you all came this morning, so that might show you're all forgetting. But seriously, how can you engage with the community and seek acceptance when you make no effort to understand all let's say the relative complexity of regulatory decision making? By the way, all the information is available, etc., etc. The next is once you get to that level, so you think you have got your purpose right, you think you actually understand, you know, generally speaking, how decisions are made. Then it's a good time to start the engagement and have a discussion. Because at that point, you begin to realize that regulators and the regulatory community, and I include myself there, don't have all the answers. Because what we're proposing to do is use a completely different technology, new types of methods, new types of information to make decisions where we've been using completely different type of information. So asking a regulator, what do you want? Tell me what you want. It indicates to me somebody who hasn't made the effort to try to begin to understand. Because then at that point, you start the co-design, co-creation process. Because the first step in that is the co-formulation of the problem. How can the decision-making community that's using the information from the community that's generating the information, how can we formulate the problem in a new way that will allow us to all to make progress? And that's why I keep saying about the animal testing, even though we want to move away from animal testing, of course, is that, oh my God, how many people who are developing new technologies, including MPS, are trying to replace a 90-day rodent study?
they're formulating the problem as we need to replace a 90-day rodent study. That's the wrong formulation. And already you can see that we run into a problem in terms of how anything could be accepted.
So regarding to your comments, so I have a question that which sector prepare the experience of using the new method? So it's the industry side prepare the basement of the data for the method assessment, or the regulator also get the some result to get the feeling of the new method. How do you think? What do you think?
Yes, me.
I guess this was now also the invite to the audience. So to engage with you, what are your questions? Come up and ask. Don't be shy.
Can I add something? I mean, sometimes as a regulator, we go out and look for new methods. So we had thought the neuromuscular junction on a chip would be good for Botox testing. And we at FDA felt we had a way to qualify it. And we went to Dan Tagley to give us assistance on it. And he's just announcing a program to work with him and with the FDA for a chip that we think will replace a very bad test. So we're open when we see needs that we have, we're open to looking for other ways to do it too, through our partnerships with EFSA or PPP or other things. So it's not always you're coming to us. We're sometimes going out and, well, we started the whole chip program that way by saying, hey, we do need this. And we're going to work with NCAS to try to develop what's here today.
I was talking with Laura Suter-Dick earlier, and we mentioned the fact that a lot of the people working in this technology originally were working in toxicogenomics, which again reminded me that, there's an excellent book called Crossing the Chasm, which was written originally for the computer industry, but it talks about these early adopters and these people that are interested in new technologies. And there's this big gap when you try to move to the majority of people. So don't make the mistake a lot of us made in toxicogenomics where, you're talking among yourselves with early adopters in your company, and you're not talking to the basic regulators, because, you're not going to get your company to move in this direction if you can't get the heart and soul of the regulators and the more conservative people in your company bought into these technologies. It'll just stop for quite a while. Yes, go ahead.
Hi, Katie Conley, Humane Society of the United States. I'm just curious whether the regulators play a role in incentivizing the companies to get the data in front of you. I'm here, definitely hearing that we accept it, submit it to us. How can we incentivize? Is there a way to do a shorter time frame for getting through the approval process or something like that would incentivize?
I can come a little bit on that. I just want to be also clear. It's a debate, isn't it? So I'm throwing things out there. Is having the opportunity to submit is not the same. I'm afraid, Susie, of saying that somehow then it's implicitly accepted.
Right.
We have so in Europe we have very strong incentives, legal ones for companies.
Right.
In fact, it's a legal obligation to use an alternative approach if available. But for example, under REACH, there is, a legal provision very clearly under Annex 11. It's where you can, what we call, I'm sorry, I'm using lots of regulatory speak now, but adapting information requirements. So you can provide alternative information generated with non-animal approaches if it's considered adequate to satisfy the information requirement. But the vast majority of those submissions are rejected by the agency based on lack of scientific credibility, lack of documentation, lack of whole lots of things. So there's an example where we have a full legal framework. You can submit away all the weird and wonderful data, but very little of it is actually accepted. And I'd be curious to know in other agencies, particularly in the US, is that really the case? because I wonder, the problem is in the you know, it's not my domain specifically, but in the pharmaceutical sector, when you're talking about companies' submissions, it's a confidential process. We don't know what's being submitted, and we don't know what we hear some success stories like what was announced yesterday, but they're you know, they're flash in the pans. You know, it's very hard to get a sense of that. But I'm glad Donna mentioned toxicodynamics. Like that technology has been, it's almost old, you know. There hasn't been one out of 27,000 chemicals registered in Europe, like 3 have omics data. And I know for a fact that omics data was actually never used in the regulatory decision-making. It was kind of just sprinkled on top. So incentivize, I mean, you know, even with legal incentives, it's still difficult to really make progress there, I think.
Thank you.
My name is Nordine Ashamake from Michigan State University. Thank you for the discussion. You covered drugs and chemicals, cosmetics. What about the food additives? How they are regulated, and will organ and a chip have a role there, given the fact we discover more about the microbiota and its interaction with different organ systems and up to the brain, and the influence of those food additives will become more important in the future. Thank you.
So your question is related to food additives and how they're regulated, correct?
Yeah, and would organ and a chip have a role there?
Yeah. So in terms of legislation in Europe, we have a guidance document which basically sets the scene for what type of studies are necessary. The first one is the food additive absorbed or not by the intestine? And then, of course, if it is, then you have the whole battery of in vivo data set. You can imagine that with the MPS, we could first look at absorption, and that's something we've been discussing a lot with Susie. And then if, let's say, it's absorbed, then we look at intestinal metabolism versus liver metabolism and so on, comparatively between rat and humans, but also the impact of the microbiome, if it's hydrolyzed or reduced or oxidized and so on, and potentially the impact it may have on the microbiome at certain levels of exposure. So potentially these MPS could be very useful in looking at these aspects. I don't know if I've answered your question, but I tried.
So in the US, most things come in through GRAS, food additives, so we don't get any data. Packaging, but you know, we already have things we're looking at MPS for mixtures. Food is a mixture. There's chemicals are a mixture. There's no good way to look at mixtures now. Can we use MPS and that developmental immuno? There's no good animal studies for that. Those are important areas for anybody. Those are areas so we're out looking. We're not sitting passively as a regulator waiting for things to come in. We are out looking for technologies. We have a plan we're working on with EFSA for NAM. So we're looking for things that can help us regulate our products better. And you know, maybe we need a better job of saying what our regulatory questions are to help guide the industry, and that's something we've talked about. But we're not. I don't think anybody in the FDA is sitting back waiting for things to come to them. Do you think, Donna? No. We want these new tools. We have invested a lot in MPS, starting at the very beginning and working with Dan to look at this technology to help us out. So this is something of all the technologies that we've really invested our time, a lot of time in across the agency.
Just one very last thing. We have a lot of historical data on food additive toxicity. So, and we have QSAR models that can help us to prioritize the way we're going to, we can use this historical data combined with QSAR predictions before we run the MPS experiments to answer specific questions.
Yeah, it would be great to be updated because, I mean, even absorbed or not absorbed, but it still will have an influence as we know today, like we know more than we knew before when this division has been put. Thank you so much.
I would add that all of the centers at FDA, the researchers are using different MPS platforms. They're using commercial ones. In some cases, they're developing their own. Each center is doing research on MPS. So again, we're not just waiting, we're actually getting involved ourselves.
Hi, I know this is too early question, but I have a question from the viewpoint of validation. To confirm MPS is very reliable or not, we have to use so many positive control and negative control compounds with already known mechanisms. And you mentioned about the regulation for food, drug, and chemicals are different. In that case, number and the variety of the compounds might be different or not.
There's, validation is science in itself, and there is extensive publications by many people in the room here. I've edited a book on validation of alternative methods, and in there you'll find a whole body of knowledge answering those kind of questions about what it would be ideal design of validation study, including chemical selection and so on. So, it
depends on the purpose of the MPS.
Well, again, if you look into the, proper approaches to validation, you'll see that one objective of validation is to define what we call the applicability domain. So not all chemicals can be tested in all test systems in all contexts. So often the physiochemical properties of the chemical that you're testing dictate whether it would be suitable. Again, if we talk from a mechanistic perspective, I mean, if you have a well-characterized MPS that what pathways, let's say, are expressed, then the chemical selection then will also be informed by the. We've run one of our biggest validation studies currently undergoing at the moment, where we're validating 18 different methods for looking at thyroid disrupting chemicals, and there actually the chemical selection was done with an international group of experts who were expert very much in the mechanistic mode of action understanding of chemicals. So as I say, science validation is a serious business, and I don't understand sometimes why really accomplished MPS developers who are obviously very clever, when they come to actually then address, actually demonstrating the scientific credibility of their MPS and their method, actually look like kindergarten kind of approach, So I'd love that the MPS community take, demonstrating the scientific validity and the utility of what they're doing to a decision maker would be. So plenty of literature out there, plenty of knowledge. ECFAM has been doing it for 30 years, so we've put a lot of, and I mentioned SAR earlier, our big massive database. Go there, download a validation study report of the many tens of hundreds of methods we've validated. Look at the scientific opinions, and you'll see there what is important to see in a good validation study. So look, if you're serious about regulatory acceptance or the information from your systems being used in any decision-making context, whether it's within industry, safety assessors, or whether it's in a regulatory assessment, then take qualification and validation seriously.
But the cost for validation is very important to regulatory acceptance.
Well,
right. So that's
there's
why
no point,
I'm so curious about the number of the.
Well, here's here. I did a little back of an envelope calculation that I did a calculation that out of the European money, I estimated over the last 20 years, we spent €1 billion on directly on alternative method development. Okay, €1 billion, between about 80% of that coming from public sources, 20%. Now that's the direct funding, not the indirect. And I estimate that probably about 1.2, 1.5% of that money has been spent on anything that would look like validation. I mean anything. I mean basic, like do your experiment twice type stuff. So I don't understand why people say who are spending, I just looked at a system in there, 150 grand for the couple of pumps and an $800 or whatever. Now, I'm not saying that they're overcharging, but any of you guys can afford that I think you could afford, investing a bit of, money in, yeah, you know what I mean, showing the reliability and the relevance of what you're doing. Sorry, I'm sounding very agonistic here, but I don't mean.
Yeah, thanks. I support everything Maurice said. And regarding the reference chemicals, I think there's a huge need for curated data sets of reference chemicals that are universally accepted. Not reference chemicals that are picked here and there to validate one technology or another or method, but there's really a need for having globally accepted reference data sets that then any developer can use and come up with their proposed solution. And so there's no need to invest for the developer to invest every time in, defining what is the curated data set of reference chemicals they want to use. And I'm saying that for complex endpoints, this is going to be what is limiting the emergence of innovative approaches is the lack of universally accepted reference chemicals for some of the complex endpoints for which we really need to have solutions in the future. We are starting to see some possible solutions, but yeah.
Otávio Presbrave from Brackman. Taking into account the theme of this roundtable, I'd like to say that me personally, I believe that the MPS is the most powerful tool we have nowadays. But I still have some problems in understanding the validation process of MPS. And especially I think that it's very important for the regulatory agencies to understand, they need to be confident about the powerful of the MPS and the meaning and how the results mean for them to register or not a product. And in terms of validation, I'd like to ask you, what kind of validation should we perform for MPS? Validate the methodology itself or the process, and if we can follow the guideline 34 of OECD for validating an MPS also. Thanks.
Yeah, there's not one size fits all response, I'm afraid, for that. I think the principles of the OECD guidance document 34 about demonstrating relevance and demonstrating reproducibility are universal. It's about the credibility, the scientific credibility of the solution you are proposing, be it an MPS or some other type of combination of in vitro, in silico method. And then it's, of course, the processes, how you perform the validation, you need to adapt depending on the MPS, the complexity of it, and but generally the principles remain universal, and this is not going to change. What may be adapted is, it depends if parts or elements of the MPS have already been validated in some other forms in the past. You use the existing information you have, and building on that, you complement with what needs to be added to create this confidence in the proposed solution. And I think involving the regulators is really important from the onset because it's a process, and the appropriation of the technology, of the innovation by regulators is really key. They are going to come and challenge, and this is normal. They want responses, they want answers to their concern, and only when they feel confident most of the concerns are addressed, then they can accept the innovation.
Can I add to that maybe? Just go back to what Anne said about the chemicals is extremely important. It's a good place to start. It's a key aspect. But there are lots of incredibly valuable byproducts of validation exercises over the year has been these highly curated chemicals lists, but very importantly has all the metadata about the chemical in terms of its properties, modes of action, etc. That can be used. And in fact, over the years, we've published several lists covering all different toxicological endpoints. Again, if you look in our status report, you'll find what we call a list of lists. We've got, I think, 85 lists of reference chemicals for all kinds of toxicological endpoints. And so I suggest please don't reinvent the wheel, use those for your own purpose. Just to talk, as head of BRACFAM, you should be asking yourself the same question in the sense of what's the best way to validate. I mean, look, validation needs to innovate just as much as the MPS, developments need to innovate too. And so we're always looking for ways of optimizing. Validation needs to be as fit for purpose as your MPS that you're developing. And so I would say to you, the first step is do something intelligent. First of all, you mentioned guidance document 34. Look, again, it's often you hear about this OECD validation. Look, again, I go back to my presentation. The aim of the game there wasn't to do the most costly, time-consuming and expensive validation ever. The aim of the game there was to develop an internationally accepted guideline that you can run under GLP and have mutual acceptance of data between 38 countries. Now, as a stepping stone to reach that highest level, there are expectations about the level of validity you would need to demonstrate, reliability and relevance. And a lot of people focus on the ring trials. Oh God, ring trials are so expensive, and transferability. The reason why that's such an important element at the OECD is not because somebody's trying to break your balls on things. It's because how can you deploy a method in 38 different countries, including Brazil, if you can't demonstrate that the thing can be reproduced between laboratories? So we often get this question like, oh, that's an essential. A second, what are you talking about? That's a requirement because of the nature of a test guideline. Do you know what I mean? It's not the universal requirement for validation. I'm very much a supporter of thinking about companies that have proprietary technology that use it in situ in one testing CRO. How can we discuss establishing the validity of those methods why would you want to do reproducibility between laboratories when there's no intention of and that's where I think performance standards become very important,
Thank you.
Yeah, Brad Calvin of Data Sciences. I just want to jump on the validation bandwagon here. Half my career was spent, or over half my career has been spent in the regulated side of diagnostics. So sitting on assay development teams, running R&D organizations. And so, I know what it takes to bring a regulated test out to market for diagnostics and the statistical rigor that must go through, and that development process from proof of concept to feasibility to development and then to verification validation. I think it's important to understand and sort of get the terminology right, because for most of the presentations that I've seen over the past couple of days, these are proof of concept, right? It's 6 compounds, it's 10 compounds, it's 20 compounds with maybe 2, that are so-called negatives for specificity. That's proof of concept, right? It's not validation. So I think it's probably worthwhile for a lot of the groups to get statisticians. There should be more statisticians here to show what validation really is. So I'd like to see what your comments are. Maurice, maybe.
So I guess I should ask, are you a statistician?
No, I'm not, but I've hung around a lot and I've had them on my development teams.
Okay, thank you. Very good point. So we're running out of time. I want to thank everyone It's a great session. We have two announcements. One is that the city tour has been canceled. So good news is you have 2.5 hours of free time. Tonight, there's a shuttle bus that's going to go to the party. It leaves a Hilton entrance at 7:45. And I do want to make a wonderful plug for Lena, who's done an amazing job getting this conference together. And I don't think she's been thanked enough. So thank you, Lena. Thanks, everyone.
Hello, everyone, and welcome to the session on computational modeling and AI in dialogue with MPS. My name is Nicole Kleinstreuer. I'm from the National Institute of Environmental Health Sciences. I lead the NTP Interagency Center for the Evaluation of Alternative Toxicological Methods. And along with my co-chair, John Wixwo from Vanderbilt University, we're delighted to welcome you to the session this afternoon. Hope you had a very nice big break in the middle of the day and you're excited as I am for this afternoon session. So our first presenter is Jari Haitanen from Tampere University in Finland, speaking to us about biophysics-based computational modeling as a tool for translational research and drug screening in MPS.
So good afternoon and nice to see you all here, that you managed to come here after this long break. Thanks, Nico, for introduction. So my talk will be more like a little bit like a story, how we have kind of done quite a long time of work on collaboration with stem cell scientists and especially on story about iPS-derived cardiomyocytes and how we have kind of combined in silico and in vitro. And let's see whether this works. Okay, no, that's small delay. First of all, we actually something like 60 people in University of Tampere, part of this kind of Center of Excellence and Body on Chip research, and even longer than this, work together. 6 PIs, 60 people, really spanning different areas from stem cell sciences, biomaterials, microfluidics, and myself, who is more like, in a way, computer science background, I would say more. And my lab is really doing in computational modeling, biophysics, in silico modeling, you could say, also electrophysiology, biomechanics, and being part of this, we are doing work in various cell types, but this story is really about cardiomyocytes. And there's a couple of reasons for that. First is cardiomyocytes is really, I would claim that the organ and cells that are mostly modeled. None other cell type is so well known actually on the basic functions as cardiomyocytes. And this is my kind of motivation intake on kind of personalized medicine. So we have these two areas that always people refer, like personal data, gene RNA, other proteomics. But then one area is computational models, and of course the fourth one is the MPS that we all are here for. So I would like to kind of claim that all this is needed for future personalized medicine. And I think most of us here agree very well. I don't need to stress it too much to really get to the patients. If you think about the modeling of human physiology, of course, the tradition has been that we have the preclinical animal and population level models for humans. And now we have the MPS really emerging. But traditionally, the modeling has been really on looking to kind of, I would say, the organ level, human level modeling, and a lot of work has been done on there. And now with the MPS systems, it's been opening us for us modelers a really nice way forward to look in the cell functions. Because nowhere else we have had this kind of nicely controlled system for computational modeling. We need data. And those of you who are making these MPS are kind of treasure trove for us to give the data so that we can learn more about the cell functions and have nicely controlled input-output relationship that we can then convert to computational models. And I hope that all this will lead that we can kind of close the loop as well and go back to the patient, the modeling on whole body level and maybe to the patient itself. Just a little bit about the in vitro side. So we have actually worked a long time with iPS-derived cells. One reason is that Professor Aaltosetälä, our cardiologist, was actually in California in the same lab where Professor Yamanaka was at the same time, and we got those cells quite early. But this just kind of, the video there just shows that beating cardiomyocytes And of course, you can add drugs and have the ECG of the cells, action potential derived field potentials, and see how it changes with the drug. You can also see, of course, that they are beating. So there's a contraction there, and that's one thing that I would like to address later. And just about the data. You need data for modeling, and if you would like to model the cardiomyocytes, you would like to get electrophysiology. Calcium is important as well because it drives then downstream the contraction. If you get the contraction, we have a kind of nice assay, and we have actually worked quite a lot on that. And this one example of paper where we show that you can actually drive all these three assays or measures simultaneously from the cardiomyocytes in vitro. So we have the data. And just kind of importance of this is that, for example, in certain genotypes, the effect what is happening there on mutation could be ion channel level, it could be calcium level, or here it actually on the action, on the contraction level, where the contraction mechanism is affected by this mutation. So if you try to model these, of course, these are the levels of modeling as well. Just to show that what the in vitro world is going on in Tampere, so we are really actually looking more on injury and ischemia and try to look for that. But anyway, it's kind of nice that we have these disease in vitro and you can model and look the mechanism as well. And of course, the question is it valid for the patient? And in a way, that's been here many times. This is the cells in vitro might not be really the same as in patient. Neither they are in cardiomyocytes. They don't have the structure. They lack certain kind of, let's say, mechanistic events. Some ionic currents are not the same. So really, sorry about this quite complicated slide, but this is a nice slide made by my postdoc, Jussi Koivumäki. And idea is that from the patient, we can, of course, get a lot of data. And traditionally, it was made for whole body modeling. And IPS, we get a lot of insight on how these cell works of the patient cell works, how the calcium contraction, action potential, other works. And to really close the loop, we need these IPS-derived cardiomyocyte models. They are not the same as real cardiomyocytes, but these are really the thing is that we are making model of the model. Your in vitro model turned to the in silico model. And if we have that, then we can really look inside the mechanistic things. Hopefully we can translate to the patient. I think to my knowledge, nobody has really done this whole circle. The pieces are there and hopefully in future we can. But just think about what these models are in silico. So of course they try to replicate the cardiac function, what we see. And if you think about electrophysiology, there are really a bunch of differential equations. Like here is one exchanger. Sorry, here is the color. There's this exchanger and you have the equation, nice equation there. And from all these, of course, you have another equation. A lot of parameters, a lot of equations. But these really define the electrophysiology event, and they can produce these kind of action potentials. And nice thing about that, of course, is that we have all the data, all these ion currents, all these opening and closing of ion gates dynamics, and we can tweak these and produce new set of models from different diseases, like for the different, let's say, like long QT is here example where you have prolonged action potential. We can add drugs, so changing these ionic currents, because a lot of drugs are really meant to change the ionic mechanism of cardiomyocytes. So this is a nice tool to play around. And I think we actually started this business on IPS-derived cardiomyocyte modeling. Michelangelo Pacchi came to my lab as a PhD student and later came as a postdoc, and his model, I think, is one really first one on this. And how it came to be is that some people have really done painstaking work and measuring these ionic currents from IPS cells. So a lot of data was needed to make the model. And then Michelangelo really fit the data to these current traces and make the model. And so we had the model, and we could kind of show the that in a way it really produced the same stuff that you can observe in vitro. And there's actually, this is an adult model that has been well let's say characterized. And these are two different models, ventricular and atrial IPS-derived cardiomyocyte models. And we were able to see what is the difference, how the ionic mechanisms are different, whether the drug effects were the same. We observed that it was about the same effect. Some cases, the IPS-derived cells actually have bigger effect of drugs than adult cells. So they were more sensitive. So this was quite reassuring that they were quite good models. And of course, this was very nicely balanced. The same things have been observed also in vitro. But of course, it's kind of boring. You have a nice model, but it's just like having one cell in your dish and saying that now we have a proof of concept. And how to go forward really was think about how to go this population level, high throughput in silico. And what we did, what Michelangelo did, was to make thousands of models, tens of thousands of models. And you can do it many times, many ways to kind of build this population and this kind of brute force, small changes in all the parameters, and you can get a lot of huge set of models with different kind of behavior. And like here are the action potentials of all these thousands of models seen there. Of course, they might not be the correct ones. How we select the ones that are not physiological. What we did was to we went back to the data. It's available. So all these boxes here are from different data sets measured from in vitro, and different biomarkers of action potential observed. And we selected those ones that are outside this and omitted those. So we pruned the population and took only those fit to all the biomarkers measured in vitro. So we generate this kind of nice set of models. And for example, it's kind of interesting effects start to happen because you could really have a population modeling and have population drug effects. So like this kind of small example where you have this hERG channel block, and in some cells, it really produced the effect that was expected. Some produced arrhythmia, and actually some of our cells died. Luckily, it was actually in computer, so nobody was hurt in this experiment. But of course, now we could actually look back and see what these models are and then go back in vitro. Unfortunately, we haven't really done it properly, but that is now, we can look this population and see what other kind of cells that are in danger of having arrhythmic or some other effects. And interestingly, we did also this long QT study on this and producing population of long QT. And when you add this mutation in your cells in silico, we observe that we have actually there two populations. We have populations that are sick and populations that are not sick. So we have kind of patients that are asymptomatic and others are not. It's like in real human population as well. Not all who have the mutation leading to long QT are actually long QT patients. And now we had actually had drug and test the drug with asymptomatic and symptomatic population. A little bit complicated to read there, but so we have pre-population control. Then we have this mutant asymptomatic population, we add drug. There's not very much change, but when you have a mutant symptomatic that has the long QT and you add the drug, you really have a clear change there. It's quite nice results. On considering that this was really in computer. And of course, it's quite hard to think that we have the original model, still the same one that we derived from the traces of ion channel kind of opening and closing. So we thought how to make a new one, or how to let's say make it easier. And this is one attempt that we just take the traces of action potential calcium and use those biomarkers and tune a new model based on just those. So we didn't have any traces of real gating of different ion channels. And we were able to make this. And this kind of lead to us to really try forward and think that can we make these kind of things purely on data from in vitro. And we were able to join forces with Emilia Incheva. She had done quite large still, even modern standards, quite large set of data from, let's say, 170 multicellular samples, 200 cells at least each, and they had traces of calcium and voltage from there, and they have tested also 12 compounds. Problem was that they actually did this all work in room temperature, and all our models were really for the human, and they were on 37 degrees. So first task was to trace or transfer our 37-degree model to room temperature. And it was actually one kind of example how robust these models actually are. We were able to do it by really changing the gating variables based on literature review on how the temperature is affecting the different ion channels. And based on this, we were able to actually make nice set of populations and test these 12 compounds and get surprisingly similar results with our in silico models that Emilia and others have made in vitro. And in addition, they were quite interesting that we were able to see similar behavior. So we observed kind of individual models that produce early after depolarization, similar things or similar kind of amounts that you could find in silico or the depolarization failures or irregular rhythm. So in a way, the population in silico and in vitro were surprisingly similar in many ways, not just respond to drugs, but this kind of, let's say, I would say, kind of personal traits of the cells. So in principle, what we can do is to have these cardiomyocytes, have the traces of calcium voltage, hopefully also the contraction, and have this kind of population model idea and transfer these data that we have measured to populations of models in silico. And of course, these models here can be certain phenotype. They can be individual. So we could have 10 wells or then MPS systems from individuals, because also our cells are not the same. They have certain variability. And we can tune these populations depending on those populations there. And of course, using these, we can then test all kinds of things. We could maybe test some drugs, toxicity, or certain conditions, whether it's ischemia or something else, and then find the populations, find the subset of cells or disease or drugs that are important, and really go back then to the bench. And if you think that this is kind of hopefully the kind of way to speed up things, they are not competing, both are needed. But we all know that if in a way, MPS systems are nice, they're excellent, but going really high throughput is difficult. And testing number of population, number of different individuals and number of compounds is a lot of work. But this is, of course, we haven't fully tested, but maybe something that hopefully we can prove later on that this really can be done. So having the MPS on the pipeline of or the in silico MPS and the pipeline of MPS testing. Okay, just to kind of show something where we're going now. Of course, these models lack the contraction, and this particular work here is by Amin, and it's under review. But you can, if you Google it, you can find it. It's online, as normally your papers are online earlier than they're accepted. And here we really integrate electrophysiologically and biomechanics, so contraction of cardiomyocytes, and also energetics, because energetics is needed, energy is needed for especially for contraction. And having this, we were able to actually look the new HCM drugs and get an insight on how they work and how they affect this. I hope maybe next time I can open this more. But just to kind of show, this is a list of papers and others that was actually data used for making this model and validation of the model. So to make this and validate what we did, needed a lot of work by many people in their labs in vitro. So to be able to do this work, we need really a close collaboration. We have a close collaboration in Tampere with these guys, but sometimes it's not enough. We really need kind of community effects to pull these together. Okay, and of course there's things to do. It's a nice heart in Katrina Alzada's lab. They try to do a kind of flat 2D culture, but sometimes the cardiomyocytes decide to do something else. Of course, this is what we would like to get, nice cardiomyocytes that are not just cardiomyocytes, because heart is not cardiomyocytes. It's I don't remember the percentage, but I think something like maybe even 50% or something like that. There's all kinds of other cells. So having just cardiomyocytes is not enough. So really to kind of think about how we can really bridge this gap from our cells there on whole heart level and patient level, and really have the feedback there. This is actually also my lab. It's a rat heart from microCT. And this is our attempt to really see the contraction on 3D cultures of iPS-derived cardiomyocytes. So I know it's interesting to see here how much work is now doing on this direction. And of course, our modelers, we need to follow this and see how we can cope, because this will bring a lot of complexity also on our modeling. More data needed and more collaboration. Just pointing out one interesting paper that just appeared by this Hubech and others. There's actually a couple of names from our friends from Norway, Simula Lab, that are also doing in silico modeling. And what they actually did in this paper, they managed to make, by having really nice MPS system, but also this metabolically driven environment, to make more adult-like cardiomyocytes. And what they did also in silico is actually they took one of our models from Michelangelo and make it more adult-like by getting the traces of some traits from their cells. And one interesting was actually the size of cells, because adult cardiomyocytes are bigger. In fact, surprising a lot on electrophysiology, but also some calcium handling. But they were able to kind of show that modeling can really explain what is happening in these more adult-like cardiomyocytes. So anyway, virtual precision medicine, I think this is what we do here in vitro body on chip MPS. It's going to play an important role in here, but I think we need also the data science approaches. I think in this, look forward to hear more of this, and also the modelers to kind of connect the dots and connect the different kind of areas. And just to thank these guys here who did all the work, Michelangelo, Paki, especially Jussi Koivumäki, Antti Ahola has done a lot of work in vitro, and Mohamed Amin, and collaborators, especially from Bologna, Oxford, and Washington. And I thank with that. And if somebody is interested to hear more about computing and cardiology, please come to Tampere in autumn. Thank you.
Thank you very much. We have time for one question.
Hi, this is Ishan from Berkeley, actually from Kevin's lab. So I had a question regarding your scaling from immature to mature cardiomyocytes in the model. Now, in the Ashlag's and Kevin's paper, they assume the functional invariance of the proteins and scale the number of ion channels based on just the size of the cell, right? Is your scaling also in a similar vein, or I didn't understand how that scaling was done in your model?
Yeah, of course, it's what happened in that last paper that it's not from ours. I think that is really what they did was looking at the size, looking the kind of number of ion channels that is really different in adult, and that is of course what we also observe. Of course, the kind of basic function of ion channels is very similar as whether immature or mature. The ion channel structure, protein structure is the same, but how many there are, how they're connected, and especially when you then start to look at the kind of how it's connected to calcium handling inside the cells and how it's connected to the, let's say, biomechanics of the cells, that is quite a disarray on these immature cells. But yeah.
Thank you very much. The next talk is by Lena Smirnova, Organoid Intelligence, the New Frontier in Biocomputing and Intelligence on a Dish.
Okay, thank you very much. I will not thank the organizers, otherwise I would thank myself and our center. I would like to thank you to choose this session over the French Quarter in New Orleans. So I will talk today about our new, more based on our research vision, what we would like to move forward in the next couple of years, or maybe 10 years, 20 years from now. So organoid intelligence, what we're thinking about, why we are thinking about that. So brain versus computer. Brain is more energy efficient. Brain has higher capacity of data storage than computer. Brain is continuously thinking and learning. Computer is faster in simple tasks, but brain is faster in complex tasks. Computer performs the task sequentially. Brain can do it sequentially and parallel processing, but computer is more affordable for us at the moment. So what we need to do, having all in mind advantages of the brain, to find the alternatives of the brain, which is a brain like biological computers. And this is our goal, to apply organoids to do so. So in our group, we developed a model from induced pluripotent stem cells, and Thomas, who is here in the audience, Helena and David, they were main developers. The model was funded by NCATS and is licensed to Axozim. And so we started from IPS, which are differentiate into neuroprogenitors, and they putting them on a shaker. We have really nice uniformed brain organoids, and we can produce 4,000 to 6,000 brain organoids per 1 6-well plate. And if you look inside of our brain organoids, we have different cell types of central nervous system represented there. So we have different type of neurons, astrocytes, oligodendrocytes. We also recently incorporated microglia into these organoids as an immune component, which is very important, not only to study neuroinflammation, but also for the neural developmental and synaptic pruning. And this is the list of different projects we have in our lab running or have been running. And I put all the QR codes of the publications we've done over the last couple of years using this brain organoid model. And you can see that it's a very versatile tool. So we can study neural development, development of neurotoxicity, neurodegeneration. My particular interest, for example, is autism and gene-environmental interaction in autism. So, but what we didn't address so far in this model, whether we can study cognition, whether we can study a cell memory and learning on a cellular level, on the level of the brain organoids. And I would like to step back and go back to the monolayer cultures. So this group in 2001 led by Steve Potter showed already that monolayer neuronal culture is able to control a robot, which you can see can move, and is entirely controlled by the just simple neurons which are sitting on multi-electrode arrays. Can you please start the video? And group in Corticals Lab, which is located in Melbourne, Australia, and we are currently collaborating with them, and you can find this Barkov paper already. They moved further and they teach their monolayer neuron cultures to play Pong. So basically, if you see this bar on the left, white bar moving, this is the cell cultures which control this bar, which is very fascinating, showing that our cell cultures are capable of memorizing simple tasks. But I'm not going into details why 3D is better than 2D. You probably heard it yesterday from John. He gave a very nice list of an overview why we need to move to 3D. And in particular, in terms of the brain and nervous system, it's more important to have very high dense cell-to-cell interaction. And we need more different cell types that's only neurons. So, and this is our team who is currently working and optimizing our brain organoids model to have nice synaptogenesis. And you can see on this electron microscopy graph, the synaptic terminal. We have myelination in our model, and this is really key for electrical conductivity, is it's insulation sheet wrapping around the axons. We have 20 to 30% of the astrocytes, which is also key nutrition providers and supporter cells for the brain neurons. And as already said, we have microglia, which is extremely important for synaptic pruning. So we can look into the synaptic plasticity within these brain organoids. So how we can look into the synaptic plasticity, we can record the electrical activity of them. And this is the schematic representation of different models which evolve over the last couple of years. It's now like a mushrooms after rain, growing different options how you can record. So for example, first you can see MASH electrodes which are covering the brain organoids. Then you can plate brain organoids on the platter multi-electrode arrays, either low density or high density. You can cover brain organoids, as you can see in this shell. We call it brain organoid EEG. And also you can put them on a Pocky electrodes, or you can use a shunt electrode by introducing nanopixels into the brain organoids. Simple calcium imaging is also very helpful. And there is a really cool experiment showing that the electrodes which are incorporated inside the brain organoids at those wires mimicking the neuronal structures without damaging the system. So in our lab, we are trying different options. So we are working with monolayer, multi-electrode arrays, low density, high density. We have brain organoids EEG and calcium imaging. And we also have collaborators where we're about to start to use shunt electrodes and neuron-like electrodes. But before I show you our results, I want to point to this publication from our collaborator from UC San Diego, Alison Moultrie. In 2019, this hit a lot of press because what he showed with his team, that when he played his brain organoids on the simple multi-electrode array, which is low density multi-electrode array, and kept them growing for 6 months, they were continuously increasing the complexity. And what was importantly, he showed that the network level of those events he was recording on oscillation was similar to premature born oscillations in the human brain, which was obviously fascinating, showing the capability of the brain organoids. And he moved further and attached those brain organoids to the robot which they built in the lab, and the robot was also able to walk. So now back to our model. So here you can see simple calcium imaging, and probably it's a bit underestimated assay. And actually the beauty of this assay is it's really scalable. So you can do calcium imaging in high throughput and get a lot of data from it. We also recently started working closer with Maxwell Biosystem, and they have also booths here at the conference, and Daulet is working together with Corticals Lab to establish our brain organoid and Maxwell system. And it's really elegant model. We can see very tiny small chip, but it has over 26,000 electrodes. So it's really high density, and we can record on the level of single cell. And here you can see a recording from one organoid. So, and you can see the different activity scan. You can see firing rate and bursting. And then Here on the raster plot to the left, you can see a lot of spikes over different electrodes, and the blue dark lines showing the burst, meaning that a lot of cells are firing at the same time. So showing the synchronicity in the electrical activity, which is, of course, this is what you want to see in your system. This is another really cool tool which Maxwell Biosystem provides on their arrays. Here we can track the electrical activity from cell soma alongside the axon. So you can see how the electrical signal is moving from the center of the cell toward the axons. And then we can see the functionality of axon and extract the axonal arbor of a single neuron or measure the velocity. And as of the set, we also have developed this EEG for brain organoids, and Chi Huan and David Gracias are the main players in this project. They are from engineers from School of Engineering at Johns Hopkins. And so they developed this very nice tool where you can have a leaves covering the brain organoids. And Chi is presenting poster today 234. So if you would like to see more material and bioengineering part of this project, please go and visit his poster. So here you can see those cages in the first image. Here without brain organoids and the leaflets, and then those leaflets are populated with the electrodes. And then when we place the organoids and place them in the medium, they folded around the brain organoids, and then we can record from those leaflets. So we are recording in comparison to the platter electrode array, we're recording from the surface. So EEG. And also this is the spontaneous electrical activity from these different channels. And here when we stimulate them with the glutamate, we can see increase in activity in response to the glutamate. So yeah, we can record spontaneous activity, but they have nothing to think about without input or output, right? So it's just electrical activity. So therefore, this is where AI should come into the play. And with that, we can provide input, but also output. We can analyze this big data, which we can generate from the recording after the stimulation to see whether we can train the organoids and remember their response to the stimuli. And this is from our vision is very promising way to move. And we can tackle a lot of different aspects of the neuroscience and memory studies toward organoid intelligence. So how learning changed the brain organoid, reflection of the biological concept, and how can we do it ethically? So we ask all those questions. And of course, we cannot do it only in our lab of the Center for Alternative to Animal Testing. So we actually brought together a quite amazing group of people. And this is the core people from Johns Hopkins only. We have bioinformatician and AI specialists. We have material scientists, bioengineers. We have ethicists on board. We have retina experts on board. We have neuroscientists, electrophysiologists, big data scientists. And we have recently teamed up with Barton Group, who is leading the institute in the applied physics laboratories, and they have a huge group of bioinformaticians who sitting and waiting for us to provide them with data they can play around and see whether they can find any memory or learning in our biological system. And beyond Hopkins, we also collected the interest from over 30 different people organizations, which is led also by Alison Moultrie. So we are trying to move forward a society of organoid intelligence, a community, and established organoid intelligence in this discipline. And to kick off that, we had a couple of months ago in February, we have a first workshop on organoid intelligence, and Iti, who is also present here, she was the organizer of this workshop, where we brought all those people together, from ethicists to biologists, neuroscientists, electrophysiologists, to talk about different fields. How can we bring them together, and how can we make this organoid intelligence happen? And beside that, the Dean of School of Engineering at Johns Hopkins also was very intrigued by our approach, and he put this project as a pepper line for the vision for the next 10 years for the philanthropic campaign. So what we want to do? So this is the brain organoid, how it is. We want to make it bigger. We make it more to generate more cell mass, more complexity. And we will be doing that by, and ITSI is already working on that with our bioengineering group. We are doing that with perfusion of the chemicals and performing the gradient. Then we will be recording with our shell electrode, as I already showed you, EEG. We can stimulate them simultaneously with the shunt electrodes and record also from the inside with the neural-like electrodes. And all this recording will be collected and then again analyzed by AI. And also beside the chemical stimuli or electrical stimuli, we are planning to apply more complex stimuli such as optogenetics and combine brain organoids with retina, which we also have in our Hopkins in our neighbor lab available, so that we have input from the retina, which is connected to the organoid, and organoid will respond to that input. And of course, the ethics, it's really important. You cannot imagine how intensive was the discussion at our workshop when we were talking about ethics, already starting from the name. So organoid intelligence as a name is highly ethical question, and was a lot of discussion about that. And we want to have SSIs on board from very, very beginning. Therefore, Jeffrey Kahn, who is the director of Bioethics Institute in Hopkins, he's on our board and together with his team. And they are following on the concept, and they're actually also part of the brain NIH Brain Initiative 2.0. So we are good covered with that. Okay, so it probably sounds like a pie in the sky, and we probably don't have the biological computing yet. But with this high level goal, we can achieve many different other things on the way. We can use organoid intelligence concept to use it in medicine, to study before we can really develop in biological computing. And Iti and Dolet, our students, Iti is postdoc on Dolet as a PhD student, they're working toward that goal. And Iti is developing the system to study neurodegeneration. So we can study the decline in the cognition and frame of the neurodegeneration, while Dolet will be looking into the neural development. So for example, how we can also use different iPS donor cells from autism or Asperger and see how they perform, whether they memory and learning and memory will be different from the typically developed individuals. So we can also study dementia and side effects of drugs and so on. So there is a big, big room to play with. And of course, it takes a village. And I think I showed you that our village is growing, and nothing is possible without Cat family and also our collaborators. And of course, all the funding we are getting to move the field forward. And with that, thank you very much. And I open for the questions.
Hi, Randolph Ashton, University of Wisconsin-Madison. I kind of have two questions. The first is, do you have a neuronal spheroid, a cortical spheroid, or a brain organoid in your system? Neuronal spheroid being neuronal arrangement of cells with astrocytes, oligodendrocytes you can put in there. Cortical spheroid being neuronal cell types that represent all 6 layers of the cortical plate. Brain spheroid being those same cell types, but organized in a pseudolaminar structure with a ventricular zone element to it.
So we can talk a lot about the terminology here. So what we have, we don't have cortical layers, at least not what we observed. We do have different type of neurons, that's definitely. And we also have in the beginning of differentiation, it's the neural rosettes as a ventricle zones. And we consider this the system as organoid because we have myelination and active synapses, and especially when we're putting them on electrode arrays, it's a substantial model, but yes, we don't have, it's not the cortical layers, it's not as structured as other organoids which are out there. But to the major difference of this model to classical brain organoids is that they have very standardized reproducibility in terms of the size and cellular composition. So we developed that model for toxicity testing primarily. So, and that would be the key. Yeah? So we need to have a certain standardized model to be able to screen chemicals.
Thanks. The second question, I guess, in Allison's paper, he takes the brain organoid, which in this spheroidal suspension shape has the pseudolaminar structure, but has multiple sort of morphogenesis points within the organoid. which is lacks biomimicry in that aspect. And then he puts it down the electrode array, and it sort of flattens out. So the electrode signatures that he is assessing and watching, quote unquote, mature are essentially neuronal networks that are maturing over age as the synapses get stronger. And the EEG signature that he is matching that to in terms of neonate is sort of measurements of the timing of the neural circuits. And he's showing that the oscillarity that they get in those structures matches in the timing. But I guess my question goes back to the idea of intelligence. And in those organoids and potentially in yours, there's critical aspects of cortical plate circuitry that are missing. And so I just, as you said, it's kind of pie in the sky, which is interesting, but I just wanted to be sort of careful on the language we use of intelligence and the idea of teaching, like the Pong exercise and stuff is sort of mapping neural circuits in a dish that stay relatively standard, and you can kind of induce synaptic strength by adding in their various excitation at certain points in that network, right? And so I guess my question is, while it is pie in the sky, do you think it's really accurate to say intelligence versus more sort of creating circuitry that is definitely biological circuitry that you can program?
Okay, so I will answer with the question, is artificial intelligence an intelligence? Is artificial intelligence an intelligence?
Again, terminology, artificial intelligence is pattern matching, to be honest.
So
and
therefore,
you can teach it to learn, and from the computational sense, you're learning patterns and sort of orienting those, and you can do deep learning and stuff like that. But those are like buzzwords, which get a lot of press, but I wouldn't necessarily say are quote unquote intelligence.
right, that is the point. So I can tell you also that organoid intelligence idea came from artificial intelligence, obviously. And also just to emphasize, we are not planning to recapitulate brain development, how it is, and learning and memory, how the neuroscientists pursue that in vivo. Our goal is to have the level of learning and memory which those particular systems are capable of. So if they can play Pong, how can we move beyond to use that also for compute something?
One last question. There's a giant literature on the electroencephalogram inverse problem. Have you started thinking about the voxel size that you can invert to understand where and what's going on inside your brain organoids?
Yeah, it's a great question. And I think we're not there yet to start tackle these problems. Thanks.
Okay, the next presenter in our session is Carrie German from CFD Research Corporation, discussing a multi-scale computational framework for modeling MPS.
Okay, good afternoon. Thank you guys all for being here, and thank you to the committee for the opportunity to share our research with you guys today. I'm Carrie German, a group leader at CFD Research Corporation, or CFDRC, in the Computational Medicine and Biology Division. And as you may have gleaned from Dr. Ingver's talk earlier this week, we focus on developing computational models of physiological systems through various collaborations. So this talk will kind of just give an overview of some of our different capabilities, ways that we've implemented these models. And while it will just scratch the surface of what we're able to do, I'm hoping to kind of show how these models can, sorry, I'm hoping to show how these models can be used to optimize chip design, both prefabrication and post-fabrication modification, as well as to support the demonstration of physiological relevance of these systems. So as we all know, the drug development process consists of a series of in vitro, animal, and human studies where the overall goal is to develop a safe and effective medication. Various efforts are made along the way to translate these in vitro and animal preclinical results to the human condition. And though there's been debate, of course, over where microphysiological systems will fit into this drug development process in the future, I think the general consensus is that they will play a key role in the translation from in vitro to the in vivo condition. So an additional piece that plays a key role is the computational modeling part. So as Dr. Schuler indicated earlier, PBPK modeling has been around for decades, and it's used to basically indicate how the drug distributes to the body and predict how the drug can distribute to the body. So through various collaborations with government, industry, and academia, we have developed numerous PBPK models linked to PD for numerous animals, many of which you see here, as well as humans. And we use a first principles modeling approach where physics dictates the transport in our systems or in our humans, our subjects, and also the physicochemical properties of the drug as well as the properties of the tissues dictate how the drug distributes into the different organs. So we actually can use this same approach for in vitro systems modeling, taking only into consideration now the material makeup of the system. And this type of approach actually helps us to enhance our ability to translate from the in vitro to the in vivo much more smoothly. So when we start this, we got to develop computational frameworks for the microphysiological systems. And the way that we do this is we start by replicating the geometry. And there's typically two different approaches that we can use. Compartmental modeling, which is very, very commonly used in PBPK modeling because it's very nice for representing well-stirred compartments. They're very fast running and they're relatively easy to build. However, these models do have their fallbacks where they can't resolve the spatial gradients and modeling multiple species becomes really cumbersome. And the 3D approach can actually compensate for some of these things where we can actually have spatial resolution of gradients in the system. And it's actually due to the modular nature, we're able to model multiple species much more smoothly. It also has a nice visualization component. However, anybody who does computational modeling knows these are very expensive and can take days, even weeks to get a final solution. So we at CFD Research have kind of come up with an alternative that brings together kind of the best of these two worlds using a reduced order modeling approach, which we term quasi 3D or Q3D, to model these different channels in the microphysiological systems, as you can see here. So we still retain the spatial resolution. We resolve the gradients in the direction of flow, but we also still allow species transport in cross flow as well. So these still retain the advantages of the 3D that we can model multiple systems, and we have those visualization components as well. And then of course, we also have the opportunity to combine them all and see which one is the most efficient and best model, depending on the system that we're modeling. So just because it's a new kind of a different approach, we wanted to make sure that we rigorously tested this Q3D approach and make sure that it could replicate both the simple ODE system or compartmental system as well as 3D. And here you can just see we've done some simple test cases where the PK profiles in a 2-channel microphysiological system overlap exactly for both the Q3D versus ODE case, and as well as 2 different meshes and multiple other meshes, but for the 3D and Q3D case. So I kind of start into more of some of the examples of the things that we've been able to do. So I'm starting with a conventional in vitro system. I know it's not a microphysiological system, but it's kind of a nice place to start. We don't have flow, it's diffusion driven, and it's actually a relatively simple geometry. So we can actually use compartmental modeling approaches to model these different pieces where the different medias are represented as different compartments. Cell membranes, intracellular organelles can even be represented in these types of models. So transport within the system goes across these boundaries. We integrate over the boundaries to represent diffusive flux across the membranes, the cell membranes. And the drug metabolism is actually represented in the cellular compartment. So we typically use Michaelis-Menten kinetics here to represent drug metabolism, but this is also used for us to model nutrient metabolism, which is also important as well. So in the bottom, you can see the just a well plate simulation where oxygen distribution is represented with respect to the media height. And so you could see how we could do that, plot these for different times and actually optimize our media change protocol, as well as our volume conditions to enhance our quantification efforts. So probably moving on into the more exciting organ on chip pieces. Once we add the perfusion piece, it doesn't really completely, we don't have to go recreate the wheel here. We're still looking at the same kind of process. We still recreate the geometry depending on what we've got. On the left, we have a standard linear channel. Those obviously can be resolved with Q3D geometries. On the right, you can see a collaboration with Synvivo where we have a more rounded geometry. So Q3D is not as effective here. We have to use more 3D geometry resolutions. So after we recreate the geometry, we have to assign volume conditions and boundary conditions. This is just like your typical conditions, viscosity, diffusion coefficients, things like that. And then boundary conditions, we can set our flow rates as well as our inlet and outlet pressures, and then let the system solve itself through different well-established flow characterization equations. So once we've got this all working together and we can run a test model, we can then look at optimizing these conditions, these flow conditions to show the physiological relevance of the system. If you look at the plot on the bottom, next to the flow dependent shear stress is SS. You can see how we can modify the flow in the system to change the shear and make sure that we are replicating what we should be seeing in vivo. On the side with this in vivo, we can see how this type of modeling can be used to optimize your design of your chip. And in this case, we can change the pore density to make sure that we're achieving the concentration needed in that target tissue chamber there in the center. So after we do these optimization studies, we can then move into predictive studies, which you can see at the bottom for single channel perfusion versus dual channel perfusion. On the left of these two pieces, or I hope it's left, yeah. So if on the left, you see the simulations for these two types of perfusion, and then you see the experimental results that are in very good agreement with these on the right. So it's not just modeling the chip, right? There's a lot of pieces, and there's been a lot of discussions on this throughout the sessions that I've attended. It's also modeling these peripheral components. There's been a lot of talk about tubing and the absorption of these drugs or compounds that we're looking at into the tubing. There's a lot of other phenomena that can occur within the reservoirs, the valves, the sensors, the pumps, whatever other pieces that we want to include. So we've worked on integrating those pieces, and actually here we can kind of show some of our multi-scale modeling capabilities to enhance our model efficiency. So through previous collaboration with, Charité, they developed a liver on chip model, and we were trying to simulate APAP distribution throughout the chip. So the model itself had to be resolved in 3D. But the underlying system of tubing and piping was actually modeled in Q3D. So we could integrate these two systems and allow them to work together more efficiently to get the results that we needed. On the right, we can see the continuity of the two models, so that the APAP that was administered to the bubble trap actually does flow throughout the whole system. So another subject that we've seen and heard a lot about this week is PDMS partitioning. So a lot of these different systems include PDMS, and the loss to that PDMS has to be considered for any of these different kinds of experiments that we're conducting. So in collaboration with WEIS, we worked on implementing a model, a Q3D CFD model for drug absorption into the PDMS, where this is where we can see the spatial gradient without the actual 3D representation on the right, where there's a 10-hour inflow of FITSI and then a 5-hour washout, but we still see the absorption and spatial gradient despite that presence of a 3D meshing in the top channel. We also compared the results in our Q3D model for the concentration of FITC with respect to the distance into the PDMS with the experimental results. And we have very nice agreement for our first implementation of this model. So we're excited to see where else we can actually apply this type of modeling. So as I said previously, modeling multiple species can be an important part of what we're looking at in these chips, particularly if you have interacting species or if you have metabolism where some kind of metabolite is produced. So that's obviously, we need to be able to model both of those simultaneously. So this is just another example using the Weiss chip where we've modeled the distribution of glucose into the system, as well as ammonia production and albumin production in the system. And so these were all modeled simultaneously, which is a huge advantage, not having to recreate the model every time. So the next step after we've got our chips, we've got our outside pieces, we've done all the PK modeling. We also like to link these to some additional types of modeling like PD modeling as well as tox modeling. So we've done various efforts in looking at this. This is kind of an older collaboration with Weiss looking at the heart chip where we can see dose-dependent changes in the membrane potential. Well, on the chip, we do model the drug PK with the different losses and binding and flow, but then we can also use that to simulate drug toxicity using the drug concentration for ion channel binding. And then you can see in the bottom corner, I know it's already stopped, but it's the replication of the electrophysiology of the health, not the tox affected one, but just under the normal conditions where we mimic what is seen on chip and then what's seen for the virtual patient. So we've modeled these individual chips and we've looked at all the things that we can do externally, but now we want to look at, can we model multiple organs on a chip? So you've probably heard Casey and Diogo talk earlier about their multi-organ chip, their 4-organ chip containing skin, bone, heart, and liver. So we help to support those modeling efforts. And we just similar, exactly like we would do a single chip, we've just done it for all 4 of these different tissue chambers. We have characterized species transport, and then we characterize metabolism of the drug doxorubicin into doxorubicinol. And we did this for both a functionalized membrane and a non-functionalized membrane to see how that membrane functionalization affects the PK results. And so I won't show both because they did. So I will show how well we were able to match the experimental results for both the parent drug doxorubicin and the metabolite doxorubicinol in all 4 chambers of the chip. And then finally, I want to wrap up with kind of how this kind of can all tie together and how we see in silico modeling supporting IV-IVT. So in collaboration with the FDA and some ongoing work, we have developed this with the Office of Generic Drugs as kind of a preliminary approach. So we go through and we characterize the compound. We find as much data as we can in literature. We're trying to do this all based on literature and not having to conduct new experiments. So in our case, we also have IVRT experiments, not just in vitro, metabolism and transporters, but also IVRT. We have ex vivo IPT experiments that we can also simulate. And then we try to use those same exact parameters that we use in each of these models and keep them the same, keep them constant. So permeabilities, partition coefficients, dissolution rates, all of that is kept the same across all of our models. So this is just an example that we've done with loteprednol etabinate. It's applied to the eye, an ophthalmic ointment. We collected a lot of data from literature, but of course they wasn't all there. And this is where I see MPS playing a really key role, is determining the values of some of these parameters where they can reduce the amount of calibration that's required. After simulating IVRT and matching the experimental results for that, again, using first principles approaches, we moved into corneal penetration studies. So we again found this data in literature, and using the thicknesses of the tissues and the Franz cell setup, similar to what we would do with the well plate setups, we were able to characterize species transport through the cornea. And again, using those values, we use those same values for topical application of the ointment and are in generally good agreement with that data as well. So in terms of current and future directions, some of these things are ongoing. Obviously, you've seen some of our on-chip pharmacodynamics modeling, but we want to extend that and look at some more ways to expand that portfolio. We've heard a lot about on-chip genomics and how that can affect metabolism and different other PD effects. So we would like to integrate those as well. Obviously, more toxicity, and of course, the whole reason that we're here looking at how we can have MPS inform IVIVT. We'd also like to look at moving into modeling disease states and incorporating bioanalytics into our modeling pieces. And we're always looking for collaborators and ways we can expand this portfolio. So I think to finish it off, I will just say thank you to our many collaborators without which this isn't possible. Our models don't work the way they should. So thank you to all of them, and I'll take any questions at this time.
Thank you. We have time for one or two quick questions.
Hi, great talk. I was wondering if your models are able to account for like remodeling of extracellular matrices, and especially a lot of interest in like vascularization or tissue breakdown, because a lot of what you showed seemed more like static blocks. You kind of like pre-render the entire system, and then you do computational fluid dynamics to see like the dead zones.
Yes.
But like, let's say you have a hydrogel that over time gets remodeled or breaks down. Can your models, or are you looking for ways to also put that into your processes?
Yes. So we can do deformation, and so we have looked at that, but we haven't actually done like the erosion of the matrix or laying down of a new matrix. So I think that would definitely be an exciting avenue to pursue.
Thank you.
Thank you.
I'm Rachid from Geraldine Biotech. I have a question about the microscale modeling versus macroscale modeling for microphysiological system. First principle with the microscale modeling, there are a lot of assumptions for the rate constants that cannot be really measured in MPS systems. And how do you handle the sensitivity of the parameterization and extracting the information from the chip experiments? Is this the ODE path versus the CFD more PDE path is more reliable based on your experience?
So that's a really good question. I would say the ODE path is definitely, I don't know if it's even more reliable. It's probably the easier path at the moment, but I think we are going to have to move into the CFD path. It's just I don't know what the path is to getting there yet.
Thank you.
I had one quick question. Yeah. I've admired the CFDRC work for many, many years. Does your quasi 3D simulate the blurring of a concentration change due to Boissel flow?
So that's a really good question too. I actually would have to talk to our coding guy on the back end of that. I'm not sure.
The reason I ask is there's a very interesting photograph of a Steve Quake circular mixer / pump that shows a parabolic wavefront that is stuffed inside of itself. And I've always wondered the extent to which you can fake that without having to do full 3D.
Yeah, that's an interesting. I will get back to you.
Thank you.
Yes.
Our next talk is being given by James Tronelloni of Texas A&M Machine learned vascularized networks improve predictive power of organ chips. You are a young investigator and are in the young investigator competition. So I urge you all to come up with some very hard questions.
Great. Thanks for that intro. And I'd like to thank the organizing committee for great conferences thus far, as well as their support for me with a travel award to present here today. So as I'm sure everybody in this room knows, microvasculature is a key component of many tissues in the body. So you can see in the schematic over here, microvasculature fenestrates organs such as the brain or the kidneys. It fenestrates smaller tissues such as pancreatic islets and the villi of the small intestine. And I also have a little bit of a tissue engineering background. So when you read that literature and you look at when they tried to develop implantable tissue engineered products, vascularization is still a key component of their design. So I'm sure many people are familiar with the traditional vasculogenesis on a chip setup. Our lab uses this chip because we believe the spontaneous self-assembly of microvasculature is a good bottom-up engineering strategy for developing small capillary beds on chip or even in future tissue engineered products, because it involves a lot less user input, a lot less expensive or complex manufacturing processes. And the way this chip works is, just to briefly explain, we have 5 parallel channels that are separated by microposts, so it's semi-permeable. We can inject viscous precursor or hydrogel precursors into the central channel and the outer channels, cross-link them without them bleeding into the fluid channels. And then we could hydrate the channels with media in the center left or center right channels. And over a course of days, if we put pro-angiogenic cell types or pro-angiogenic growth factors, we can create growth factor gradients towards the center of the chip. So just to quickly go over how vasculogenesis happens in this chip, we put an individual suspension of endothelial cells in a hydrogel in the center channel. And in the outer channels, we put pro-angiogenic cells in hydrogel, or we can even add pro-angiogenic growth factors in the media next to endothelial cells. And in 4 to 7 days, we'll get formation of microvascular capillary networks like you see in the fluorescence micrograph to the right. So I also have a video on the bottom left showing individual endothelial cells sprouting into capillary networks, as well as a perfusion of microbeads from parent vessels into the microvascular bed, showing that these are fully functional and perfusable microvasculature on chip. So our group recently reviewed the previous few years of microvasculature on a chip. So this includes vasculogenesis, but also angiogenesis and lymphangiogenosis. And we found that there's a independent or interdependent combination of different components that can lead to different or morphologically different microvascular networks. One important thing that I noticed when we were reviewing this last year is that a lot of people evaluate their microvascular networks based on morphology. But as I mentioned earlier, tissue engineers especially, but also some MPS designers are focused on oxygenating the chips or oxygenating the tissue engineered grafts. So the question becomes, how does morphology relate to the oxygenation of the tissues? So to answer this, I guess first I'll show an example experiment. Here we increase the hydrogel stiffness. We could see that the vasculogenic networks for the lowest stiffness groups are morphologically different, but they look fully perfusable. The highest 2 stiffnesses don't look as perfusable, but they also look morphologically similar. When we quantify this, metrics such as vascularized area kind of match this observation. But then if we look at other morphological metrics like vessel length or branch point count or segment count, the 5 mg per mil group, the second least stiff group, emerges as the winner. But if we look at other metrics such as the mean segment length or the mean segment diameter, the 2.5 mg per mil group, the least stiff group, looks like the winner. So the question becomes, which one might have the most success in downstream applications such as vascularizing other tissues on chip or using these ingredients in a tissue engineered construct? So then we developed this MATLAB toolbox that we call AngioMT, which stands for angiomass transport. And we take the same images that we use from our morphological quantification. We input that, and then we can generate oxygen gradients in the vascular network and in the tissue areas surrounding the vascular networks. And we come up with two biological metrics for each sample. The first being a vascular potential, which is the oxygen concentration going through the vasculature minus the reaction term of endothelial cells consuming oxygen. And then we also have a second term that we term hydrogel oxygenation, and that's an area average oxygen concentration delivered to the hydrogel. So we have two new metrics that we could possibly use to evaluate our vasculogenic networks. But the problem here, and it's a problem that we've been discussing a lot this week, is that a lot of MPS technology and even this data analysis technique, it's not throughput, or it's not high throughput. We generated 500 images in our own lab, and to quantify everything with AngioMT, it took over a week to generate all the data. And if we want to, like make this available for pharmaceutical companies or other smaller companies that want to develop tissue engineered products, they can't wait so long to generate data from their vasculogenic networks. However, for the same size data set, quantifying 10 different morphological metrics, that took less than an hour. And it kind of makes it obvious why a lot of people think, or a lot of people will use this as their standard. It's easy to get this data. There's a lot of open source programs out now that make it very easy for even non-engineers to quantify their vascular networks. However, as I mentioned earlier, it's still not clear if optimizing different morphological characteristics will really give us higher quality networks. In our lab, we like to think that the higher quality networks are the ones that can transport the most oxygen, but it's still not clear which of these morphological metrics might transport more oxygen. So then we decided maybe we can use the morphological data, which is very easy to get. A lot of literature already has this data. And we're wondering if we can create a machine learned regression problem and predict the two biological values and output a singular continuous data that scales between 0 and 1 that we can use as an evaluative metric of vascular network quality that correlates highly with oxygen delivery into the tissues. So when we were choosing a machine learning algorithm, our intuition told us that we should start with multiple linear or multiple exponential regression. And you can see from the scatter plots, those might be good choices, but we're dealing with a data set that had high multi-collinearity between all the independent variables. So multi-collinearity is just a statistical concept saying one independent variable is highly correlated with other independent variables. And we could quantify this with the variable inflation factor equation, where a VIF value above 10 indicates multi-collinearity. And we could see for 5 of the 6 metrics, all of them far surpass 10. The 6th one, segment count, has a value of 8.7, so it's pretty close to 10. So we know that multi-collinearity is a huge issue with this data set. So it eliminates a lot of simpler machine learning algorithms. But we could use other algorithms such as decision tree regression, or in this case, I tried random forest regression, which is just an ensemble of decision tree regressors. And we could see for the 2 morphological predictors, as well as a third predictor, connectivity, which has been reported in the past, but that's still a morphological value. All of these, when we look at the residuals of the expected value minus the predicted value from machine learning, the residuals are normally distributed around 0, but a lot of them fall close to minus 0.5 and 0.5. And when you look at a plot of the expected versus predicted, as you increase the true biological metrics, the predictions become less and less accurate and they're more widely varied. So it's indicative, for this algorithm and a lot of the other non-deep learning algorithms that follow the same trend, that simple non-deep learning methods really aren't suitable for this regression problem. So then we turn to deep learning, and the first example I'll have is neural network regression, where we take each of the morphological metrics as input and we sequentially combine them in hidden layers of certain amounts of nodes that the machine will learn. And each node can be summarized as an input value multiplied by some weight the machine learned, plus some bias that the machine also learns, and then filtered with an activation function. And all of this data is summed at each node and then outputted to the next node or to the output layer. So in this specific case, or in this specific schematic, I have an output layer with just one value. But with a neural network regression problem, we can have a multi-output regression problem similar to the regression tree. So when we were testing different algorithms or testing different architectures of neural networks, we first tried a multi-output neural network that predicted both vascular potential and hydrogel oxygenation. We found that to still suffer from the same problem that the regression tree had, whereas you increase the true biological values, the predictions became less, or I guess more random and inaccurate. Then we decided, what if we created two different neural networks that separately predicted each of the values? So we have one that predicts the vascular potential. We have one that predicts the hydrogel oxygenation. We found that the accuracy increased a little bit and the residuals are still normally distributed around 0, but we can see it still suffers from some wildly inaccurate predictions of the true biological value. So then we came up with this idea: what if we take the 6 morphological values plus the vascular potential value from NGO-MT and just simply predict hydrogel oxygenation? Because in the end, you know, in the first slide I mentioned, hydrogel oxygenation is the ultimate goal. So why don't we just predict that? So when we do that, we have a trend, or we have predicted values that generally follow the expected value trend, and the residuals are tightly bound around 0. But the problem with this is it still relies on the angio-MT vascular potential values. And as I mentioned earlier, that would take, for 500 samples, that would take a week to quantify. And if somebody's already quantifying vascular potential, they might as well quantify the hydrogel oxidation as well. So in response to that, we devised a neural network architecture that still only uses morphological data, where we first input the 6 morphological metrics into a neural network and first predict vascular potential. Then in the second neural network, we have the same 6 morphological metrics, and then we add a 7th input, the predicted vascular potential, that is predicted from our first neural network and is calculated in seconds. And then we input this into a second neural network that predicts hydrogel oxygenation. And then we design this neural network so that this output value is some value between 0 and 1, where higher oxygenation comes closer to 1 and lower oxygenation comes to 0. So this output we simply use as our quality index. So the output of the neural network ends up being some kind of evaluative metric that can be translated from lab to lab. Granted, they use the same image processing techniques. So when we look at the same accuracy plots, we see that this neural network is tightly bound around 0, but it's slightly less accurate because it's using predicted vascular potential instead of true vascular potential. But we see that the widely inaccurate predictions are far less compared to the single output neural networks or the multi-output neural network. And if we look at a box plot of all the architectures we tested, we could see here, these 2 are from the multi-output neural network. These 2 are from the single output neural networks. This one that looks the most accurate is from the neural network that uses true angio-MT data. So it's not addressing the problem we aim to solve. But the chain neural network looks most similar to this one. It has ranges that are closely bound to 0, and it has the same number of outliers as what we deem to be the most accurate. And so when we perform a Pearson R correlation test, we also see that our new value, the VNQI, which takes into account all the morphological data, that correlates most highly with the true oxidation compared to all the individual morphological metrics alone, which is the current standard. And I know it's a little small, but we also have traditional machine learning accuracy metrics that I've pasted here. And if anybody wants to discuss later, I could maybe discuss that. But an important thing with neural networks too is that we can employ explainable algorithms in order to understand what's happening on a deeper level. A problem with machine learning often is that it becomes a black box solution, but we chose to dive a little deeper by employing Shapley additive explanations, or SHAP. And that's just a game theory based algorithm where it takes all of the inputs and subtracts just one of them and sees how much of an error is left in the true predicted, or in the predicted value compared to the original predicted value. So from this analysis, if we look at the graph on the left, we can see that if we remove branch point count, because it's so important in this quantification, it leads to the highest errors when we're only predicting with the remaining 5 morphological metrics. So branch point count, mean segment length, those are the most important for calculating vascular potential, whereas traditionally optimized morphological metrics such as segment count or vessel length, those actually turned out to be the least important morphological metrics for determining the vascular potential. So not only do we have a new algorithm for determining what might be a good evaluative metric, but we're also seeing that the current standard is not agreeing with this. We've been optimizing vascular networks incorrectly based on morphological metrics that we see don't really impact the vascular potential that much. On the graph on the right, we can see that as you increase branch point count or mean segment length or any of these, increasing these leads to higher vascular potentials. So for all these metrics, higher values mean more vascular potential. But then if we look at a SHAP analysis of the second neural network, we can see that certain metrics like branch point count and the mean segment diameter, if you increase those, you actually decrease the oxygenation. So it also highlights that you can't just only try to optimize and generate as many of these morphologies as possible. You can't be trying to make the branchiest microvascular network or the microvascular network with the largest segment diameters, because you're actually decreasing your oxygenation. And this is key because a lot of the previous literature is showing, optimal networks will have high values for all of these metrics. Additionally, we see for predicting the hydrogel oxygenation, the predicted vascular potential is most important. And that was kind of expected because we never got an accurate model until we started incorporating this chained architecture, which uses vascular potential. So then when we look at our case study again, we can look in the context of VNQI and see that the two lowest stiffness groups don't have a significant difference, but both of them are more oxygenating than the two lowest stiffness groups. And then also if we run another Pearson R correlation test, we can see that VNQI correlates with the true oxygenation to an R squared value of 0.94. So it's very highly correlated with the true oxygenation. So then the question becomes, why is this important? Why do we do all this machine learning just to determine a new way to evaluate microvascular networks? Well, the answer to that is when we wanted to start using organ chips to design cellular therapies or tissue engineered products, we might want to limit the amount of experiments we do, especially when we validate those experiments with in vivo testing. So I'll show an experiment coming up modeling islet transplantation therapy. And that's a therapy that currently suffers from an inability to vascularize islets due to severe hypoxia when the host vasculature doesn't integrate with the graft. And there's no FDA approved islet source except for cadaverous islets. So if we can increase the rates at which islets revascularize and decrease islet death during the grafts, we can potentially give islet therapy or islet transplantation therapy to more patients. So here we just have islet spheroids injected with the endothelial cell suspension, and then they form capillary networks around, or they're fenestrating the islets. And then in this experiment, we use different vascular network ingredients that scored low or high with the VNQI methodology. We challenged all the chips with hypoxia. We did a glucose stimulation assay, and then we measured insulin. And we found that chips cultured with high VNQI vascular networks increase their insulin almost twofold, kind of validating all of this machine learning, showing that if we can use VNQI, we'll generate better performing islet chips as well as potentially downstream tissue engineered products. And just to conclude, this work, we have a few outcomes. The first being a vascularized islet chip, which is still a goal in the MPS field. We want to use this for immune cell co-culture and therapy testing for mimicking type 1 diabetes, but it could also be used for type 2 diabetes. And then obviously we have all the tissue engineering applications, such as testing other islet transplantation therapy biomaterials, such as pro-angiogenic biomaterials or immunoprotective biomaterials. But all of this is to increase the weight of the in vitro testing so that we can reduce the in vivo testing we have to do downstream. And then quickly, yeah, sorry. I'll just thank my fellowship funding sources, NSF, AHA, Chris Jafdari. He was an undergrad that I mentored, and he helped me generate a lot of the vascularized organ chips. And then Tanmay Mather, he was the graduate student who generated AngioMT, from which I was able to do a lot of the machine learning. And then thank you, and I'll take any questions.
I'll just ask one quick question before we move to the next presenter. So I'm curious why you confined your feature set to the 6 morphological features when you're doing dynamic measurements in your system. Have you thought about opening it up to some of those dynamic features that are time dependent, that like the perfusability of the networks?
So you're asking why I use 6 morphological metrics instead of using other more complicated morphological metrics?
Or right, or not just morphological, but actual dynamic features.
So we limited to that because we use a tool that most people will use. We also use other tools. So there's AngioQuant, Reaver, which is what I use. And all of them only give those static morphological features. There might be more complex data, but if we, from all the literature I reviewed last year in that review paper, everybody was just using those standard metrics. So we figured if we want something that could be adopted by other people, we might as well define our machine learning algorithm in terms of what people are already using.
Okay, thanks. Okay, our last, thank you again to the speaker. So there's been a change in the lineup for our last presentation, same topic, but different speaker. So Jan Lichtenberg from Inspero will speak to us about ex vivo human 3D NASH models as a screening-based discovery approach for selecting and prioritizing drug candidates.
Thanks a lot, Nicole, John, for hosting the session, and Thomas Lehner for making this all happen. Sue Grapper, who was supposed to give this presentation, her vocal cords were victim of the festivities last night, so she can't really speak. I'm a last-minute replacement here. It was a great party. I'd like to kind of build this bridge between phenotypic screening and machine learning as an enhanced readout to make our phenotypic assays more relevant than just relying on standard biochemical readouts. We are interested in NASH, non-alcoholic steatohepatitis. Why is that so interesting? Well, first of all, it's a quite complex disease that needs lipid uptake in the liver, inflammation, and then fibrosis, cirrhosis. Secondly, there is no current therapeutic solution on the market. And I think it's a great example how 3D microtissues can be used to model a complex disease in vitro, and how we can read these systems out. So the 3 aspects that I'm going to touch today is how are we building this model, and how are we stimulating the model to develop the disease phenotype? What are the endpoints and readouts that we're using to measure what's going on? And then how do we use image-based or machine learning-based image quantification to measure the complex morphology of the fibrils that are formed during that fibrotic step? So the system is based on our gold standard liver tox model, which is a co-culture model of hepatocytes and non-parenchymal cells like Kupffer cells and endothelial cells. But what we add to the system are stellate cells, which are extremely important to develop the fibrotic disease phenotype that we are after. Now, we start with a lean and healthy model, and then we induce NASH by feeding the tissue a cocktail of free fatty acids and high glucose concentration. So it's basically a diabetic medium. It takes a few days until the tissues take up the lipids and then start to develop a disease phenotype. But we also have to create an inflammatory insult using bacterial LPS that then triggers the disease progression in the system. And now that we have a diseased phenotype, we can start to look at compounds that are either anti-inflammatory, anti-fibrotic, or metabolic to reduce lipid uptake in such a system. What's important is that we have a pretty substantial battery of assay endpoints that, on the one hand, are screening compatible because we want to upscale that system for automated screening. On the other hand, we also have endpoints that have high clinical relevance because ultimately we want to convince pathologists and researchers that this is a functional model and that they can relate this also to clinical endpoints. So the primary ones that you see today are biochemical assays on the screening side, and then either high content imaging based or histology based IHC endpoints to look at tissue morphology from a histological perspective. So this happens all in our Acura 96 well plates, a single tissue per well, always the same cellular composition, always the same size. Uniformity is key here because otherwise we'll never really get to a screening compatible solution. So let me take you through the three hallmarks of NASH: steatosis, inflammation, and fibrosis. And we start with steatosis. What you see on the left are high content images of these tissues. We use Nile red staining to make the lipid uptake visible. You see DAPI for the nuclei in the system. The lean control is on the left, and then the induced NASH is on the right. And you see a clear increase in Nile red staining here. If we just by pixel counting do a simple quantification of this image, we already see that we have a 10x increase from control to NASH condition. So it's a very strong signal. We have a great dynamic window in this model. The imaging endpoint is great, but it's fairly time consuming to generate this data. So for a screening application, we'd rather prefer to have a biochemical readout, and that's what you see on the right side. Using a triglyceride assay, we can pretty much generate the same data. We see an 8 times increase here between control and NASH. So again, very large dynamic assay window in this system. Now for the inflammatory response, we use a Luminex assay, and we're able to quantify a large number of cytokines and chemokines in parallel with the system. We have built panels that help us to look at pro-inflammatory markers like IL-6 and IL-1 beta, or at fibrotic markers like TNF-alpha and IL-10 in parallel in such a system. And what you see is that between control and the NASH model, we see a substantial increase in chemokines, in cytokines across the board. And this is also been true for chemokines on this slide, where we again have a good mix of pro-fibrotic and pro-inflammatory markers in the system. Now, the important aspect is how are we quantifying fibrosis in such a context? Because it's not just about the amount of collagen that's deposited in the system. It's also about the structure of the fibrils, how mature they are, how strongly assembled they are. And I'll show you how we attack that in this proof of concept study. We took an antibody, an anti-TGF beta-1 antibody. Why is this relevant in this context? TGF beta-1 is one of the drivers of fibrosis in endothelial cells. So you see that the TGF beta-1 triggers the TGF receptor 2, which then the receptor 1 recruits ALK1, ALK5, and then triggers this whole signaling chain through SMAT, and ultimately leads to an upregulation of collagens and TIMS. And if we are blocking the TGF beta-1, we should see a decrease in collagens and TIMS in the system. And that's what I'm going to show you in the next couple of slides. So our screening compatible endpoint to look at collagen is measuring procollagen 1 and 3 in the supernatant. You see the procollagen 3 data on this slide. What's really nice is you see the dose response that works both on day 5 and on day 7 to 10 in this data set. We have a pretty good consistency from model to model, so high statistical relevance in this assay. And what's important to note here, this is a measurement that's taken in the supernatant, so we can also look at longitudinal changes over time inside the same tissue and multiplex this with inflammatory marker quantification using the Luminex assay. So you get a lot of data from this complex model over time. On the next slide, you see the same quantification now for tissue inhibitor of metalloproteases, TIM. The TIMs are very important in NASH. Typically in NASH patients, the TIMs are upregulated. The TIMs then inhibit the MMPs in the patient, and the MMPs are necessary to break down extracellular matrix molecules. So if TIMs are high, then MMPs cannot work, and the extra collagen that's deposited in a NASH liver cannot be removed. And so we want to change that by a pharmaceutical intervention. And you see that as we add the anti-TGF beta-1 antibody, we see a substantial reduction in TIMP1 and TIMP2. So again, correlation to the clinical findings for this antibody. Now, the TIMs and procollagen have another important significance for us because they are the two of the three aspects of the so-called ELF diagnostic test. The ELF test has been developed by Gilead and Siemens. It's one of the few non-invasive diagnostic tests for NASH. The third quantity in that assay is hyaluronic acid. So PC3, TIMP1, and hyaluronic acid together are part of this ELF test. And this is kind of where we go full circle because we want to have readouts in the in vitro case that have a clear relevance to the clinical setting. Now, what are we doing about fibrosis? We are able to look at procollagen in the supernatant. It's a precursor of the collagen deposition. But of course, we really want to have a good understanding on a morphological level what happens in these microtissues so that we can assess the impact of a drug on fibril deposition and also the formation of fiber networks in these systems. We've started about 1.5 years ago to collaborate with a company in the US called PharmaNest, and they've developed an artificial intelligence or machine learning based approach to quantify fibril networks on sections to allow us to get a better understanding of how a drug intervention changes the morphology of the fibers that are deposited in the system. And this is quite complex. You see the serous red staining for fibers on this image on the left side. If we're again just doing a pixel quantification, we're losing a lot of biological information that's actually contained in these models. So the quantification approach in the FibroNest system consists of 3 different buckets. The first bucket is how much fibrils do we have and what is their overall structure. The second aspect is what's their shape. That's the morphometric approach. And then the third aspect is where are the fibers across this tissue and how are they organized? Are they fully assembled? How many nodes do they have typically per fiber and so on. So using that approach, we get about 350 to 400 unique quantifiable fibrosis parameters. And based on these parameters, we've developed a compound score that takes basically a weighted sum of 37 of these parameters to create a compounded index that gives us a measure of fibrosis that we have in the system. So how does that work practically? You see the serous red bright field image on the top with already a visible change or visible reduction of fibril deposition as we add our TGF beta-1 antibody to the system. But the FibroNest software now allows us to really understand what kind of morphological changes we see. So in the second line, you see in pink the assembled collagen and in green the fine collagen. The fine collagen fibers are fairly young collagen fibers that are not yet fully embedded into assembled networks. They are an important drug target for us because they are much easier decomposed by MMPs in the system than a much more solid and rigid assembled fibril network. And then on the lower end, you see dense and faint collagen. So we get a lot of extra information from this machine learning approach. But of course, we want to ultimately have a kind of understanding of what happens to these tissues. And that's where a heat map is really quite helpful. So you see the control NASH and then 2 concentrations of the TGF-beta antibody. Each column that you see here is a different region of interest, and the region of interest for us is one tissue in the section. We are typically embedding 12 to 16 tissues into one block before we section it, so that we have a bit more histological real estate to generate data from. And the algorithm is automatically identifying the tissues and then numbering them as regions of interest. And so what we get are very interesting fingerprints that show us how specific drugs change these sets of parameters in their specific way. So you see the control on the left side. In NASH, all of the parameters basically get red. And then we see a concentration dependence as we add the anti-TGF-beta antibody to the system. So a big question here is, what is the physiological meaning of these different parameters? How does it actually link to clinical aspects, and how does it link to patient outcomes? And I'll come to that in my last slide, because this is a subject of current investigation with our partners. The main focus on how we use this approach at the moment is that we are using different compounded indices that are basically drafting information from a selected subset of the 400 quantifiable fibrosis parameters, and then give us a compounded score that is a figure of merit for us to decide, is this a drug that has an antifibrotic effect or not? And is that effect dose dependent, which are 2 key criteria for moving forward with a drug in the clinical trials. Now, where do we want to take this technology, and why are we so interested in scaling it up, both on the biological side, by being able to have miniaturized uniform tissues in 384-well format, but also on the assay side to be able to generate data at fairly low cost. The reason is that we believe that a key to solving the NASH problem in drug discovery is to use drug combinations to address the different mechanistic effects that lead to the disease. NASH is an extremely complex disease that involves dietary aspects, cardiovascular aspect, metabolic aspects, gene predisposition. So to be able to attack such a complex disease, most likely we need more than just a single compound, but a combination of compounds. And this makes the whole drug discovery aspect a lot more complex, and it requires a lot more data. And that this is actually helpful, we've shown in a study together with Gilead. You see the results here at the lower end here. What we did was looking at elafibranor and firsocostat, both drugs that didn't really make it through the clinic. You see that each of the drugs, this is control, this is NASH, each of the drugs in itself is not very powerful, but when we start combining the 2 drugs, we see a synergistic effects by this drug-drug combination. And this, for us, is a way to go, also with other drugs under investigation, to see how we can improve the efficacy by combining them in the right combination. So to be able to do that, we need a high throughput compatible solution. We need to be able to measure the hallmarks of NASH in a highly integrated, low-cost biochemical assay setting, but we also need to be able to verify our findings in clinical relevant readouts. And all of these aspects we can fulfill with a fairly simple and easy to scale up spheroid system. As I mentioned, one of the aspects of investigation is how can we relate our in vitro findings to actual patient outcomes. And we have a poster about this at the EASL conference in 2 weeks, collaboration with FibroNest and Arun Sanyal at Virginia Commonwealth Hospital, where we are taking data from 19 NASH biopsies from actual patients and then benchmarking them against our in vitro findings. And what I can already say is that 90 out of these 400 fibrotic parameters show an excellent correlation with clinical outcomes. So we're extremely happy about this. Let me thank the liver disease group that at Inspiro that made all this work possible. Francisco Verdeguer is our VP working on this with his team, and also Mathieu Petitjean and his colleagues at PharmaNest here in the US for helping us with the machine learning. And thanks very much for your attention.
We have time for a question or two, and then there will be a brief discussion of the session. One of the great advantages of organoids is that you have lots of them. So if you look at the multi-drug combinatoric problem, have you done a power analysis to figure out how many organoids you would have to do to design a multi-drug cocktail?
Yeah, that's a great question. So we haven't looked at it from this angle. The way how we want to approach the combination testing is actually on a more iterative level. So by selecting compound sets with different modes of actions to see which type of compound in combination is moving the needle, and then improving these compound sets iteratively as we go to zoom in on a combination that works.
There are questions, though, about if you have nonlinear interactions, that becomes a little bit more complicated on the iteration.
Agreed with that, and we've seen that in a different context in combination treatments where the response was nonlinear in the sense that sometimes lower concentrations were actually showing higher efficacy than higher concentrations of combination drugs. So there is a risk with this approach that we are kind of falling into kind of a local maximum, but we're not really getting the best combination. But I think that's a bit of a trade-off between the size of the screen and the capabilities that we have.
Okay, thank you very much. Oh, we have a question. Good, great.
My name is Job Bomen from the University of Twente. Thanks for the very interesting talk, and especially comparing it to the pathology in patients. What I was wondering, do you consider having a test and a train set with the patient samples? Because you're now going from 100 parameters in all potential combinations and permutations.
Yeah, definitely. So I think in the current setting from the 90 biopsies, and this is not a large number, I'm totally aware, right? We've selected 15 for a blinded validation test. Or qualification test, I should rather say, and the others are used for training the system.
And these biopsies, how do they relate to the clinical outcome?
So the biopsies are primarily at the moment relating to the stage of NASH that the patient has, right? So as we don't really have a pharmaceutical intervention against NASH, it's hard to, we don't have responders and non-responders, right? But we have different progression of the disease. And our main interest at the moment is that we are able to basically classify the 3D tissues into the same buckets as a human pathologist would classify the biopsies. That's limited to a certain extent because clearly the tissues are quite small. They lack some of the features of a biopsy. You don't have a portal vein, for instance. So I think there is still a bit of translation that we need to do. But on the other hand, a big benefit is that our calculated index is continuous. So we're not just looking at like 5 classifications, 1 to 5, but we can also have a much more granular assessment. And I think this will be crucial to see these fine changes that some of the pharmaceutical interventions will entail.
Thank you.
I can't imagine that Lance has anything to say about the liver, but I would love to hear his question. Lance.
Jan, very nice talk.
Thank you.
Are you able to control the oxygen tension in your plates? And what is the oxygen tension? And does it relate more to zone 1 or zone 3? Because there are differences.
That's a great question. So we don't see zonation in these tissues. It's also not really expected due to the size of the tissues. But it's been a recurring topic for us. So I think one of the interest that we have on the microfluidics aspects of microphysiological systems is actually at some point creating a liver system that would allow us to show sonation in that device. And if you have a good idea, Lance, then I'm happy to pick it up.
Use the mic.
Lance,
Yeah.
can you use the microphone?
So Lance's point was uniformity and how you could drive a single tissue into a specific zone. So that's something that we're currently not doing. We're keeping constant oxygen tension. Yeah.
Well, one of the interesting questions following up on Lan's comment, or request for information about whether you're doing oxygen tension, is whether in fact zonation can be studied with independent zones. So you build a plate that's oxygenated and a plate that isn't, or has lower oxygen and even lower for, let's say, the third zone, and the extent to which they have to talk to each other or can be treated independently. I would like to hear from either you or Lance about the liver physiology on things like zonation.
Yeah. So I think the two possibilities to address this is, as you said, it could be a discrete system where we have different types of tissues that are cultured under different conditions, but you could also imagine a tissue culture that is that has an imposed oxygen gradient, so that you can drive the system as in a holistic manner in this field. I think our primary interest here is building a system that allows us to run a screen to find compounds against the disease in an efficient way. So we've decided not to add these extra layers of complexity to the system. But from a scientific perspective, I totally see the point. Yeah.
But in the Insphero plates, you have serial perfusion of organoids. And the net result is that there can be metabolic gradients along that flow, depending upon your flow rate. Because Kim and Ingber, looking at the CACO2 cells, clearly showed that there was a difference in expression, or this happened to be of villi, but it is a general phenomenon along the length of a channel. So to some extent, you have a situation with spheroid after spheroid after spheroid, where there's going to be, by definition, a metabolic gradient, depending upon your flow rate.
Yeah. So the work that we do here is under static conditions in the 384 well system, right? So we have no flow because again, this is a screening setup. But point is well taken. We also have the Acura flow system where we are lining up tissues. Olivier and his team and our colleagues at ETH have done quite a lot of computational fluidics analysis. So it really depends on the cell size versus medium volume that you would really see a depletion of specific nutrients or an increase of, let's say, metabolic byproducts, right? But this definitely plays a role. Yeah.
Okay, I hate to break off such a stimulating discussion, but we have to keep to time so that the next session can start. So I'd like to thank again all of the speakers in the session and you for your attention. And while we're transitioning to the next section, I'll just remind folks, if you haven't heard the announcement already, that for the reception this evening, there are 3 shuttle buses that will be leaving from the front of the Hilton at 7:45 to take you to the reception. And we'll be coming back at various stages throughout the evening. The last one's coming back at 10 p.m.
Sit down. So welcome everyone to the last session, symposium session of the day, which will be on reproductive system MPS. And I'm very excited to share that session together with Ben Capiello from Axozim. And my name is Lena Smirnova, Johns Hopkins University. And without any further introductions, I don't know why people are all leaving. It's the wrong direction, guys. Anyways, I will introduce our first speaker, who is Lauren Richardson from University of Texas Medical Branch. So Laura, yours.
Hi, yeah, please stay. I was so excited to see that there's a reproductive health section in this conference. You know, women's health needs a lot of research, so I'm very excited, and thank you so much for allowing me to come speak today on this very important topic. Let me make sure I can get this going. Okay, yes. So I'm gonna be using the pointer on this screen, just so if you want to follow along. All right. So throughout gestation, multiple intrauterine organs develop alongside the growing fetus to allow it to support and grow and mature. But closer to term, around 40 weeks gestation, this is when fetal and maternal signals will increase the inflammatory load, lead to immune cell activation. This will weaken the fetal membranes that you see there in yellow, allow for cervical ripening and myometrial contractions, and eventually happy delivery of the baby. Now, these systems are totally normal and happen physiologically at 40 weeks, but they can be induced pathologically before 37 weeks, and this is called preterm birth. Now, preterm birth is a global crisis. 1 out of every 10 babies is born premature. And even in 2021, the rates of preterm birth in the United States are on the rise. Now, one of the most common phenotypes that induces preterm birth is called intrauterine infection. And this is when bacteria ascends from the vaginal cavity, crosses through the cervix, transports through the fetal membranes, and reaches the amniotic cavity where the baby will be sitting. And so this infection, as it propagates, also increases inflammation and reactive oxygen species that then leads to almost 40% of preterm births. But regardless of the pathology that induces preterm birth, these premature babies now have increased risk for adult onset diseases such as neuro or cardiovascular disease, early onset asthma, diabetes, and even certain types of cancers. And these topics might sound familiar to you because a lot of you are studying them on chip now, but they could be pre-programmed in these babies in utero. Now, during gestation, there are 2 organs in particular, the placenta and the fetal membranes, that play very vital roles as the fetal maternal interface. And while the placenta is critical for fetal survival and growth, the membranes are also very important in the role that they play to induce labor signaling. So because of this, the fetal membranes are going to be the topic of my talk today. Now, the membranes are comprised of multiple cellular and collagen layers. This is not a simple tissue that just surrounds the baby in the intrauterine cavity. The tissue is comprised of an amnion epithelial layer that contains amnion epithelial cells, or AECs, and a chorion trophoblast layer that contains chorion trophoblast cells, or CTCs. And these 2 epithelial layers are separated by multiple collagen layers that contain mesenchymal cells, or AMCs. Now, these 3 fetal layers are connected to the maternal decidua that used to be the endometrium. And these maternal DEC cells connected to the fetal cells create the fetal maternal interface. Now, during gestation, the fetal membranes grow and they undergo constant remodeling to maintain that structural integrity of the intrauterine cavity. And they play very important roles on the immune, endocrine, and mechanical functions that are required to support a pregnancy. Now, in particular, the amnion layers, these first 2 layers of the fetal membranes, play key roles in the mechanical integrity, by undergoing cellular transitions, such as epithelial to mesenchymal transition, or the reverse, mesenchymal to epithelial transition. So they remodel the collagen, they strengthen it, they fill gaps, they maintain that barrier that's needed. While the chorion decidua interface, on the other hand, that lower layers, they play very important roles in immune regulation. They have high expressions of human leukocyte antigens that keep maternal immune cells at bay during gestation. But at term, what we know is that these two processes go awry. You have increased EMT, or epithelial to mesenchymal transition, in the amnion that degrades the collagen and weakens the membrane. And you have a downregulation of HLA-Gs that now allow for the maternal immune cells to infiltrate through the tissue, increasing that inflammatory load, promoting those labor signals. But how do we study these types of processes in the lab? Now, traditionally, you might start off with just a single 2D cell culture system to test a method or an idea. If you're lucky and you're connected to a hospital, maybe you can collect some biopsies or explants and study the tissue ex vivo. But in tradition, you have to go to a small or large animal study to really see what's going on at the fetal maternal interface. But there's a problem with this. And the problem is that the fetal maternal interface has a very different function and anatomy in animals versus humans. So what we really need is an advanced human-based cell culture system that can fill this gap. And for us, that is organ on chips. So today I'm going to tell you about our most advanced organ on chip that recreates this fetal maternal interface, or the FMI-OC. So our FMI-OC device is comprised of PDMS, and it contains 4 concentric cell culture chambers that are connected by arrays of microchannels. This also contains an on-top gravity-fed media reservoir for long-term culture. Now, we have recreated every collagen and cellular layer of the fetal maternal interface within this device, even going down to the detail of filling the microchannels between the amnion and chorion with basement membrane collagen, or allowing the microchannels to be left open, as you see here, so that the cells will form that nice, tight cell junction interface between the chorion and decidua. Now, within each of these circular chambers, we're able to grow primary cells that we've isolated from cesarean deliveries of the fetal membranes, and then we've immortalized them and characterized them so that we have set established cells for these chips. These cells are grown in a mixture of 2D and 3D cultures. We have AECs in a monolayer. We have AMCs and CTCs in a 3D growth that we culture with primary collagen and matrigel. And we have maternal DECs in the center that's also in a monolayer. Now, these human cells can maintain their morphology, their collagen production, seen here in purple, their intermediate filament expression, like vimentin and cytokeratin, and also their viability on chip. And since we've previously validated this device for its physiological model, in this talk today, I want to focus on how we're advancing this system to study pathologies. So our 2 objectives for this project was to, 1, model ascending infection. And so that is when you have a maternal treatment and we're looking for a fetal response. And the second objective is to recreate intraamniotic infection. And that's when you have a fetal infection and you look for the maternal response. So to start off our objective 1, we were able to establish culture within this device by loading our microchannels with matrigel the previous day and allowing it to cure. On day 2, we load our cell and collagen mixtures into the appropriate channels and fill the media reservoirs. Then, to model ascending infection, we use lipopolysaccharide, or LPS, to mimic infection on chip, and we treat 100 nanograms per milliliter into the center maternal decidua chamber. We then monitor LPS propagation, cellular level changes, and inflammatory profiles over a 72-hour period. I'm Immunocytochemistry confirmed that LPS propagated from the maternal decidua chamber to the amnion mesenchymal chamber within 48 hours, as seen by these little red dots. And they also reached the fetal epithelial chamber within 72 hours, showing a propagation of infection from the maternal to the fetal side. Additionally, we looked at a prominent LPS signaler receptor, toll-like receptor 4, and saw that it too followed that same LPS propagation timeline as seen in this green fluorescent staining as it propagates from the maternal to the fetal compartments and quantified in this graph. Next, we wanted to look at cellular level changes. And so what we noted is that maternal LPS treatment did not induce cellular transitions like EMT in the amnion, as noted by the static levels of vimentin and cytokeratin ratios, but it did significantly downregulate HLA-G expression in the chorion after 72 hours. And so this shows us that maybe maternal LPS treatment could be inducing immune dysregulation at that choriodecidua interface. Next, we wanted to determine what maternal LPS would induce in the inflammatory profile across our device. And what we noted is a very cell and time-specific cytokine production that created a pro-inflammatory environment of interleukin-6 and IL-8, and GM-CSF across this device, starting in the maternal side and also all the way to the fetal side. So in summary for this objective, we were able to document that this infectious agent can propagate from the maternal to the fetal compartment and that it induces a fetal inflammatory response, such as increased TLR4 and downregulated HLA-G. We also saw that this inflammation was not associated with cellular level changes in the amnion. Next, we focused on our second model. This is a model of intra-amniotic infection. So we established culture on this device in the same manner as objective 1, but now we're treating lipopolysaccharide in the outer AEC chamber. We monitored LPS propagation and cellular level changes and inflammation over 72 hours. Immunocytochemistry confirmed that the fetal LPS traveled to the CTC chamber within 48 hours and to the maternal DEC chamber within 72 hours, showing infection propagating from the fetal to the maternal side. Additionally, TLR4 expression followed this LPS propagation in a time-dependent manner seen by this green staining here. When we focused on the cellular level changes, we noticed this time that fetal LPS treatment did significantly induce EMT in the amnion layers, as noted by this significant increase in vimentin to cytokeratin ratio and increased mesenchymal morphology in the amnion. But it did not change the levels of HLAs in the choriodecidua layers, suggesting that this fetal LPS could induce cellular transitions and weaken the membranes. Next, we wanted to see how fetal LPS would induce an inflammatory profile across this device. We noticed time and cell-specific cytokine productions, and in this case, specifically IL-8 and interleukin-6, that was present in the fetal amnion layer as well as the maternal decidua layer. And this was over a 72-hour time point, showing propagation from the fetal to the maternal chambers. So in summary for this objective, we were able to show that infectious agents can propagate from the fetal to the maternal chamber of our chip, and that this induces maternal inflammatory responses such as TLR4 and cellular transitions in the amnion layer, but it did not affect HLA-G expression in the chorion decidua. So lastly, we decided to validate this FMI-OC model by conducting in vivo mice testing to go along with our ascending infection model of our FMI-OC. So to do this, we treated CD1 mice on day 15 of pregnancy, vaginally with live E. coli with 10th to the 11th power colony forming units. And we monitored the ascension of this live E. coli through the uterine horns from the proxial to distal horn over 6 and 24 hours using IVUS imaging. Here you can see the IVUS imaging showing that after 6 hours, the DID stained E. coli was propagating from the proxial to distal horn, and by 24 hours, it had completely covered that horn. And along with this, when we looked at the fetal membranes, what's interesting is that these membranes at the proxial horn, the closer horn, were showing increased TLR4 and also phospho-NF-kappa B signaling, which led to inflammation and preterm birth in this model, deemed by delivery before day 18.5. So these pathways are the same pathways we're seeing in our ascended infection model on CHIP of TLR4 inflammation and weakening of the membrane leading to preterm birth. So in conclusion, we have now developed this FMI-OC, and we're able to recreate a healthy and diseased phenotypes of pregnancy. And we're able to do this because we have these well-characterized, genetically stable cell lines that we can culture within this chip, and they can maintain their intracellular interactions and provide distinct responses that allow us to make these different states. Additionally, we've conducted in vivo testing to validate this FMI-OC model and to try to prove its utility to model in utero events. And hopefully this will allow for a decrease in animal models and studies in the future. Lastly, since we have these two types of disease, the physiological and pathological states of this chip, we'd like to start to conduct preclinical testing of various agents in the hope that we can reduce these adverse pregnancy outcomes. Now, there are some limitations with our device. We don't have resident immune cells within the decidua of our chip, but we are actively pursuing to add immune cell components to this device. And as of now, we are using a static gravity-fed media reservoir instead of dynamic flow. So these are some things that we are pursuing and looking into advancing our model with. And so with that, I would like to thank the multiple labs that and the people that have contributed to this project. This project is a collaboration with Dr. Menon's lab at UTMB and Dr. Hahn's lab at Texas A&M. And specifically, I'd like to thank the people in red that have contributed majorly to this project. Also, NCATS for a good chunk of the funding for this, as this is part of our UG3 section of that grant, NICHD, and other small pilot projects. And with that, I will take any questions, and thank you for listening.
Great, thanks, Lauren. Fantastic talk. Can we have some questions come up here? Here comes Dr. Griffin.
Hi, it's wonderful to see work on the fetal maternal interface at this meeting. So congratulations on a great talk. One of the things we struggle with a lot in working with these models in vitro is how to manage the steroid hormones, particularly progesterone, which is very rapidly metabolized. So we tend to often use MPA, which I'm not super happy about. But the other thing is that in experiments where we use PDMS, such as Ellen Kahn, the student who gave a talk earlier, we have to work really hard to think about the design of the experiment because progesterone just soaks into PDMS like there's no tomorrow.
Okay.
And so I'm wondering, because these issues with preterm birth are often associated with progesterone resistant phenotype, which comes with inflammation. How are you managing to grapple with that progesterone issue in your cultures? Because progesterone is such a huge part of the pregnancy. Of the decidua.
That's such a great question. And so I think that we're able to actually slightly overcome that in a couple of ways. Since our cells are primary derived, we've actually measured their production of progesterone naturally. They produce a lot of progesterone, specifically the chorion and amnion layer, continually throughout their cycle as a cell. And from being at this conference, we now know a lot of the limitations of PDMS, and I think that we will try to get away from that. But we are seeing that, the cells are functioning the way that we're expecting. And I do know that there's a lot of kind of controversy on, like, do you need to add progesterone to prevent preterm birth, or, can you use these treatments? We've also detected the progesterone receptors, the membrane progesterone receptors in these cell layers, and to document that they're not decreasing, and we know what they should do during oxidative stress. So with your concern, I think that we will go back and validate and make sure that those progesterone receptors are maintaining the right characteristics and expression on chip to make sure the progesterone is acting normally in this system. So I thank you for that comment.
It's a really hard problem, and we can talk about it later because it's really gnarly, and there's a lot of characterization you have to do. So we can continue discussions throughout other conferences too, because it's an ongoing challenge.
Yeah, thank you for that. Yes.
I'm sorry, I really like the discussion about the experts, but we have to move forward with the next speaker, and we can address other questions maybe after this session. So our next speaker is from Binns Institute, Harvard University. Zore Izadifer.
Good afternoon, everyone. So in my talk, I'm going to talk to you guys about the human cervix and vagina chip models that we have developed in our lab to model the human host microbiome interaction, as well as the physiology of the mucus. So in the female reproductive tract, the two main organs in the lower part of the, So the two main organs of the lower female reproductive tract of the cervix and vagina, and their main role is to provide a gateway to protect the upper female reproductive tract from the direct exposure to the outside environment and the pathogenic invasion. And also to regulating the passage of the sperm during the fertilization time, but more importantly, to protect the upper reproductive tract from the pathogens that I mentioned. And cervix and vagina do this function through these two main approaches here, in addition to anatomically constrain the access of the outside environment. As you see, the channel here is pretty narrow to separate the uterus from the outside, but they do it through these two main approaches that I have mentioned here. The physical barrier that's done through the epithelium, the impermeable epithelium that covers the surface of the vagina and the cervix, and also the mucus layer, which is an important component in the female reproductive tract. The cervix tissue is basically the factory of the mucus production in the female reproductive tract. It provides a physical barrier on the surface of the cervix and vagina that separates the interaction of any particles and substances that want to reach the epithelium, and that protects the epithelium and the upper reproductive tract from those invasions. The mucus layer also constitutes of a lot of antimicrobial properties that also contributes significantly to the health defense mechanism of the reproductive tract. The other main component here is the host microbial barrier. As we know, on the surface of the epithelium, we have a microbiome, we have a microbiota, a population of the bacteria that are residing on the mucosal layer. And in the female reproductive tract, the most abundant type of bacteria is Lactobacillus species. And these Lactobacillus species, they maintain a stable and healthy symbiotic with the epithelium, which the epithelium provides them with nutrients, and they in exchange provides the antimicrobial properties against other pathogens. For instance, the Lactobacillus crispatus is a lactic acid bacteria, so it produces a lot of lactic acid in that microenvironment, which reduces the pH of that environment, make it acidic, and as such inhibitory to the pathogens. These bacteria also produce a lot of immunomodulatory components that also contributes to the health mechanism of the cervicovaginal microenvironment. So as I mentioned, a healthy balanced microenvironment in the cervicovaginal constitutes of the epithelium, covers with a layer of mucosal layer on top, And all these residential commensal bacteria, which is dominated with Lactobacillus species. The Lactobacillus species constitute of 99% of the bacteria that are there. So they outweigh any other opportunistic bacteria that they want to thrive on that microenvironment, and keeping the population under the control. And this is called the symbiotic microenvironment. But what happens is that once that symbiotic microenvironment is disturbed, then these opportunistic bacteria start to take over. They deplete the population of the Lactobacillus bacteria, and they start to replace that microenvironment with a range of different pathogenic bacteria that are mostly anaerobes. And what happens is that anaerobic bacteria produces a lot of sialidases, proteinases, and mucinases enzymes that what they do, they attack this mucosal layer, they destruct that physical barrier, they make direct contact with the epithelium, which colonizes inside the epithelium and then calls a lot of immune cells to come. Infection happens. Then these pathogenic bacteria colonize there, form a thick biofilm on top of them, which makes them very difficult to get rid of. This condition, the unbalanced dysbiotic condition, clinically well known as bacterial vaginosis. Unfortunately, BV affects over 25% of women in their reproductive age, which is like just in US, about 21 million women. And unfortunately, the only clinical treatment that's available is antibiotic, metronidazole, which what happened is women get that treatment, they go back, they're good for some time, but 60% of them come back within 6 months. And this condition never gets completely removed. And as Laura mentioned very nicely, it has various health adverse conditions, including preterm birth that Laura mentioned, spontaneous abortions, higher chance of getting HPV, HIV, and other health inflammatory diseases in the pelvic. So the big challenge here is, the problem is we don't know what's going on there. That microenvironment is very complex because there are different factors contributing to the physiology and pathophysiology of that microenvironment. And for years, I would say over 70 years, this condition is known, but the pathophysiology of it is really not understood well. And as such, they can't come up with the better treatments. So what we did at WIS, we decided to make organ-on-chip, use organ-on-chip technology to make human vagina and cervix tissue organ-on-chip models to look at the host microbiome interactions. And we used the emulate dual-channel microfluidic device and recreated the epithelial stroma interface on this organ-chip model here. We plated the epithelial cells, vaginals, and cervical cells on top of the ACM-coated membrane, and the stromal fibroblast cells on the bottom of the channel. And we used an optimized method that we developed in-house, and within 12 to 15 days, we are able to create a thick epithelial, vaginal, and cervical layer on the chip with supported stromal cells on the opposite side of the channel. We characterize the vagina chip, and the vagina chip shows characteristics of a stratified in vivo-like phenotype. As you see here in the cross-section of the chip, we see a stratified epithelium that has the basal layer and also showed a well-maintained barrier functionality throughout the 15 days of the culture, as we show here for 2 different donors that we tried. The epithelium also shows main markers of the vaginal epithelium, including CK5, CK14, and involucrine. One of the main proteins that the vaginal epithelium secretes is the glycogen. Glycogen is the main source of food for the commensal bacteria in that microenvironment. And we found that the vagina chip produces glycogen that we can collect in the effluent of the chip. And as you see here, increases in concentration with time of the culture. As I mentioned, the cervix tissue in the body is the factory of the mucus, and that's one of the main functions of the mucus in that microenvironment. Sorry, the main functions of the vagina chip, cervix in that microenvironment. So here you see the cross-section of the cervix chip, the side view of the main channel at day 5 of the culture, when we have cells on their expansion. So we have only one layer of the cells there. And as you see, there's no mucus layer formed there. But by day 12 of culture, when we are well in differentiation, we see formation of a thick mucosal layer on top of the epithelium. which is also positively stained with beta-germagglutinin, which is a lectin binding marker that binds to the mucin type 5, the primary source of mucin in the cervical mucus. In collaboration with Dr. Lebrila's lab at the University of California, Davis, we performed some glycomic analysis of the mucus. And here you see a comparison of the N-glycan and O-glycan analysis on the mucus that we collect from the cervix strips as compared to the clinical mucus. And you see here that we can very nicely recapitulate the diversity of the glycosylation of the mucin that we see on the clinical mucus. In collaboration with Dr. Rebeck's lab at MIT, we also developed a method for non-invasively look at the biophysical property of the mucus on the chip. As we know, biophysical property of the mucus is one of the mechanisms of defense in that microenvironment. So our method allows us that during the culture, we can look and see how the biophysical property of the mucus is changing. And in this case, we just looked at the mucus at the end of the culture time, which is like 12 days of culture, and compared that to the clinical human mucus. And we found the diffusion constant of the mucus that we get on the cervix strip is actually comparable to that of the clinical mucus and much better than the mucus that we get from the static transfer culture that is commonly used in studies. Menstrual cycle, we all know what happens. There's changes in the sex hormone levels, including the estrogen and progesterone. And these changes in the hormone levels have a major role in changing some of the physiological functions that are happening in that microenvironment. And they are directly correlated with the composition of the microbiome in that microenvironment, and as such, to the health defense mechanism of that microenvironment. So it is important for us to be able to recapitulate that on the organ chip model. So we treated the chips with two different hormone levels, ovulatory and non-ovulatory. So ovulatory is obviously when we have very high levels of estrogen, and non-ovulatory luteal phase, high level of progesterone. And we observed that in this side view imaging, you are seeing that the thickness of the mucus is significantly higher than that of the one we get in a non-ovulatory phases. As you see here, were quantified based on the images. Also, biophysical analysis of the mucus showed that when we have non-ovulatory hormonal regime, which is high progesterone, the diffusion property of the mucus significantly reduces compared to the ovulatory phase, which is similar to the trend that we see in the human clinical mucus as well. And this is pretty obvious because during the ovulatory phase, the transfer of the sperm, the environment needs to be ready to transfer the sperm, so it becomes more diffusive. In addition to that, we looked at the glycomic analysis of the mucus. And this is, again, in agreement with what is observed in vivo, that in general, when we are in the ovulatory phase with high estrogen, we have higher abundance of O-glycosylation and N-glycosylation on the mucus compared to the non-ovulatory phase. We also have developed an organ-on-chip system in-house that we integrate transepithelial electrical resistance sensors on them. So we can dynamically monitor how the electrical resistance of the epithelial layer is changing with time. And we use that for our system here when we are developing the cervix strip model under the two hormonal conditions and look at how the barrier function is changing. And as you see here by day 11 of culture, we see a significant difference in the barrier of these two models. And that is again in agreement with what we observed in vivo. During the ovulatory phase, there is higher progesterone. The tight junction loosen up because they want to allow transfer of more fluids from the interstitial tissues to the cervical lumen to increase the water content on that microenvironment. So after we characterized the two models, the next step, of course, we wanted to look at the host microbiome interaction. That was the main goal in the beginning. So in collaboration with Dr. Ravel's lab from University of Maryland, we used clinically isolated optimal Lactobacillus crispatus strains that were collected from women that have a stable Lactobacillus dominant communities for years. And they were collected and we inoculated the vagina chip with either a single strain Lactobacillus crispatus or 3 different consortia of 3 or 4 of these strains. So we inoculated the vagina chip with these different bacteria and co-cultured them for up to 3 days. And we observed that at the end of the 72 hours, we are able to maintain a stable population of the Lactobacillus crispatus consortia on the chip compared to the inoculum. Interestingly, when we have Lactobacillus crispatus population on the vagina chip, the pH level of that microenvironment is maintained within 4, which is an acidic level, and it is comparable to the pH level in the vagina in vivo. Also, the strain ratio analysis showed that the 3 days of co-culture of this consortia on the vagina chip does not change as much the ratio of these different strains that we are using on the chip. In fact, the microenvironment that we have created on the vagina enables for maintaining of this ratio between different strains. Also, cytokine analysis shows that the presence of the Lactobacillus crispatus and its colonization for 3 days significantly reduces the pro-inflammatory cytokines here, presented here as IL-6, IL-8, IL-1 alpha, and IL-1 beta, both for the consortia as well as for the single strain. Also, we know that the lactic acid, as I mentioned, is one of the main components that the Lactobacillus crispatus secretes in that microenvironment and contributes to the defense mechanism of that the cervix and vagina environment. And we found that we can tell the difference. Sorry. When we have among these 3 different consortia that we tested, 2 of them were found to actually induce production of D-lactate. The lead lactate is one type of the lactic acid that's specifically contributing to the defense mechanism, and we could recapitulate that on the chip. So we established a model for a symbiotic microenvironment, and we wanted to see if we can model a dysbiotic microenvironment as well. So we used this isolated dysbiotic bacteria, again, from the women that have dysbiosis, and we come up with 2 different consortia called here BVC1 and BVC2. The BVC1 is a consortia that consists of 2 strains of Gardnerella vaginalis and Prevotella bevia and Autopobium vaginae, and the BVC2 is just the 2 strains of Gardnerella vaginalis. And again, we co-cultured them with the vagina chip for about 72 hours, and we found that we can maintain the population, this population of bacteria on the chip for both of these consortia for the 72 hours. And the pH level on the vagina chip for one of these consortia significantly increased. This is interesting because in the clinic, one of the major diagnostics approach is to just measuring the pH. And once the pH on the chip in the patient is high, that's one of the indications that they have developed BV, and we were able to recapitulate that on the chip as well. And as you see here, the presence of these dysbiotic bacteria also caused cytotoxicity. The number of the mammalian cells also significantly reduced. And also cytokine analysis shows that with the presence of this dysbiotic bacteria, we see a surge of inflammatory response from the epithelium for all of these 4 cytokines that we looked at. So we also went ahead and developed the symbiotic and dysbiotic model on the cervix chip. And here you see that we selected one of the L. crispatus consortia and one of the dysbiotic consortia, mainly the G. vaginalis, inoculated them on the cervix chip and maintained for 3 days. And here you see that these models, we are able to, on a daily basis, look and see how much bacteria are actually coming out of the chip. So this enables us dynamically monitor what's going on the chip. Here you see the quantification of the bacterial load in the effluents as well as at the end on the chip. The pH level, interestingly, on the cervix chip didn't change as much, either for the C6, which is the Lactobacillus consortia, as well as the G. vaginalis. And this is, again, in agreement with what we see in vivo. In vivo, the cervix has a lot of mucus, and mucus is known to have a buffering capacity to control the changes, the significant changes in the pH. And we also were able to recapitulate that on the chip. And on the right here, you see a representative bright field image of the cervix chip. It's hard to see here, but I hope you can trust me that there are Lactobacillus crispatus colonized on the epithelium. Here is just the immunofluorescent staining of the epithelium that you see with the presence of the G. vaginalis. The integrity of the epithelium is completely destructed. And the lower number of the mammalian cell is also observed on the chip. And continuously monitoring the barrier function on the chip shows that with the presence of the G. vaginalis, at 72 hours, we see a significant compromisation of the epithelial barrier on the cervix chip. The cytokine analysis also showed that the presence of L. crispatus consortia on the cervix chip minimally changes the cytokine levels on the cervix chip, some improvement with TNF-alpha. But with the presence of the G. vaginalis, we see again a huge increase in all the 4 studied cytokine levels on the cervix. Having the mucosal layer gives us the advantage to look at the interaction of the bacteria and the cervical mucus on the chip. Here you see a cross-sectional image of the cervical layer, and the middle and bottom. The middle one is where the chip is inoculated with the L. crispatus, and the bottom is G. vaginalis. And I hope you can appreciate some of the qualitative changes that are happening in the cervical mucus with the presence of these 2 types of commensal and despotic bacteria. And when we quantified that, we found that the thickness of the mucosal layer when we have L. crispatus is significantly higher than the 2 other groups. And also the intensity of the mucus signal is significantly higher than when we have the G. vaginalis. And the biophysical analysis also showed that the presence of the L. crispatus significantly reduces the diffusion constant and diffusion diffusion constant and diffusion exponent of the mucus, which both of them indicating that when we have L. crispatus, the mucosal microenvironment becomes more constrained and more confined, which is one of the major defensive mechanism of the mucus. It becomes less diffusive, so the bacterial and pathogens and virals can have minimal activity inside of the mucus. And also the O-glycomic analysis showed that the presence of the G. vaginalis significantly reduces the O-glycosylation profile, and specifically the sialic acid O-glycosylation. And again, this goes back to having G. vaginalis has a solidases enzyme, and that directly cuts off a lot of sialic acid from the mucus. And in summary, I can say that the vagina and chip models that we have made represent physiologically relevant and functional models of the human vagina and cervix tissue in vitro. We can use these models for studying the physiology and pathophysiology of the cervical mucus and the host microbiome interactions, both for healthy and disease situations. And these models also enable us to look into the contributions of the different ethnic backgrounds and different genetic contributions that peoples from different parts of the world have in the disparity of the disease and health condition, as we all know. And also these models are currently being used as potential preclinical models for evaluating new biotherapeutics. And at the end, I want to acknowledge all of my team members at the VIS and also collaborators at MIT, University of Maryland, University of California, Davis, Boston Children's Hospital, and Massachusetts General Hospital, and the Bill and Melinda Gates Foundation for the funding. Thank you very much.
Great, thank you. Unfortunately, we won't have time for questions here, but please come talk with Zora afterwards. Sure, thank you. Next up, our next speaker will be Shu Zhao from Rutgers.
Okay, good afternoon, everybody. At first, I would like to thank the organizing committee for having me right here. So today I will briefly introduce how we use the alginate hydrogel encapsulation method to make the mini ovary model, and how we use this model to study the female reproductive biology and also toxicology. Okay, so our first 2 speakers already give you a brief introduction about the downstream female reproductive function, female reproductive tract. In our lab, we primarily focus on the gonad, the female gonad, the ovary. And there are 2 major functions for the ovary. At first, the ovary will produce hormones such as estrogen, inhibin, and progesterone. And the second, the ovary also mature the oocyte or egg after ovulation for maturation, ovulation, and then support the full-term pregnancy. So for the ovary, there are different developmental stages of the follicles. So today I will mention follicles many times. So for here, that is the follicle development process. So actually primordial follicle is the earliest stage of the follicle. For women, they have 1 to 2 million primordial follicle right after birth. So right after puberty, primordial follicle will be gradually activated to the primary stage and then further develop to the secondary, early antral, and antral stage for maturation. And during the follicle maturation process, the follicle will also secrete increasing amount of the estrogen and also inhibin. And then when the estrogen reach a peak, it will stimulate the pituitary gland to release a surge of luteinizing hormone to stimulate the ovulation of this mature follicle. So the hallmark of the ovulation include the rupture of the follicular cell and also the resumption of the oocyte meiosis. So the follicle can release a fertilizable egg for potential fertilization. For example, after the fertilization, the egg will develop to 1 cell, 2 cell, until blastocyst stage of the embryo. And at the same time, the embryo will also transport from the fallopian tube to the uterus for implantation and the placentation. So since the ovary is pretty or highly hormone regulated, it's also potential target of some toxic like compounds such as clinical drugs and also environmental endocrine disrupting chemicals. So why we need to culture the ovary tissue in vitro and also in 3D? So far we know that the gold standard for studying female reproductive toxicology still rely on whole animals. But we also know that the whole animal, they are pretty expensive, time and efforts consuming, and also cost a large number of the animals. So specifically for the ovary, it is also pretty challenging for us to study the dynamic ovary function. For example, the dynamic follicle activation and the development process I just introduced, and also the dynamic hormone secretion, ovulation, and also luteinization process. So on the other hand, some other people, they use individual ovarian cell lines, such as the granulosa cells, the theca cell, or the oocyte for toxicology study or for physiology study. However, these individual cell lines do not faithfully mimic the whole function of the follicle or the ovary, because for the follicle, there's like a lot of bidirectional communications. For example, for a growing follicle, there are 3 major cell types, including the outer layer of the theca cell, inner layer of the granulosa cells, and the center germ cell oocyte. So actually the theca cell will produce androgen, which will be uptaken by the granulosa cells to convert the androgen to the estrogen by the aromatase. And also the granulosa cells and the germ cell oocyte will also talk to each other. For example, the oocyte will secrete GDF9 and BMP15, those TGF-beta family proteins, to drive the granulosa cell differentiation. And on the other hand, the granulosa cell will also provide the amino acid pyruvate to support the oocyte maturation. So this indicates if we only culture 1 cell lines in the ovary, that cannot mimic the whole function of the ovary or the follicles. So in our lab, we primarily use the alginate hydrogel encapsulation method to 3D grow the ovarian follicles in multiple species, including the bovine, human, and also rodent. So if we use mouse follicle as an example, we can use the alginate hydrogel to encapsulate the follicle, and then it can give it a 3D environment. We can culture the mouse follicle for 8 days. The follicle can grow from the multi-layer secondary stage to the early antral stage on day 4, and then to the antral stage on day 8 to reach full maturation. And then we can stimulate the antral follicle or mature follicle with the HCG or luteinizing hormone to stimulate the ovulation. And then the mature follicle can rupture and release a metaphase 2 egg or oocyte. And the remaining somatic cells will differentiate into the luteal cell, which is a process of the luteinization, and form a corpus luteum or CL-like organoid. So if we collect the conditioned media during the culture window, we also see that actually the cultured follicle can secrete a human 28 days, like a menstrual cycle hormone secretion profile. For example, for the figure right here, the red line indicate the estradiol and the black line indicate the progesterone. And we can see the increasing level of the estrogen before ovulation, and then it will greatly decrease after the ovulation induction. And then for the progesterone, it is relatively low before ovulation or during the follicular phase, but the form, the corpus luteum organoid will secrete increasing amount of the progesterone after the ovulation. So this is the hormone secretion profile. And for the ovulated egg, we can also perform in vitro fertilization to fertilize it. And it can develop to the blastocyst stage of the embryo. And if we transfer this embryo back to the pseudo-pregnant mice, the mice can be pregnant and deliver live pups. So this data indicate, at least at the morphological level and the hormone level, we can mimic the most of the key ovary functions. So recently, we also collect those cultured follicles at different time points to perform the single follicle RNA sequencing analysis, because we want to see whether the mini ovary or the ovary organoid we cultured in vitro can mimic the key genes or signal pathways like that can support the ovary function. So for here, I won't go too much in detail, but the overall message is it can, because most of the key ovary signal pathway and the genes, they are well preserved and pretty similar to what we have been found in vivo. Just use one example. For example, the LHCGR, that is a luteinizing hormone receptor. So actually, before ovulation, the induction of the LH receptor can ensure the follicle to be responsive to the LH, and then it can ovulate. So this result indicate that our mini ovary model can mimic key ovary functions, and we can use this for studying the toxicology, biology, and also the ovary disease. So these are our current project in our lab. For example, we use the vitrification method to cryopreserve the individual follicles. In this can be used fertility preservation method for young female cancer patient, and we can also use this to make a high content like a follicle biobank, so we can do the high throughput toxicology study. And we also use our in vitro ovulation model to screen a lot of compound, which is supported by the Gates Foundation. So we can do the drug repurposing and see which can be the a candidate of novel birth control pills. And we can also use this model to study the toxicology and also connect to other reproductive organ or non-reproductive organs to make it more in vivo-like. So today, because of time limitation, I will only introduce our toxicology project. So currently we use this ovary, like a mini ovary model, to screen different classes of the compound, including some clinical or the drug on the market, and also some environmental contaminant, such as the bisphenol A and the bisphenol analogs, and also the flame retardant, PFAS, nano and the microplastic, and also the cyano harmful algal bloom toxins. So today I will just use hepatoxin as an example to let you know how we use this model for the toxicology study. I'm not sure whether you have heard about the cyanobacteria or the harmful algal bloom, but you may have seen some like a picture, or you have, if you go to the lake, you see some green water, or if you go to the beach, you can see some brown or like a blue water. Actually, those blue water are primarily caused by the cyanobacteria. And it's like a great public health concern and also environmental concern. And it's primarily caused by the temperature rise due to the climate change and also the agriculture running off. So the reason we care about the bloom very much just because the cyanobacteria, they can secrete or produce some toxins, which are also called harmful algal bloom toxins. And the microcystin-LR, that is the most common and also potent type of the hepatoxins. So it has been shown that it may cause like a liver toxicity, liver cancer, and it also has like a neurotoxicity. However, we still don't know whether it can be toxic to the ovary in the women or other female mammalian species. So in this study, we use our mini ovary to do the in vitro exposure using different concentration of the microcystin. And our data indicate that, so actually during the follicular phase or before ovulation, microcystin-LR did not affect the overall estradiol secretion and also the follicle survival. However, what we found that after we introduce or induce the in vitro ovulation, the micro-CCLR can dose dependently inhibit the ovulation. And for example, at 10 micromole, like less than 10% of the follicle can ovulate. So that's indicate exposure to micro-CCLR, especially during the bloom season, like through the drinking water or through like if accidental swallow of the water, like when you swim in the bloom like a lake, it can potentially affect women's reproductive health. It may cause like anovulation, infertility, or even some reproductive disease such as the reproductive cancer or the polycystic ovarian syndrome. So for the next part, we also want to know whether we can use this model to study the mechanism. So what has been found is that micro-CCLR is a selective inhibitor of the protein phosphatase 1 and also 2A, PP1 or PP2A. And we also know that actually PP1 is a key mediator to activate the PI3K AKT signaling pathway during the follicle like maturation. So we hypothesize that maybe that is also the mechanism that micro-CCLR can inhibit the follicle maturation so it cannot ovulate. So using this mini ovary model, we do the same exposure, and we also found that micro-CCLR can inhibit the PP1 activity in the follicular cells. And we also see significant downregulation of some key mediator, such as the phosphorylation of the AKT and also the FOXO1. And actually, our real-time PCR data also indicate the micro-CCLR exposure can also significantly downregulate the a target gene of this signal pathway. So for example, I already introduced the RH receptor. You see the micro-CCLR can dramatically decrease the RH CGR expression, and that might be the reason to make the follicle not responsive to the RH surge, and then it can cause the ovulation defect. So for here, you may have a question. So all your results are based on the like a mini ovary or in vitro exposure. So whether you will see some similar or you can validate your data using the in vivo model. Actually, the answer is yes, because we also perform a like an in vivo validation experiment by using the mouse superovulation model. And you see this experiment design, and here is the result. We did consistently see that the micro-CCLR treated mice, they have fewer ovulated eggs in the fallopian tube. And if we collect the ovary for histology, the data also show that we can see a lot of more unruptured or unovulated follicles in the micro-CCLR treated group. And these results are consistent to what we have found using the in vitro or the mini ovary model. So actually, in addition to using the mouse sample or the whole animals, we also collaborate with IVF clinic and also validate our data using the human like primary granulosa cells, and we also see consistent data. So overall, this result indicate that micro-CCLR or the harmful algal bloom toxin, they may have potential effect for women. Like for example, they may cause irregular menstrual cycle, or the like annual ovulation, infertility, or some reproductive disease such as a polycystic ovarian syndrome, because in like abnormal ovulation, that is a key marker of the PCOS. So for the currently, we also collaborate with Dr. Julie Kim at Northwestern University and try to make our model more in vivo-like. For example, we want to connect the ovary with downstream reproductive tract tissue, such as the uterus, so we can make a whole female reproductive tract on chip for studying the physiology and also the reproductive toxicology. And for myself, we also want to co-culture the liver tissue and the ovary tissue so we can introduce the metabolism for our reproductive toxicology screening. So overall, I hope based on this result, I can convince you that the mini ovary model is pretty cool model and also pretty powerful model for us to study the, like for example, fertility preservation, the development of novel birth control pill, and also the study the ovary biology, toxicology, and also study the whole female reproductive toxicology. So at last but not least, I would like to thank all my lab members from Rutgers University and all my collaborators from other university, and also the financial support from like NIH, NSF, and other agencies. Thank you so much.
We have time for one question.
Thanks for the very nice talk. So can you comment on the ovarian tissue concentration of HEB toxin at the serum level that you mentioned? Do we have an idea of that?
Oh, you mean the chemical concentration we use, right? Yes.
And what is observed? So what is your expectation to see in vivo in tissues? What would be like the real exposure?
Yeah, that's a very good question. So for the chemical concentration, we did dose response concentration from 0.1 to 10 micromole. And both from the low dose to the high dose, they are pretty environmentally and human relevant, especially during the bloom season. But if there's no bloom season, for example, in our drinking water, we don't have to worry about that too much. And another concept in toxicology is about dose response relationship. Actually, we observe 90% of the ovulation in reduction, but actually based on the a benchmark dose we get is around 0.1 micromole. That is even more human relevant if for the human exposure in our environment. Yeah, thank you.
Thank you so much. And our last speaker of the session will be Jeremy Wang from MIT.
All right, thank you for being here and for the amazing primers that everyone here in this session have. I'm going to finish this session with talking about modeling immune endometrial crosstalk in MPS systems. And I would like to thank the organizers and for an amazing conference thus far. As we've previously been primed to, the human endometrium, which is responsible for reproduction, is really a complex, hormonally responsive and hyper-regenerative tissue. Now, the endometrium is this layer which actually over the course of 28 days scarlessly regenerates over 1 centimeter. And it rests upon the muscle layer, which is called the myometrium, which is responsible for contraction and for birth. And it's comprised of primarily a luminal glandular epithelium with a bunch of stromal cells, and there's tissue-resident macrophages, NK cells. And these kinds of interactions are of particular interest because they change over the course of the 28-day menstrual cycle. In particular, the stromal cells undergo the decidual process, which prime the tissue to be prepared for the pre-implantation embryo. Now, what happens when these processes are dysregulated? Well, one of a fairly prevalent, but actually publicly not really recognized disease is called endometriosis. And the pathology of this disease is it becomes lesions on ectopic lesions within the peritoneal cavity. And it's thought that the process of the menstrual shedding is dysregulated in this disease such that when there is reflux menstrual tissue that leaks into the cavity, it attaches and forms these lesions. And what's interesting is that 90% of women experience some form of reflux menstrual tissue. However, only 10% globally, well, that's a fairly large percentage, actually develop and maintain these lesions, right? And so the implication is that there is immune clearance deficiency within this disease, right? And it presents itself as these really painful, so there's dysmenorrhea and also contributes to infertility. And as I said, it's also hormonally implicated. And so how in the Griffith lab and in the Laufenberger lab, we're very concerned or interested in properly modeling this disease. So how do we actually model this lesion microenvironment? Especially how do we move into MPS or physiologically relevant systems? Well, if we consider what I was just talking about, maybe we can provide some glandular epithelial organoids, maybe seed stromal cells, and then have these monocytes and macrophages. So it quickly becomes a pretty complex system. So when I entered the lab just shy of a year ago, I was particularly interested in kind of stromal monocyte crosstalk because of what was previously identified as this kind of immune inflammation component, which is dysregulated in disease. So I'm going to go over this this entire disease modeling pipeline with you today. So first we'll start with the hypothesis generation and then go through some of the very key tailored ECM environments that we generate in the lab and how we actually combine these with patient-derived cells, both stromal cells, epithelial cells, and monocytes derived from patients. And then first, place them into static three-dimensional culture. But ultimately, what is a developing story is integrating these systems into MPS devices. And there's just like a feedback loop that ideally through understanding more of these mechanistic drivers behind disease. So let's first begin with the hypothesis generation. Very recently, Lauren Baugh, which is a former postdoc in the lab, did a multi-omic screen of uterine biopsies, both from patients without endometriosis and with endometriosis. So she did single cell RNA sequencing and multiplexed proteomics, and then utilized pathway analysis and also NicheNet to understand ligand receptor interactions. And the upshot of all this is that she found that TYRO3, which is a gene which is implicated within immune clearance response, is significantly downregulated in disease, right? And so this this kind of pointed to a potential immune deficiency in terms of tissue clearance, which I previously described is phenotypically present, but actually molecularly hadn't been characterized up till this point. Similarly, others in the lab, in particular Juan Neco, who just recently got a faculty position at Tufts University, utilized a model that I'm going to describe a little later, where he stimulated co-cultures of stromal cells and epithelial cells with an inflammatory stimulant, IL-1 beta, and induced a diseased phenotype well known within endometriosis, which is progesterone resistance, right? And so this leads to the thought that, okay, it replicates this disease characteristic, but the question when I came to the lab was very curious was, how do stromal monocytes interact, right? Because in a pro-inflammatory environment, monocytes are known to potentially secrete IL-1 beta. So can we gain insight into the mechanisms behind endometriotic disease by modeling these interactions in vitro? So this was the hypothesis, that diseased stromal cells disrupt immune clearance mechanisms by dysregulating monocyte activity in a cycle-dependent fashion. So now I'll quickly talk about how we model these systems, first with the PEG-based ECM. So if we are to look at tissue compartment crosstalk, you need a low noise environment. And typically in a lot of animal generated or animal derived ECMs, it's difficult to actually know or control lot-to-lot variability. So we in the Griffith lab utilize a PEG-based hydrogel, which is based on an 8-arm reactive PEG macromer functionalized with a vinyl sulfone group, where we can add binders such as SYNK-RGD or G-Foger, which mimic collagen and fibronectin, and fibronectin binders or basement membrane binders that replicate collagen-4, laminin, and fibronectin, and generate these hydrogels, which are very reproducible and which are also MMP cleavable. And we can tailor not only the constituents of this gel, but also the stiffness of the gel and tailor it to the environment that we are trying to engineer. So this gel has been utilized in a lot of not only cold cultures, but monocultures. So Alex Brown published a paper demonstrating endothelialization and vascularization, and Alan Kahn had a beautiful talk back on Wednesday that you can look at back in a recording. And Juan Neco recently submitted to bioarchives both the stromal and epithelial cultures. And today I'll talk a little bit more about the monocyte stromals. But why do we even move into three-dimensional gels? So quickly, I'll talk about that. So if we were to look at the decidual response of stromal cells in 2D versus 3D, we actually see that morphologically in 2D, they become very flat. In 3D, they're a little bit more biomimetic, but and a characterization of that is that the secretion of prolactin is significantly increased in three dimensions versus 2D culture, right? And this is work done by a talented undergrad called Kira Butley at MIT. So now I'll talk about how we combine the stromal cells and monocytes and what kind of insights that we gain into endometriotic disease. So in our co-culture model, we derive the stromal cells from endometrial biopsies from our collaborators at Newton Wellesley Hospital. And we gather CD14 positive monocytes from our collaborators at MGH, and we combine them. So we pre-decidualize some of these cells, and we co-encapsulate them to replicate the 3 phases of the menstrual cycle. So the non-differentiated cells would be co-encapsulated with monocytes in this proliferative phase model. And then in the secretory phase, we pre-decidualize these stromal cells, co-encapsulate. And then in the Menzies model, we have the pre-decidualized stromal cells and then withdraw the addition of MPA, which is a synthetic progestin that Linda just talked about that we use. And in these droplets, we gather them up and we're able to not only culture them, but for a period of 7 days, but do a bunch of different experimental outputs, such as look at time course images, flow sort and sequence the individual monocytes and stromal populations, and also do some downstream analyte detection. So I'll talk primarily about these two first. One of the results that we found is that monocyte proliferation in our co-cultures is blunt in disease. So what we do is that we take a composite image of the stromal cells and CD14 positive monocytes that are encapsulated. And at day 7 of culture, we also do an EDU proliferative stain. And if we isolate just the CD14 positive monocytes, and we can overlay and use cell profiler to quantify the amount of proliferation that the monocytes that we observe in culture. But in disease, it is significantly blunted, right? So that's one metric. But what about other molecular characterizations? So what we did is that we can also dissolve these gels, these co-cultures, and then separate out using flow sort the stromal and monocyte populations. So I'm going to tell you briefly about the monocyte story. So if we were to take volcano plots of the differential gene expression analysis and compare the 2 different cycle between proliferative and secretory, you can see that in this control group, the monocytes, there isn't a significant difference in the amount of differentially expressed genes. But in disease, there are a lot of genes implicated. And so when you do gene ontology analysis, what you find is that terms related to cytokine production and pro-inflammatory factors such as TNF-alpha or IL-6 are enriched in these gene ontology analysis. So that's interesting. But then the question is, because this is a co-culture model, what are the ligands that the stromal cells potentially are secreting that are pushing monocytes to become dysregulated? So we took a reference data set from our collaborators at the Reagan Institute, Brian Bryson, and they did a very interesting study where they took these CD14 positive monocytes and they stimulated with 60 single conditions, right? So CSF2 or IL-17, IL-7. And what we did is we took our sequenced monocyte data and we plotted it on. So this PCA demonstrates that, okay, the secretory and control co-cultured monocytes don't seem to at least on this PCA plot, be very different. But in proliferative and menses, you see this kind of drift. Now, this is kind of a dimensionality reduction technique. So I was curious about, can we infer what kind of monocytes these are, these kinds of co-cultured disease monocytes are becoming? So we created gene sets from these reference data set. For example, if I would take differentially expressed genes that are upregulated or downregulated between IL-17 and a control, you would take this and make a gene set, and you can actually run single sample GSEA, gene set enrichment analysis, to compare it to our modeled monocytes. And what this demonstrated was something very interesting, that pro-inflammatory, that we believe that these kind of monocytes that were co-cultured with diseased stromal cells became more pro-inflammatory, okay? So in both kind of the menses and the secretory phase compared to controls. So there is a level of dysregulation there. We also found that in our stromal cell sequencing data, we recapitulated what was observed in vivo, that TYRO3 was actually downregulated, but interestingly, in only the proliferative and the menses model. So there is some kind of cell cycle dependency observed. This is kind of corroborated with what we observed in the monocyte, this kind of drift in both the proliferative and the menses data sets, right? So it points to this dysregulation, but that is cell cycle dependent. And finally, we did a little fancy footwork in terms of trying to understand what are the ligand receptor interactions and how is it disrupted in disease. So we took genes which are from a reference database of cellular communication called OmniPathDB, and we took our data sets. For example, we took genes which are stromal ligands and we crossed them with monocyte receptors. And this PCA plot shows that this actually separates in the blue, for example, secretory model separates very fairly distinctly between the menses model and also the proliferative model. But in disease, this kind of relationship is significantly disrupted, right? And so the menstrual cycle conditions don't clearly distinguish these ligand receptor pairs. So we're still ongoing analyzing the implications of this. But for example, some pairs which are implicated are prostaglandins or TNF receptors as well. And some of the ongoing work is to understand. We also took we saved the media, but it's really difficult to distinguish co-culture media. So that's ongoing work as well. But now a little bit more about how we're trying to integrate this into MPS systems. And so in order to recapitulate these cycling hormones, in our model, we actually just had a static addition of estrogen and MPA. However, we really want to move into systems in which we can recapitulate not only the cycling hormones, but control oxygen levels and different additives. To do this, so in that more complex platform, we are currently recapitulating this into a static chip testing platform. So this kind of COC material, which is a plastic, thermoplastic, we also enlarged it so we can do some pre-testing, and I'll show you a little bit of that story right now. So there is a comparable decidual response in this static chip. So if we compare these droplets that I've just talked about to this chip, and we attempt to decidualize stromal cells in it, there is a comparable prolactin secretion over time. And we also demonstrated very recently that we can co-encapsulate stromal cells and monocytes and culture them over a long period of time. So, and in the future, we hope to be able to integrate this kind of stromal monocyte encapsulation with endothelial perfusable networks. And so this kind of is the hope, is that we can potentially push these systems into this kind of multi-compartment integration to understand more to understand the mechanisms behind endometriotic disease. And yeah, this is the the overall arching pipeline. And I'd like to thank all the members of the Griffith Lab and of the Laufenberger Lab and all our collaborators. And thank you for your attention. And please go to the posters of Elise Gubbins and Aaron Tavonian and Priyatana Roy in the poster session after this. And thank you for your attention.
Yeah, we have time for one question. Here comes Chris.
Hi, thanks very much. I'm interested in these stromal populations that you're putting into a droplet assay. Do they maintain the same phenotype throughout the 7 days, or do they kind of drift once, or do you keep in the same hormone regimen that they were in at the time they were harvested?
Yeah, so if in the proliferative condition, they were on DMMF12, 5% heat inactivated FBS and estrogen. So they are, but they were first cultured in 2 dimensions. And then in the co-cultures, they are in 3 dimensions. We haven't done, for example, time series RNA-seq to actually understand if in these encapsulations, they do drift, for example. So, but that's a good question for sure. Yeah, but yeah. In the secretory, the plus MPA, they do decidualize, but in the we haven't done a temporal characterization of only estrogen only. Yeah.
Great. Thank you so much, everybody. 2 quick announcements for you. One is that we'll be moving across the hall for a poster session. And also for the sunset reception afterward, there will be 3 shuttles, 7:45, 8, and 8:15. So it's also just a 10-minute walk. So please be ready for that. Thank you very much for being here.